in very real time what the community is doing and might like to do with ASAXS, and just share ideas throughout the day. We have more than 20 presentations, and there's about 10 people joining us virtually, so the crowd is about twice as big as it appears to be here, so that's good. Let's go through a few introductory slides here about the facility that we're talking about here. There we go. So as you no doubt know, since many of you, if not all, are users of the APS is dark now, while the entire optics of the ring are being changed. But as it says, we're here on the 5th floor of the main APS building, and the sector that we're talking about is almost diametrically across the storage ring at sector 15. The current layout in the hutches and things that we have had is displayed on the right-hand side of this. But we're making major changes to this, as I'll talk about in a minute. But ASAXS has only been around for a little over 5 years. It was funded in the last renewal of the NSF grant that supports ChemMatCARS. And you know, for a little bit of other background, you should realize that there are 60-some experimental stations at the APS. Half are operated by the APS, half are operated by collaborative access teams, and ChemMatCARS is one of those collaborative access teams. So it's actually operated by the University of Chicago with full support from the National Science Foundation. So we always refer to this as the National Science Foundation's ChemMatCARS. That's what this facility is. The ASAXS part, as I said, was funded in the last renewal in 2017 when we got it started, and the users have been growing. As I'll mention, so far ASAXS has only been allocated 15% of the time, but that's going up. So there'll be expanded capacity and availability. That's what the instrument looks like, but Myrna will tell you more about that. We operate through the APS proposal system, so we don't have a separate selection process for ASAXS users. This delay is somewhat. There we go. The big improvement is shown here. In the current X-ray beam before the APS upgrade, you get a narrow slit of X-rays impinging on your sample, but the focusing optics will change so that you now get a spot, as is illustrated there, and that will be 500 times brighter, more coherent, and more physically localized. And in addition, the NSF supported the construction of an additional beamline here. So that's what's going on during the upgrade. There's a lot of work going on at ChemMatCARS that involves all of the instruments that are there, which include not only ASAXS, but crystallography and liquid surface scattering. As I said, our beamline scientists and staff are working hard during this time. So the new beamline will increase the ASAXS fraction of time by a factor of 2, from 15% to 30%. And we believe, and that's what we're exploring here today, that this will allow exploration of many new possibilities in chemistry, material science, biology, and so on. And we really want to bring this to a broader research community. And we're in the midst of writing our renewal proposal, the NSF proposal for ChemMatCARS. So we're really surveying what the community wants and understanding what the community's interest is. during this time for the 2024-29 cycle. So that's what you're doing here today. I don't think I'll review the agenda in detail. It's in your packet that you will have picked up. Perhaps the most important thing to say is that we want you to give a 10-minute presentation in your 15-minute time slot, so that there's a little bit of time to talk about things. We have, like I said, 10 or more people online, so we'd certainly like to have discussion, but if you want to say something, please position yourself near a microphone and turn it on so that everybody can hear what you have to say. As you gathered, the event is being recorded, so don't say anything you don't want anybody to know about for posterity. And keep the microphones off when we don't need them. For those of you online, if you want to ask questions, please put them in the chat and we will get to them as much as we possibly can after the talks. If you have any questions about travel reimbursement, those can be directed to Natalie Chen, who I think has contacted you, but you can certainly talk to Mirnal or me. Anybody have any questions right at the beginning here? Good. Well, why don't we unshare now and turn the floor over to our NSF program director, John Papanikolas from chemistry at the National Science Foundation, to say a few words. I should say that John has been very supportive of what we've been doing, and you know, together with us, brainstormed about the role that this workshop could play in generating a strong renewal proposal for ChemMatCARS. So, John, please go ahead.
Thanks, Matt. Let me just introduce myself. My name is John Papanikolas. I'm a program director at the National Science Foundation. I actually spent much of my career at the University of North Carolina at Chapel Hill, where I was a faculty member for 25 years or so. And then sometime around like 2017, I came to NSF as a rotator, and I enjoyed it so much that I decided to stay. And as a rotator and eventually as a full-time program director, you know, I've been involved in lots of different things. I'm currently the lead of the Chemical Structure Dynamics and Mechanisms program in chemistry, which is essentially the experimental physical chemistry program. I've worked in the MSN, or macromolecular nanochemistry, supermolecular chemistry program. And other things as well. And one of the things that I do is sort of handle kind of lead the effort in the chemistry division on its facilities, and of which ChemMatCARS is one. And I took over ChemMatCARS, as I think all of you know better than I, has this very, very long history. And the most recent program, or the program director before me, Carlos, when he passed away a couple of years ago, I took over the ChemMatCARS, or the management of ChemMatCARS. I want to say, you know, like to Matt and Mark and Benoit, I hope you all are there. Thank you so much for your patience with me and like the things that I ask. And I got to say that the leadership of this facility is incredibly responsive, and I've appreciated that quite a bit. And I also enjoyed the site visit last summer. Actually being able to come to the site and see what it looks like and how big a hutch actually is. When I heard hutch initially, I was thinking like a rabbit hutch or something, but you know, it's these ginormous things. So so I learned a lot there, and I see this as sort of a continual sort of learning process. So when Matt asked me to say a few words, I really had no idea what I was supposed to do. This is not the type of thing I've ever done before, and you know, and so so I thought about a few things, and so I have a couple of slides that I think I'll show, and then it's not going to take too long, and then I'll certainly, you know, if you guys have questions for me, I'd be happy to sort of address them in any way that I can. So what I wanted to say about chemistry and about, you know, and about facilities and ChemMatCARS is, first I thought I'd start with like kind of how the chemistry division supports research, or different modes of research that chemistry has. And so some of these are pretty obvious, right? We support individuals and small teams. These are through our disciplinary research programs. I think many of you, probably maybe even all of you, have an individual investigator award through the chemistry division. These awards support research, and like I said, typically they're you know, maybe individuals or small collaborations. The division also supports big collaborations, and this would come in the form of centers like the CCIs and things like this. And these are large-scale investments that the division makes, and they are also there to do research. Okay. And here what you do is you have, like, you know, a larger number of people. You're focusing a significant number of resources on that group of people to solve, you know, specific problems, you know, related to sustainability or polymers or whatever it is. And the point I want to make here is that centers are, it's sort of like an inward focus, right? The focus of the money, the resources are onto a group of people. And if you're in that center, it's awesome, right? You have lots of collaborations, you have lots of resources, lots of things, you know, happening. And but if you're on the outside of that center, it's still great for the community, but you're kind of more on the outside looking in. The other type of investment that the chemistry division has are facilities, and facilities are kind of the opposite. I see them as sort of the inversion of a center. Facilities, you provide resources to a small group of people that then turn around and use that money to provide state-of-the-art tools that enable science for a broad user base across multiple fields. At NSF, we call this infrastructure. I think that's synonymous with facilities, you know, the word facility and so on, but it's an operation, it's an investment that benefits a large number of people, you know, by a small group, in this case, the ChemMatCARS team, creating these tools and making them available so that users can come and use those tools and instruments. And so for a facility, the interaction with the user community is key, right? It's really important that the facility is constantly, and I think ChemMatCARS has demonstrated this over the years, that is constantly sort of evolving and upgrading and moving to provide better and better resources to that community. And in order to figure out what to do, or what to, you know, where the best investments are, they need to have that interaction with the community. And these workshops that ChemMatCARS is holding, I think are sort of one way or a key way in which ChemMatCARS is interacting with that community and getting the feedback from the community as to what to do, where to go, and where the big, you know, where the big advancements can be made in this particular area. I encourage everyone that's on this call to, like, give their feedback to Matt and Mark and the others. They want to hear it. They want to know what it is, and it's going to be integral to their planning for going forward, not just into the next renewal, but, you know, as they go forward on sort of a longer term. So Matt, I'm glad you're doing this. It's great to see, you know, such a large number of people participating, and I hope I hope you get kind of the feedback that you really need. I have a couple more slides here. So more broadly, if we look at facilities within the chemistry division, the chemistry is new to this game. Okay, so, like, you know, if you think about facilities at NSF, large, you know, there's a whole organization called the Large Facilities Office, and those facilities are things for, like, they run telescopes, the South Pole, big boats, you know, and all these sorts of things. And there are communities, like astronomy is one, where, you know, the community is really led, their community-led priorities that govern what type of telescope they're going to build or what their energy range they're going to look at, and so on. Chemistry as a community doesn't really have a history of doing this. ChemMatCARS has been around for a very long time, and it's and I think it's a model example of how a facility can impact a large number of people. But the chemistry community doesn't really have a history of doing this, and chemistry, the division, at the moment, we only really have 2 facilities in our portfolio. One is ChemMatCARS. It's, like I said, it's been around for 20 plus years of operation. It has this long history of serving many people. There's another facility that a mid-scale facility that chemistry is starting. It's in the National Extreme Ultrafast Science Facility, or Nexus. It's located at Ohio State, and it's also using or creating X-rays to do experimental science. Although its focus is more on time resolved, ultra-fast, attosecond, femtosecond sort of dynamics. It's currently under construction. It should be open for business kind of in the fall of 2024. But at the moment, these are these are kind of the 2 facilities that the chemistry division is sort of are in our portfolio. And if I step back and look at facilities and what should the chemistry facility portfolio look like, right now it looks like it involves a lot of X-rays. And there are, but we could be thinking that maybe there are other types of facilities that are out there. And NSF, and this is sort of, I'm going to take 2 seconds of your time here, Matt, and sort of kind of put this out there in kind of the ether. NSF has this program in what they call mid-scale infrastructure and facilities. And mid-scale is sort of, there's it's defined by a dollar range. So down at the low end, you have MRI. So these are, many people I'm sure here have written MRI proposals. These are projects up to like $4 or $5 million. And then at the high end, they have what they call major research equipment and facilities construction. And these are things that are above 100 million. So you're thinking like, I don't know, the South Pole Station or something like that, right? So these really big facilities where you got like, you got small things like NMRs, mass specs, and so on. There's this gap between about $5 million and $20 million, which is called mid-scale. And mid-scale is they run, they divide it up into 2. There's MSRI1, which is projects below 20 million, and MSRI2, which is projects above 20 million, 20 to 100 million. And they've had, in this gap prior to 2019, these were all supported by individual divisions. In 2019, 2021, and now this year in 23, they've run 3 competitions. We're in the middle, we're in the midst of sort of the 3rd competition for MSRI1 and MSRI2. And I've listed sort of the document numbers here so you can go and find the solicitations. For teams to submit proposals to develop facilities, okay? And what's going to happen after next year and beyond, that's like, who knows? We don't know what's going to happen until they sort of issue a solicitation, and when they issue a solicitation, then everyone knows. So at the moment, there's just sort of the current the current solicitations out there. What I'm saying is, if you're out there and you're thinking, you have an idea for a facility, an idea for some piece of type of infrastructure in this price range that would benefit, that you think would benefit the chemistry community, then talk to me and let me know. There might be a way to sort of help you get started, sort of building that community support that you'll need, that'll be so integral to sort of getting a facility off the ground. But certainly chemistry is looking to broaden its portfolio and add to its portfolio of facilities. Anyway, I'm going to stop there and say just, once again, thanks for doing this. Thanks for your patience, Matt, with me over the last couple of years, and I'm happy to answer any questions that you might have.
Thank you very much, John. Thank you for joining us. Are there any questions for John? I'm not seeing any right now, John. So
okay. All right. Well, have a good meeting, and I'm going to be popping sort of in and out. I have to go to my normal staff meeting, which is right now too. So I'll probably come back maybe later in the day.
We're here until late in the afternoon. So
Yeah, right. All right. Good luck. Bye-bye.
okay, so we're ready to get underway and turn the floor over to Myrna. We're 10 minutes ahead of schedule, so and there are 20 talks, so everybody can talk for 10 minutes and 30 seconds. So
okay, thank you all for coming, and thank you all who are joining us virtually. So I'm Rinal Bera. So I'm the beamline scientist responsible for the development and user operations at NSF ChemMatCARS as far as the ASAXS part is concerned. And ASAXS, as you can see, is basically, if you just take the full name, it's Anomalous Small Angle X-ray Scattering. So now I will be talking about some of the developments and some of the research highlights which we are doing from the research at NSF ChemMatCARS. So to begin with, why we should do any ASAXS measurements at all. So the reason is it gives us an opportunity to look at the structure in an element specific way. For instance, there are several problems in chemistry and material science which deals with ions, element specific structure in the terms of like if you have a multi-element nanoparticle like this, how the nanoparticles are distributed within how the atoms are distributed within the nanoparticles decides how the nanoparticle will perform or provide applications. Again, in terms of biological materials, it's the ion distribution surrounding them which kind of decides how they will interact and how they will perform in a biological system. Similarly, some new experiments are coming up which deals with this kind of molecular rulers, where they tag some kind of nanoparticles in a biological system to measure the distance or sizes of those systems. And there also ASAXS can be very useful. Another particular system where ASAXS can be very useful, and some of the users which we are dealing with are using this, is to look at the supramolecular structure. So there are metals there in the system which can play a big role in their assembly, and how they are kind of arranged there is going to decide how the system is going to perform in terms of application. So that's where also, so I'm kind of giving you some brief overview, and over the course of talk today, we'll hear more examples where we'll see how ASAXS can be more useful in terms of a very strong tool. Said that, now how we do ASAXS. The question is how the ASAXS works. So basically ASAXS works on a principle that how X-ray interact with matter. So each and every atom in the periodic table has a unique fingerprint in terms of how X-ray interacts with it. So as we know, X-ray interacts with electrons. So in an atom, electrons are they're arranged in a shell. So depending upon how X-ray interacts with those kind of shells, electrons in the shells, they have a particular response in terms of scattering, and that is given by this complex function. And as you can see, I'm just showing you an example of the gold edges, gold L3 edge and silver edge. And you can see near the edge, they have some peculiar function. And that's why this particular technique is called anomalous, because it gives you some behavior which is different from the normal operation. So here, as you can see, this is a function of energy. So at least in order to get this thing done or get the ASAXS experiments done, you have to do experiments in terms of energies. And I'm showing you an example here to explain. So here, if you just consider a core shell nanoparticles and you want to see how the elements are distributed within this, so what you do is you perform ASAXS measurements as a function of energies, which is very close to, for instance, in this case, close to the gold and silver edge. And as you can see this from this plot itself, that at the gold edge, you will see this drastic difference between the silver and the gold scattering contrast. And at the same time, when you do it at silver edge, you will see the same. So by combining these two energy dependent ASAXS measurements, you can get ASAXS data, which will be, as you can see, are very close to each other, but still you will see a lot of differences in the data. So in short, what you are dealing with is X-ray scattering signal, which are very close to each other, but still you find some differences. And by proper use of data analysis, you can extract the information which will tell you how the elements are distributed within nanomaterials. So said that, what we are doing at ChemMatCARS is providing you a very broad, experimental infrastructure, where we are providing a X-ray source which can go from energy 5.5 to 70 keV. So it's a broad energy, spectrum which we are providing. And with that spectrum, what we can deal with is we can look at the elements, which is shown in this periodic table, from Z equal to 25, Z greater than 25. So most of all the high Z elements we can look at by using our facility. And on top of that, we have an undulator source, which gives you a high X-ray flux. So we can do experiments pretty fast. And also we, at the same time, the undulator source is kind of energy tunable. So that gives us the tunability. And on top of that, what we have so far as an experimental section is ASAXS composed of, the whole instrument composed of monochromator. So there is an optics part of it, which gives us the energy and energy resolution. And then we have a focusing mirror, which goes through this. And then ultimately it comes to the experimental station. So in an experimental station, as we are looking at small angle X-ray scattering, so we have a big vacuum tube between the sample and the X-ray source. And your sample is somehow here. We should put the sample here, and this is corresponding to the position here in the image. And then we have this long tube, and this long tube is basically to avoid the air scattering which is coming out of. If you have no tube and put the detector right away there will be a lot of air scattering. So you want to avoid that. That's why this long tube is needed. And on top of that, in order to get accurate, because I told you that the ASAXS measurements need to be done in a way that the signals are very close to each other. So we have to detect signals which are not very much different. So we need an accurate measurements in that sense. And for that, what we have employed is a very optimized photodiode setup. And on top of that, there is a diamond, BPM, that means the beam positioning monitor, which tracks the beam as we change the energy. So we can have a feedback mechanism which keeps the beam focused at a particular position. So those two are very much needed for having a accurate setup to measure ASAXS measurements. On top of that, what we have is not only SAXS, but now we have like 3 different setups by which we can change the sample to detector distance, and we can have like discrete Q ranges. So at this point in the facility, we can do it like 3 different ways to get to the different Q ranges. So I will come into later about the development part of this. But at this point, as you can see, we have a discrete Q range. So overall, we have a well-tunable X-ray source, which can give us a broad energy range, which will be useful for getting to a lot of high Z elements, as you can see from this. And then we have a accurate measurement system, which is sensitive to this kind of small signals. And then we have a discrete Q, Q range, which comes from this setup. Okay, now coming to an important part of this program, because ASAXS is not a new thing in the scientific community. ASAXS has been done for like 30-40 years so far. So the first experiment was done in 1980s, but it is not in the normal user realm, because it is a very specific technique and it needs a lot of analysis and expertise in analysis. So far the experiments are done by most of the expert user groups, which are kind of like 4 or 5 in the world so far. But what we wanted to do when we took this ASAXS is to make it as general as possible. And the major hurdle in that sense is experimental capabilities for sure, but on top of that, the infrastructure to do that data analysis. So we took an initiative to develop a software program or software infrastructure, where we can not only do the experiment very easily, but also to do the data analysis and data reduction in a way that with few simple clicks you can do that. So in order to bridging the gap between the ASAXS experiments and to get the results, we have developed series of software tools, and those are already available in GitHub, so anybody can download and use that. And on top of that, when we were developing, we were interacting with users to see what kind of functions they need to address their particular problem. And there are already functions developed. Any user who is using that particular system, if they are very close to each other, they can use that directly, or they can collaborate with us, or they can write their own software, because it is written in a way that if a person is good in Python, they can write it themselves, the function to analyze their data. So this is one of the big initiative which we took, and I think for the last 5 years, as we are operating, we are seeing some advantages of this directly. Okay, now I come to one of the examples which will show you how powerful it can be. So this is research which is done in Matt's group. So in his group, one of the postdocs was interested in looking at the element, the dopant distribution within a upconverting nanoparticle. So upconverting nanoparticles are necessarily a core shell type of nanoparticles, which has this kind of rare earth dopants within it, and it is the distribution of this rare earth dopant which determines the efficiency of this conversion, to convert it from near infrared region light to a visible one. And it has a lot of broader application in terms of in biology as well as in material science. And the way they have designed the experiment is to look at the dopant distribution as they grow the particle, because as it has to be a multi-layered particle, so it has to be done in several stages. So they have done it with to create a seed first, and then they put different kind of shells with different dopant concentration they know. And in general, what is used in this community is TEM and EDX measurements to show how the dopant distribution is, but it is not that accurate, first of all, and second of all, it is some sort of qualitative way to look at it. And another thing is it is averaged over depth, so it doesn't give you a 3D distribution of the atoms or of the elements you want to look at. So we did ASAXS on this system to show that we can not only look at the morphology of the particles, the particle size distribution, but also we can look at the distribution of the dopants, which is in this case the ytterbium. And as you can see, as it grows, it has some kind of monotonous distribution happening within the non-monotonous distribution of ytterbium atom happening in this nanoparticles. So this gives us, in that sense, I want to tell you that ASAXS is a very powerful tool which can give you not only the size distribution or those kind of things, but also the element distribution, which is really crucial for applications like this. Okay, so in terms of user support, so far we are kind of discussing with our users to really, so whenever we do experiments, we discuss with our users. Either we use the setup which we have built for other users, or we discuss with them to build some custom made things for all the experiments which we do. So as you can see, most of them are custom built. Only thing we recently purchased is this Linkum sample stage, which is for temperature control. But other than that, we kind of make all our sample stages by ourselves, because we have some particular need in terms of sample thickness and the area of the sample which we need to probe. So that's why we always interact with our users to get it. And on top of that, we provide some ready-made tools as well. And as I mentioned, software is one of the big things in our facility. So we also provide help in terms of data analysis, and at the end, when we write manuscript, we provide support there too. So coming back to the upgrade part of ASAXS. So as I mentioned, we have a system where we can have discrete Q ranges, as you can see from here, and as I mentioned it in detail in the last few slides. So we have a discrete Q range, and we want to move away from this discrete Q range to a continuous one, where we can change the sample to detector distance within the bigger vacuum tube, and that will give us a simultaneous SAXS setup, as well as the Q range which goes from like 0.003 to 6 angstrom inverse. So we are working on this, and probably it will be available just after the upgrade of the APS. So we are doing it in sync with the APS upgrade. So next thing is the development in terms of sample environment. So ASAXS is giving us, after the upgrade, a very small beam, right? So we want to use this. It's not only small, it has a coherence which is higher, and also the intensity will be 500 times higher, as Matt mentioned. So we want to use all this property to discuss something or develop something new. And as you have seen, the ASAXS so far is done as a function of, like, in an ASAXS measurements, what we do is we measure ASAXS data as a function of energy. So it takes time. For instance, for getting a particular data, you get at least 50, you need at least 15 or 20 minutes of data collection just to get the full energy range to get the information done. So you cannot do time resolved studies with ASAXS. So what we are planning to do now is combining microfluidics with ASAXS. It has been done with SAXS to show time resolved capabilities. So we want to do the same thing. So the idea is very simple. So in a microfluidic setup, we have the channel. We have the channel through which the analytes are moved, or the sample moves around. And depending upon the reaction which is happening here at the entry of the channel, then the time is basically proportional to the length of this channel. So basically, when we measure these structure at a different point in the capillary tube or the channel here, you get a time evolution of your system, and we can do energy dependence study as well at different points. So which gives us ASAXS. So in that sense, we can do time resolved ASAXS in this setup. Another thing which we are trying to do is, so far we have done bulk measurements. So we haven't done anything near the interface. So by doing the same experiment in the grazing incidence geometry will give us an opportunity to look at grazing incidence anomalous SAXS. So which are also trying to incorporate in our facility. So in conclusion, ASAXS facility provides a very powerful structural characterization tool, which will be useful for soft matter research in chemistry, materials, and biology. So we have developed both hardware and software in a way that it can be used by general users who are not experts in ASAXS per se. And with the upcoming APS upgrade and the planned upgrade, which we are trying to do, which we are kind of plan to do in this upgrade time, is going to give us new techniques to do time resolved ASAXS and grazing incidence ASAXS. And then I come to the acknowledgements. So I acknowledge most of all the users which we are collaborating with to do the experiments here, and then our ChemMatCARS PI and our ChemMatCARS staff, and of course the funding which we get from NSF. And yeah, I'd like to have some questions if you have any.
So we certainly have time for questions. Mirno is the guy who runs this place.
Yeah, we have more than 5 minutes.
Anything from our online participants?
Can I maybe then ask a question?
Yes, sure.
So this was great, thank you. I was just curious about, you showed a couple of little sort of purchased setups that you got, and how do you go about sort of getting different setups? So I would imagine folks, at least people in my group, would be interested in being able to stretch the material while you're doing it and things like that, right? How does that work? Is it you talk to the students or you talk to the researchers and say, wouldn't it be great to have this? And maybe you can talk just a little bit about how these different setups for the sample.
Certainly what happened with one of your students who came for experiments.
Mm-hmm.
So they bring this kind of stretching mechanism that manually they can stretch. So they wanted to see if it is possible to load it in our system, and we did it. Like we have a technician who is kind of making these things very quickly for us. So we did it within few hours to load it on the system, and we collected data like for the first time when they brought it. And later on, it became kind of, we did like 2-3 times experiment. Later on, it became very, very routine there.
So you have an infrastructure here to help you maybe
Yes.
construct things. I was just trying to get a better feel because the students came back with all this cool data. And I thought, oh, you could do that.
Yeah,
Right. So I
I
was just
think
kinda
it will
curious.
be better if we discuss it ahead than it will.
Sure, of course.
Done it earlier, but
Right,
during
right.
the beam time, if it is necessary, we can do that as well. Yeah.
Thanks.
I can also have a question.
Yes.
So when you say in the future you want to do time resolved measurements, what kind of temporal resolution are we looking at?
Yeah, it depends on the whole field of microfluidics. So depending upon the microfluidic setup we are building, so that can be custom made to get to the time, time resolution which we want. So there are two issues here. One is the signal to noise ratio. So we have to look at that. And on top of that, the scattering material, what you're looking at. If it is a high Z material, probably you can go to much lower time scale. But if it is a low Z material, then probably the contrast will be not that good. Then the time resolve will suffer. But so far, what I have seen, so ASAXS can do millisecond so far I have seen, but we haven't done ASAXS on that. So this is something which we have to explore.
And it's likely to be better with the upgrade in APS.
Yeah. Huh? Okay.
There's a hundred.
Oh, hand raised, okay. Can you ask the question, please? I don't see it here though.
Hi, Renal. Shall I ask a question?
Yeah, sure. Go ahead.
Hi, thanks very much for a great talk. It's very, very informative and much appreciated. I mean, I think you kind of answered the question, and thanks to Matt also. And you kind of answered my question with the slide that you showed of Matt's data and the ytterbium. But in your slide 3, you were talking about core shell gold, silver nanoparticles. And so I was just wondering what kind of a sense you have in terms of the range of sizes, also what optimum sizes for those kinds of colloids you think that ASAXS would be useful for, in terms of thicknesses of the shell, if there's a minimum or maximum kind of thickness.
Yes. So the thing is, if I just talk about ASAXS, because we are talking it loosely in terms of, just ASAXS, but ASAXS can be AVAX as well, right? Means we can do the same in a wide angle X-ray scattering setup, we can go to much lower size. So far what we have seen is we can, with the current setup, we can go up to, let's say, few angstroms even. Does that answer? Do you want the lower limit or the higher limit?
So, I mean, pretty tight resolution, basically, pretty small thicknesses is quite obtainable.
Yes. So lower limit is, as I can say, like few angstroms, and then the higher limit can be like the setup. What we have currently is it may go up to like 150 nanometer, something of that order.
Great, thanks.
Anything else? Otherwise, why don't we get Ganbo up there? And we're almost exactly back on schedule now.
Thank you, Matt. Thank you, Marino. Okay, so I will talk about a little bit about my research. So we are asking this question some years ago. That is, we are all familiar with simple ions and colloids. We know that different theories, stable versus unstable, solution versus suspension. So the question we were asking is that what happens if we make simple ions larger and larger in size, or we cut the colloid particles smaller and smaller? There are still two different things. But when their sizes are meeting with each other, what happens in the middle? Now we know the answer in the middle when we ask the question, what happens when the soluble ions reach the size of nanoscale? Actually, they have their own behaviors. We call them macro ions. So these kind of nanoscale, highly soluble ions, we have many model systems from inorganic ones to organic ones to bios. They all follow the same trend. So what we learned here is that those microions, they have their own behavior. So in these kind of solutions, microions charged, counterions, coions. Coions means the small ions carrying the same type of charge. And in water, of course, in polar solvent, everything is hydrated. That's it. So very simple system, but they have their own behavior. So first thing we start with the behavior is that counterion association. Those kind of microions are bigger than the, microions are bigger than counterions, but not significantly bigger. So therefore, counterions are loosely associated, not condensated onto the surface of the microions. So this is the first point that ASAXS can help us a lot. So when microions are in solution, this kind of loose counterion association leads to a counterion-mediated attraction. People in polyelectrolyte field probably know that for a long time. So we continue this attraction. They carry the same type of charge, but attract with each other in dilute solution. They start to form monolayers. When the layer is large enough, they start to close and bend. So that is the electrostatic interaction controlled self-assembly, leading to the structure. We call that BlackBerry type of structure. So this is a general self-assembly behavior favored by the nanoscale charged large ions. Okay, so we can always use different metal counterions. Stronger counterions making the distance closer between the microions and the bending more and more difficult. Then the consequence is that larger and larger self-assembled structures. So overall, this is a picture representing our effort of quite a few years. So what ASAXS can help us is that at early time, we noticed that our models, we can always pick up the well-defined a few nanometer size microions. They are molecules, they are all the same. So therefore, on top of that, they have all the advantage, containing a lot of metals with counterions. Counterions are also metals. So even in dilute solution, we can easily have beautiful diffraction patterns. If there's any counterion association, these patterns will change, and that is a part ASAXS can contribute greatly. So this is a very early work. We proved the first point that counterion association indeed is there, and once they are there, they lead to the self-assemble process. Okay, so that was achieved here at Argonne 14 years ago by Mark Antonio. Very exciting recent progress is achieved again at this beamline, and Marino helped us a lot. So right now, what we can get is that we can accurately determine the number of associated counterions and their distance to the microion. So these two informations are really important to us, number and the distance. So we use the 2.9 nanometer cluster, spherical. Okay, so after the analysis details, please ask Marino, he's the expert. We can calculate, okay, when we use different counterions. One is monovalent cesium, one is divalent strontium. They have different distributions. Okay, if we just ignore all the experimental details, we tell the story is that, look at this, rubidium, strontium, even rubidium is 1+, strontium is 2+. We see that rubidium ions can be closer to the central cluster. Okay, so that explains the phenomena that people knew a long time ago, and expert was May. She's there. So that is, see, 1+, 2+. But the solubility of this cluster is higher when we use the 2+ strontium. Why? We try to explain that is that because strontiums, actually they stay further away from the cluster, according to ASAXS. Okay, so that because why is that? Because of the merge of the hydration layer as one of the reasons. Okay, so that is what we can do. And we can continue. That is the next step we're going to do is that we know different ions will behave differently with this kind of interaction. And previously they're called a Hofmeister series. So we can ask question, central microion, how smart they are. Can they accurately distinguish different counterions? And if several of them are together, can they pick up the most preferred ones? We know they can do that, but let's prove it by ASAXS. Okay, then let's continue. So our science started there, self-assembly, bending, BlackBerry structure. We can always ask this question, what happens if the contrast, the metals are really strong, making the bending very difficult. They're close to each other. So then the 2D sheets will stay as 2D sheets in dilute solution. And that leads to another phenomena, that is phase transition or gelation. So remember, this is a simple dilute aqueous solution of inorganic salt. We expect that they're either soluble or insoluble, but actually they can have very interesting phase behaviors like polymers. We identified two, gel, coacervate. Okay, both are polymer phases. So how come that they can form those kind of phases? We expect that gels are formed because the counter ions are strong, they cannot bend, okay, they stay the sheet, a large excluded volume. How about coacervate? We all know coacervate, positively and negatively charged polymers interacting with each other, liquid-liquid phase separation, both are stable. But look at here, we don't have any polymer. These kind of clusters, they are charged. This is one of them, but we don't have the opposite one. The opposite ones are those mantle counter ions. So therefore, how the mantle counter ions interplay with this and we interplay with each other to lead to those phases, we try to answer these. Okay, so at this moment, we know that gel phases, coacer phases, they are there. And actually, we start to understand that indeed, those rules can be understood. So gelation, critical gelation concentration, critical salt concentration when we add heavy metals, here is yttrium, or some other transitions. So gel phase can be regulated in a triangle area for different microions. In other words, where and when we can identify them, where at the space that we can predict that. Okay, so final thing, that is chiral recognition. So what's that? We start from here. Microions, self-assemble. What happens if we put two similar microions in the same dilute solution? Ask the question, do they self-assemble individually or separately? And we prove that they strictly go that way. That means they self-assemble separately. That means they can self-recognize with each other accurately because the long-range electrostatic interaction. Okay, so that's their feature. We can push always forward. Now we have two microions, one is left-handed and one is right-handed. And they self-assemble separately, leading to chiral Bradbury structures. Okay, one step further, chiral discrimination. Okay, so we all know that chirality, that's just this, right? They are expected to have the same physical and chemical features, but it's not. Like in our body, we have only one type of amino acid and one type of sugar. So how to achieve this? So we have the central macroions. Here is metal organic cage, I think. Counter ions are very normal. We just add a small amount of chiral co-ions far away. They lead to a complete suppression of the self-assembly of one type of enantiomers, but promoting the self-assembly of the other one. So that is different from the current theory. If chiral counter ions are there, people know that they can do that, okay? The three-point theory and so on. Chiral co-ions are very far away. How those small co-ions far away can completely control the assembly, that means the interaction, we don't understand. But we believe that ASAXS will be the key to answer those questions. Okay, I think I will stop here. And I will thank my students, my collaborators, especially Marino's help at ASAXS and funding agency. Thank you so much.
The system that you showed, which had this solution to gel to phase change or coacervation, looked really interesting. I always wonder, especially with that word coacervate, whether or not, there's actually kind of liquid-liquid coexistence. Is there something you can say about kind of the level of order or disorder that you see, or the viscosity in that more concentrated liquid-like domain after
Yeah.
it phase separates?
First, it's very stable. Number two, the viscosity, what we observe is relatively high. They are highly viscous solutions, and sometimes they are gels. Okay, so my understanding is even they are gels, but if there's a clear liquid phase separation, this is coacervate phase. Is that right? I think so. I talked to them.
Yeah, I don't know.
Not gel. They're phase separation. Yes. So you see that we still don't understand the mechanism very well. This is basically our proposed mechanism. That is the 2D sheets. They stack like a graphene gel. If you add more metal ions, they further stack with each other stronger. They stay at the bottom layer. That's coacervation. We may be wrong, but this is the current explanation. So
Okay.
we expect the coacervate phase are viscous.
All right, thanks.
Up to the microphone.
Mine's on and I can't turn it off, so there you go.
It's just a question about the structure of those sheets. Are they hexagonal packing or
Good question. We don't know.
Or-
Uh,
you?
We-
Okay,
We really don't know, but that's critical question. I don't know how we're going to answer that. We tried the ASAXS, but it doesn't work very well because this is basically a single layer. The signal is weak. Do you have any suggestion how we can
yeah, either you put it on the solid substrate, can you do that?
TM or
Or X-ray, or
X-ray?
you can, are they floating on the surface of water? That's
No,
another way.
no,
to
they are
do
in
it.
solution,
Okay.
homogeneous
Then
solution.
a solid substrate thing, how big are they?
I cannot control. They can be big, can be small. If they self-assemble, they are all in the same size, but the 2D sheets, they can be any size.
Yeah, I think it GSAXS or
Just like,
the solid interface.
okay, I will try
X-rays gathered.
GSAXS. Brookhaven has a nice GSAXS setup. Maybe I will try that.
Okay.
Thank you so much.
Thanks.
Oh, sorry.
Oh.
So very interesting talk. I'm wondering, you mentioned the aspect of sorting ions through the assembling process. And what do you mean that the aggregates morphology afterwards will be different than you separate from that way? And then in that sense, if they are all monovalent ions, and what will be the size difference when they aggregate? And would there be a mixture of ions as the as the counter ions that will be in them?
The question is that if we use different monovalent ions, what happens? Yes, there's a difference. The self-assembled side will be different, accurately controlled by the counter ion. How strong is the metal counter ion?
How big is the difference? Of the
Okay, difference is pretty huge. I would say it's radius here 20, here maybe 100.
Just within the monovalent ions?
Oh, then it's probably 20 to 40 or 50.
Okay.
So my question is about the time scale. So when they are forming, so you said it can be like much more hierarchical, right? Smaller domains can form and then they form the big ones, right?
Mhm.
So that small domain formation can be in the time scale of seconds or no?
Oh, it's very slow.
it's very
Slowly is the case. It takes days.
Okay, okay,
Yeah.
okay.
Energy wise, it's a high energy barrier.
Yeah, I was thinking of, as I was mentioning about this microfluidics, is it possible to do something in that?
Yes,
But-
it's slow is better for you, right?
Yeah.
Okay. Okay, thank you.
Time for one more, I guess. Okay, thank you. Turn the floor over to Stuart Rowan from the University of Chicago.
Thank you very much, Matt, Arno. Thank you for the invite. I traveled great distances to get here. All of half an hour, down 55. I'm going to talk today a little bit, give you two little vignettes, two little stories, and I'll try to stay within my time frame, on how we've been using, our students in my group have been using ASAXS to explore order and soft materials. The first class, I'll talk about these are cellulose nanocrystals, or talk about our cellulose nanocrystals. So these are bio-based materials here that we obtain from plant sources. And as nature grows cellulose, it does it as a fibrous structure here. And what you can do, scientists have figured out, is you can take this fibril structure and through either mechanical or chemical processes, you can maybe dissolve away all the amorphous regions here and be left with nanocellulose. And this is what nanocellulose looks here in this little picture. Okay, I need to get used to this. Right, okay, so it looks kind of like cotton, but what it is actually, if you look at it in detail, is these are high aspect ratio nanofibers, right? And they're, say, highly crystalline. And depending on the biosource that you get them, actually dictates the type of nanocellulose that you get, the size, its morphology. So there's a lot of data here, but the only point I want to make here is there's been a lot of work looking at how nanocellulose, or these bio-based nanoparticles, can be used in ion separations for water purification applications, membrane technologies as absorbents. And I'm just showing you, there's a sort of a whole range here on this table of different metal ions that have been explored and removed from aqueous components from doing this. And here, this table in the middle here shows the difference between just normal soluble cellulose here and its binding capability or absorption capability to copper-2. Cellulose nanofibers are very, very long fibers, a mixture of amorphous and crystalline cellulose. But then the cellulose nanocrystals, and then depending on whether you've got carboxylic acids on the surface or phosphates on the surface, you can see much greater enhancement in the binding of these ions. After Turnbull's talk, I probably need to call these nano ions and not macro, right? These are, so I need to change my terminology now. But one of the, and just to give you an idea, this is how we actually separate them in the lab. So these are MXG, so this is Miscanthus giganteus. This is a bioenergy crop. It grows like 6 feet every year and was, investigated quite a lot in the early 80s as a potential source for bioethanol. And so there's farms all over the country. And we had figured out basically how to take these MXG stalks, it's just a big tall grass, and maybe access the nanocellulose from it. And so these are what they look like on a TEM, and this acid process at the end here basically converts dissolves away all the amorphous cellulose and leaves you just with the crystals. You do that with HCl, you get uncharged CNCs, and then we do a sort of an oxidation process to take the primary OH groups here and convert them into the carboxylic acids. We can do other types. If you use different acids, you put different negative charges on the surface of these. So it's quite a flexible platform. And even here, actually, at Argonne, the MERF here at Argonne scaled up this technology. So, so we've got a lot of this stuff to play around with. But it was this stuff that really Harrison, who is a graduate student between myself and Matt Terrell, and Morale started to start thinking about how they might be able to utilize ASAXS to study the ion absorption onto these crystals. And it's going to be very much like just what you just saw from Turnbull's talk. So here's an AFM of these particular crystals. And we have 2 different flavors with different carboxylic acid densities. So either one that has 740 millimoles per kilogram and another one that has 1100 millimoles per kilogram. And these are the dimensions from AFM. The width dimension, of course, of tip broadening, take with a massive grain of salt, right? So I wouldn't rely on that too much. But what Murnau did was, and Harrison did, was to basically mix this initially with some rubidium. And what they were wanting to do is like, let's look at what's the effect of rubidium, strontium, and deuterium and on the binding of these materials. So you can see then the ASAXS data here. And of course, what they then had to do is to develop a model that would mimic the core of the CNCs so that they could extract the data from this. And their model that they came up with actually gave us very good agreement for when they fit the data to it, to the dimensions that we see by AFM on these particular structures. But you see 3 kind of different models here. There's the sort of the core of the CNC up here. there's sort of just a stern layer around the CNCs, and then a sort of one that has a multi-ionic layer. That's the model. And so fitting that data gave a lot of information. For these particular materials, what they found out was that, of course, ASAXS initially confirms that the rubidium ions are absorbed onto the CNCs. You see that in the resonant feature here. And that we get a higher concentration of the CNC, of the rubidium close to the CNCs than we do further away, right? So both consistent with the idea of the rubidiums absorbing onto the CNCs. If we look, what became, I think, interesting, something I was not expecting, is if we look at the effects of just changing the charge density. So we've got two sets of data here. We've got one with the higher charge density and then one with the lower charge density. And it turns out from this data, which we would never have got any other way, I don't think, other than ASAXS, is that with the lower charge density, we only see a stern layer. We just see a sort of a monolayer of the rubidium around the CNCs. And at the higher charge density, this is where we see this additional diffuse layer that Tambo has seen in his macro ions, right? So it seems to be charge dependent as to whether we get this particular diffuse layer. Not 100% sure why, but that's what we're seeing. If you look here at changing the initial ion concentration and sort of the density of the rubidium in these systems, the dark blue color here is the higher charge density CNCs, the lighter color is the lower charge density CNCs. This dotted line here is essentially the density of the carboxylic acids on the surface. And so one of the things that we see from this data is our stern layer is actually a higher concentration than the carboxylic acid anions. So it's more than just electrostatic interactions that are that's absorbing onto these CNCs. So what we think is the density of the rubidium around the CNCs is actually greater than the density of the carboxylic acid. So it's not just a simple electrostatic interaction that we're seeing. There's probably interactions through the OH groups on the surface, but we also think we're seeing a chiotropic effect because one of the crystal surfaces on the CNCs is very hydrophobic. And so we think there's an effect there that's actually enhancing the absorption of the rubidium onto the CNC. And then again, this shows the same data that higher charge density results in this additional diffuse layer. To look at the different ions, again, this is the lower charge density system. So only, again, even when we put in the higher charged ions, we still only see the stern layer in all of these systems. But of course, you see a higher binding of the lower charged rubidium on the surface than you do for the higher charged deuterium. When you go to the higher charge systems, again, now you get to see these multiple ionic layers. Again, we get to see these diffuse layers. And sort of dramatic increases in the amount of absorption for these higher charged atoms in this diffuse layer around the CNCs. So there's a lot of information that we got from these studies that I'm not too sure we could have got any other way. And we're still trying to unpack, I think, a lot of what we're seeing here in understanding that. I want to switch gears to something a little different here for the last half hour of my talk. We've also been looking, and I briefly asked Mrinal about this earlier on, we've been looking at liquid crystal elastomers. So these are liquid crystal materials where we have large mesogens embedded into the network. And so there's a lot of people been looking at this, but when you take these materials and you stretch them, you actually can align the mesogens in the direction of the stretch, which seems then a very interesting thing that we could maybe probe with ASAXS. We got interested in this, Charlie and Alina, who've spent a lot of time up here, got interested in this because what you can take is a polydomain LCE. And if you stretch it, you macroscopically align all the mesogens, and then it will stick there. The mesogen alignment will hold it in that place, and then if you heat it, it contracts. If you start with a material where the mesogens are already aligned, and then you heat it, then you get a sort of two-way actuation process going on, right? And this is well known in the literature. What Alina and Charlie were interested in is they have these disulfides in this particular process. And what you can do is under what I affectionately call a hot yoga process, because we're just stretching at elevated temperatures. The students roll their eyes whenever I say this, right? But what you do is you stretch it at these slightly elevated temperatures, you break the disulfide bonds, and that arouses stress relaxation through this dynamic exchange process, okay? And so through this kind of process, Alina I'll just show you a couple of quick videos, but Alina has been able to kind of train, if you like, these materials through this hot yoga process to sort of align in different places of the material. And so when she aligns this material on the left where the outsides aren't trained, but the insides are, and she puts it on a hot plate, the part that's aligned contracts, right? Because that's where it's aligned. And then of course it goes up. And then as it raises up, it goes down, so it expands, and you get this kind of belly dancing motion, for want of a better term, right? If you train it around a pipette here, so again, just wrapping it around, stretching it around a pipette and put it onto train it at 40 degrees, then you get this permanent spiral put into the material. And then when you put it on the hot plate, anywhere it touches the hot plate, it wants to contract. And as soon as it moves away from the hot plate, it wants to expand. And so you get this breakdancing motion in the system. So, they're cool, they're kind of interesting little demonstrations of what these materials can do. But the key component to all of this is, where are you putting your alignment, right? That's the key. That's how all of this works. And so we couldn't do this with the disulfides, but one of the challenges that Alina really got interested in was, we didn't want to do the hot yoga process. We didn't want to have to heat it up. We wanted to do just the normal yoga, right? Where you could just stretch it to train it. And so she moved on to the selenium bond, right? The diselenide, which is a much weaker bond and should be more susceptible at lower temperatures. But of course, that allowed us to do ASAXS on these things, right? Because we're putting in the selenium bond. So it's the same process. And so if you look at these diselenide materials, this is just standard wax, right? We can see in the poly domain, when we stretch it, you can see alignment, right? But we don't see very much in the way of features. So it's like maybe something in the wax data that we see. If I can use this, there's maybe something there, right? But what we did then is, or what they did then, of course, was use ASAXS on this. And this gave us much bigger wealth of information between the material that wasn't stretched and the material that was stretched, right? What we clearly see here is a symmetric phase, and as you move away from the edge, it gets less. So this is from the selenium edge. So this doesn't have selenium in the component. So we think that this is just the LC symmetric phase in this material. But what we do see as you move away is an increase in these peaks, which suggests these are selenium-containing features. Interestingly enough, when you stretch the material, we actually get a new feature, one or two new features that are related to selenium in this material that we didn't see when we unstretch it. So there's some additional organization going on in these stretch processes that ASAXS is telling us that we wouldn't have got otherwise. So just, Elise Chen, who's a joint PhD student between my group and Juan de Pablo, is now starting to model these systems to see if we can better understand what these distances are telling us. This is still very much a work in progress. So I hope I gave you a little bit of an idea of how we've been using ASAXS, or the students. I haven't been doing anything. I just come up here and give the talks. The students in Merlot have been using ASAXS to probe organization in both the solution state of our systems, but also in the solid polymeric state of these materials. So I just want to thank all of the collaborators here in red, are the people that were involved in this project, and then thank you for your attention. And Myrna will be very happy to answer any of your questions. Thank you. Come.
Oh,
There you go.
oh no, okay. I'm wondering for the rubidium case, and is the like the stern layer versus the diffusive layer thing relevant to the anion?
So it could be, we don't know, right? It could well, the anion I think is the same in all cases.
Yeah.
Right. So whether it is a feature that can be controlled or altered, is that what you mean by the nature of the anion?
Exactly.
That's a very interesting question. I don't know if we know the answer to that. Maybe that's something we should be looking at once the APS gets back up and running again. But yeah, it's a great point. We haven't checked that out.
Beautiful talk. So I have a question about the second part. When you show the structure domains, are those actually spatially distributed? And then if so, what are the domain size they have?
We don't know, but they should. if we've just stretched the material, right, in a homogeneous manner, I would expect that there would be subtle changes. There's been some modeling done, and where when you stretch the material from a polydomain, certain parts of the domain align quicker in alignment than others. So I would imagine if you looked at different points, you might see slightly different results. More interesting to us is when if we can compare, when we partially train certain regions. So when we put it around the spiral, we're only actually training very specific regions. We're only doing the relaxation where we're maximizing the stress in the material. And that I would imagine give you very different results as you went, I say, along the film. But it's a really interesting question. I haven't got enough information from the ASAXS to tell you that, but that's something I think that I would love to be able to see is kind of more mapping on the system.
Just want to add something on top of this. So you have these peaks coming up, right? So we are kind of modeling it to some extent to see. First thing is we can get some rough idea about the domain size from the peak widths, and the thing is which we are trying to get. So this is kind of a little bit known in the sense from some of these studies which people have done before. But only thing which we are trying to get from this ASAXS data is which part of the film, like the selenium, is contributing into this peaks. And they have, it's a little bit interesting, like the peak is going down. So the first peak is going down. So this is very interesting in the sense that we are just seeing the overall contrast of the system. So as you are moving away from the selenium edge, basically you're decreasing the contrast. That's why it is going down. But if it is selenium peak, which is the other two peaks, it is going up. So this is a very beautiful way of looking at the system.
It's giving us information I don't know we'd get any other way.
Yeah. Yes.
Chris here. Yeah.
Hey, I have a question about your nanocellulose with the
My
rubidium.
nanoanions, I'm talking about
with rubidium parallel pipette. So it looks like you see evidence for association on the outside in solution. And these cellulose looks very flexible. Do you see any evidence for the rubidium diffusing in?
So these are
polymers?
basically single crystals.
Okay.
Right. So we isolate them, they're like 90% crystalline. So we don't see any diffusion in. I think all of this is at the surface. Right. Now the cellulose nanofibers are a mixture of amorphous and crystalline cellulose, and maybe doing some studies, analogous studies to this on something like where there's amorphous regions in these fibers may tell us more about what we're, if that actually happens, if we get diffusion in, but these materials are very highly crystalline. So I don't think we see anything that sort of diffusing into these materials.
Considering the amount of number of selenium in the crystals, it's compared to the carbon, very small numbers, right? So it's a contribution to the diffraction peak is pretty large in the sense. Is it because of it's more contributing to the diffraction peak somehow?
In the LCE films?
Yeah.
Yeah.
Yeah. Any questions from virtual setup? Maybe can they hear me? Yeah. I don't see any questions so far. Anyway, in that case, let's thank Stuart.
Thank you.
So our next speaker is Professor Liu Ping Yu from University of Chicago, but unfortunately cannot join us today. He has some medical emergency. So we have like 10 minutes break now with our next talk.
What if I'd known I was talking for another 10 minutes? You kept that quiet.
Our next talk is at 9:30.
Thank you. Yes, yes.
Our next speaker is Raymond Tu from City College of New York. And he will be talking about selective rare earth element recovery.
Oh, sorry, I'm messing with you. Stop. There you go. All right, and then this one. How do I control this?
Doesn't go back.
Oh, I see. Oh, yeah. All right, I'm just not gonna use it.
Okay.
Can you go back to the first slide? I'll see if I can. Oh, I can go back to the first slide. I see.
Yeah.
No, I'll make you. I'm just not gonna point very much. I'm close enough. Okay, so thanks so much, Renal, for not just allowing our team to use the anomalous X-ray scattering, but also having us over to talk about it. I'll speak up. So as far as the project is concerned, the goal of our work is to try to define a more sustainable way to selectively bind to rare earth elements for recovery out of aqueous systems. The work I'll talk about is actually a fairly big team that is funded by the DOE from the University of Pennsylvania, my institution, which is the City College of New York, Northwestern University, and the University of Illinois at Chicago. And so the work I'll talk about actually started as a project really thinking about interfaces. So looking at the reflectometer, trying to understand binding, selectivity, and separation. But then it turned out that we had a question that could really only be answered by ASAXS. And so that's kind of where the project ends. And so, or where this talk will end. And so I'll introduce the problem as an interfacial problem, but then I'll highlight the aspects of self-assembly in solution that are relevant. So I was a little surprised that, and maybe just from the titles, I couldn't tell exactly, but the talk I'll be giving is one of the few on rare earth element separation specifically. But I won't do too much background in that. I think this community knows a bit about the importance of rare earth elements and how they go into a lot of electronic and magnetic and optical devices. And so the problem with the rare earth elements is that, as the name implies, the abundance is low and they come from mining applications that typically have a very high energy, high organic solvent extraction process. And the benchmark way of purifying rare earth elements is to use a liquid-liquid extraction process, which is very costly and not very environmentally friendly. And so our approach, our proposed approach, is to try to leverage the ion selective binding associated with lanthanide binding tags. And so what I'm showing here on the left is the lanthanide binding tag, and you can see the peptide has a sequence that's not particularly long, but it has a decent amount of negative charge, right, in the aspartic acids and the glutamic acids. And so what the lanthanide binding tag can do is it can define a binding pocket and then selectively bind to different rare earth elements with different KDs, if you will. And so the goal of the project is to take this lanthanide binding tag, to make it surface active by changing the sequence. And unfortunately, you can't see it at the bottom here. The end of the sequence is an LLA, which is a leucine, and an alanine, which is fairly hydrophobic tripeptide sequence. And then what we've done is we've added an additional leucine, alanine to make it even more hydrophobic. And so this sequence actually doesn't come from our group. It comes from Barbara Imperiali's group. And she's really done a tremendous amount of work, not just on designing these peptides, but showing how different sequence modifications can bind to different ions more or less selectively. So the goal, though, of making it hydrophobic is to wind up creating a molecule that can, in solution, coordinate to the rare earth element, and then by virtue of its hydrophobicity, hop on to a bubble, right? And in industry, these practices are actually fairly common. It's known as foam fractionation or foam flotation. And what you do is you just bring a whole bunch of very small bubbles through solution, right? And then the absorption of the peptide with the rare earth element will promote these rare earth elements that are selectively bound to float to the top. And then once they float to the top, they're pretty easy to push off and then separate. And ideally, you can do this without a bunch of expensive, environmentally unfriendly solvents. Okay, so that's the idea. And we wanted to see if this idea would work. And we had to test two aspects first. The first aspect that we want to test is to see if we could modify the hydrophobicity of this peptide and then maintain the selectivity. The second aspect is we wanted to see if once we've modified it to add some hydrophobicity, if it's stable in solution, and then if we can actually have it be interfacially active, right? So that's what we did first. So if you look on the left, what you're looking at is the free binding energy, and then the effective ionic radius, so for different rare earth elements, as a function of sequence. So on the left, there's a sequence that is the Imperiali sequence, and on the right is the sequence that we've designed with the additional LLA on the far right, right? It's on the far right because the binding pocket is on the left, right? So on the left, ideally, you'll not mess with the binding pocket too much by making it a little bit more hydrophobic by adding the LLA on the right side of the sequence, right? So if you look at this, the free energy of binding versus effective ionic radius has terbium, for instance, as the molecule, as the ion with the lowest KD, right? And it has lanthanum up there way on the right, and then lutetium way over here on the left, right? If you look at what happens when you modify it with LLA, right, you have terbium, dysprosium, also at very low KD, so very strong binders. And then again, lanthanum and lutetium, and the shape of it's fairly similar. The numbers are a little bit different. They're tabulated here and here. But in the end, it seems like we can modify that terminus of the peptide without messing up the binding pocket nor messing up the selectivity too much. So the next experiment we did was a pendant drop experiment. And I won't go into too many of the details, but it's a very straightforward experiment where we just inflate a bubble in solution, right? And then we look at what gets absorbed to that bubble as a function of time, right? So we can look at the change of the shape of the bubble as a function of time, and it balances gravity against the surface tension, right? And when we look at the shape of that, we can solve Young-Laplace equation, and it tells us what the surface tension is as a function of the concentration of the peptide or the peptide with LLA or the peptide bound to the terbium, right? And so what this figure is trying to highlight isn't the dynamics, it's highlighting the what we'll call quasi-equilibrium state. And what you're looking at here is that the LBT peptide, so the peptide without the LLA, it's a little surface active, right? It'll jump to the surface. But then once you add the LLA, it becomes much more surface active. So a lot more of those molecules want to jump to that air-water interface. When then the LLA molecule binds the terbium, right, you can see that it becomes a little bit more surface active in the case without the hydrophobic group. But when you have the hydrophobic group, once it's bound to that terbium, it really likes the interface, right? So it'll jump to those bubbles as the bubbles move up through the solution, right? So this was all good news. And then comes the bad news. So we did X-ray reflectivity, and we did it in combination with X-ray fluorescence. So the X-ray reflectivity teaches us about the electron density as a function of depth. The X-ray fluorescence teaches us about the ion concentration for the different rare earth elements as a function of depth, right? And so we could do all that analysis, and what we expected to happen was that at a given terbium bulk concentration on the x-axis versus the number of terbiums per lanthanide binding tag, again, this is now the hydrophobic one, that we would get a curve that started low, and then as we increase the terbium amount, it would go to 1, right? Because each of those lanthanide binding tags, the LBT peptides, could bind to 1 lanthanide and then jump onto the interface. That is not what happened, right? What happened was, even at these lower concentrations, I should say, oh, it says the peptides are at a concentration of 100 millimolar here in this solution, right? And then as we started to increase to an equivalent concentration, it bound above 1. And then as we went to much higher concentrations, it bound almost, 1.7 terbiums for every single peptide that we have in solution. And so this is not what we wanted, right? We wanted selectivity, and wanted selectivity by virtue of binding into that binding pocket, right? And so what we wound up seeing was that, and this is actually a simulation, right? So when we did simulations, we found that there's an ion that lives in the binding pocket and another ion that lives in a binding pocket there. And then what winds up happening is in this case, the C terminus, and then there's a few other negatively charged amino acids would often bind to those terbiums as well, right? And that was an issue. And that's when we turned to ASAXS, because we wanted to know essentially what comes first, right? Do we self-assemble in solution into these aggregates that are cross-linked by the presence of the ion, or do we go to the surface and then the ions come to the surface and then start to interact, right? And I see that I planned really badly, and I'm almost out of time, so I'm not skipping slides to hide anything. We made a couple other sequences to think about it. We saw some behavior, but the key piece that made us think that we had to go into the solution is if you look at these KDs for the LLA sequence, and you do the calculation of the ratio when you actually try to get selectivity of terbium to lanthanum in this case. So this is something that binds really weakly compared to something that binds really strongly. You'd expect to see at some of the concentrations that we're at, something like, 35 times more terbium than lanthanum, right? Instead, we see something more like 1.8, right? So it's really not binding with the selectivity that we want. And so we had to look in the bulk at the aggregates and ask the question, does that 1.8 number come out in the aggregates, right? And so we did some light scattering again. I'm going to skip this just because I'm out of time. And then we did some simulations, and the simulations showed that you have aggregation a little bit because of that LLA group, but you also have aggregation due to the terbiums coordinating and forming networks. But the ASAXS taught us the following, right? I'll make one quick note about the ASAXS because we spent a lot of time with Mrinal. Actually, my student Lewis spent a lot of time with Mrinal, probably not as much as my student Gershon does though, on a different problem. And we had to model this aggregate, and I showed quickly the TEM, as 3 phases, right? So there's water inside the aggregate, right? There's terbium inside the aggregate that's free, and then there's terbium inside the aggregate that's associated with the self-assembled peptides, right? When you look at it, if you guys spend a lot of time modeling ASAXS data, right? You assign a density, a volume fraction, and then this R moles number tells us about how much peptide is associated in that aggregate with the peptide, right? So how much terbium winds up coordinating specifically the peptide, and then how much exists in the solution phase with the water, right? And in the end, this is what we found, right? And so these are aggregates now at much higher concentrations, right? This is the ratio of terbium to the lanthanide binding tag in solution, right? And then this is the ratio of terbium bound to the lanthanide binding tag in the aggregate, right? And if you look at this number, and I won't highlight that number, right, what you see is that at these high concentrations, at ratios above 4, it comes pretty close to this number, which is, around 1.8. So it's a little bit higher. We think that's because we're at a much higher concentration of peptide. But yeah, it seems like the population of rare earth elements that are coming to the interface is really dictated by the aggregated structure. And that's kind of the answer that we got from ASAXS. After we do our beamline run, we always get Italian beef because it's Chicago. And that's Lewis and that's Gershon, and that's us without much sleep. And yeah, certainly special thanks to ChemMatCARS and Mrinal for all their help with this. Thanks.
Thank you. Quick questions? Anything online? Anybody online has any quick question? Yes, May, go ahead.
So this is a chemistry question, not a ASAXS question, but what do you think drives the selectivity across the lanthanide series?
No,
If you
it's a
said
good
that, I
question.
didn't quite catch it.
I did not say that.
But it's interesting that you got this, I don't know, I described it as
Yeah,
amphiperic.
so I'm not gonna go through this slide. But what usually is thought of as deciding what defines the selectivity is a combination between the acidity and then the steric hindrance. So on one side, the acidity of that pocket wants to grab ions, and the other side, as the ions get too small, you can't close that pocket small enough. That's not the case for our system. We don't know why. It seems like it's all dominated by the acidity, especially that's not true for our system as far as the interface is concerned. That's true for bulk binding, but once it goes to the interface, for some reason, it's perfectly fine binding small guys. We don't understand that.
Okay, as we are running out of time, let's move to the next.
Thanks.
That is a virtual talk from Melissa from Pacific Northwest National Lab. Yeah, she's online. Hey, Lisa.
I'm here. Good morning, everybody.
Yeah, go ahead.
Am I sharing my screen?
Yeah.
Can you see the presentation mode?
Yes.
Fantastic. Okay, so thank you, Marina and the organizer, for inviting me. I'm sorry I could not be there in person. So I'm a sub-scientist at PNL, and I should disclose that I'm quite new to both anomalous X-ray scattering as well as the study of electrolytes. And my interest in electrolytes started when I joined the CPMs group at PNL a couple of years ago, where instead experimentalists like John Fulton or theoreticians like Sean Cutman, Greg Shenter, and Chris Mundy have been studying the structure of electrolytes for many years already. And my background is in chemical dynamics, and I've used time-resolved X-ray scattering, and I've always been curious about anomalous effect, and so I thought that this would be a great technique to try to solve some open questions in the group as well as in the field. And so I contacted Marinal, and we were lucky enough we could do some preliminary experiment last February before the shutdown. And yeah, this slide is to acknowledge the collaborators I've mentioned, and you should also acknowledge Abdullah Karaman, who helped during the experiment, and Adi Dimpagio, who did the MD simulation I'm gonna show today. So understanding the structure of electrolytes as important application in many fields, from chemistry to biochemistry, geochemistry, and material science. And I have collected here a non-exhaustive list of application. I should specify that we are mostly interested right now to understand the long-range ion-ion and ion-water correlations. So ordering that extend beyond 10 Armstrong, because it is a range which is relatively less explored in the literature. Yeah, so from microscopic properties of the electrolytes, like atom pair distribution function, we can retrieve important macroscopic properties like the osmotic pressure that has relevance in biological application. Another important example is the fact that sodium ion ordering can affect DNA folding. And long-range ordering is also important for the formation of pre-nucleation cluster, as well as ion transport and reactivity. And the conventional way to access this long-range ordering would be through small angle X-ray scattering, which can be directly compared with molecular dynamic simulations. And in this plot on the left, I'm showing some small angle X-ray scattering data of calcium chloride in solution as a function of salt concentration. And so starting from the bottom, we have the most dilute sample. And we can see that in the low Q region, the signal is flat. Then after 1 inverse Armstrong, this would be the onset of the water liquid peak. But when we increase the salt concentration, a new diffraction peak, which so-called the pre-peak, appears, which report on the fact that the ions start seeing each other and ordering themselves in the solution. And in the plot here on the right, if we plot the peak position as a function of concentration to 1/3, we can see this linear behavior. And we, at the most concentrated sample, we eventually get that this peak position corresponds to the largest Bragg spacing in the hexahydrated calcium chloride crystals. So I don't have time to go into the details about this, and I invite you to look at this publication from my PNNL colleagues. But for instance, from the structure of this pre-peak, we could also have a screening length and other parameters that can be directly linked to liquid state theories. And so I said that we can get microscopic information by comparing the data with the MD simulation, and this is just an example of a comparison for 4 molar calcium chloride of the MD simulation, which is the red dashed with the experimental measures signal, which quantitatively reproduce the data. And my colleagues could establish that by looking right at the MD simulation, that is pre-peak, and so the ordering arises from cation-cation interaction, and the fact that each cation is closely hydrated by surrounding water molecules. But from the MD simulation, we can get additional information. So this is a zoom on the pre-peak region. And it's possible to disentangle the different atom pair distribution function. And for instance, we could see that calcium-calcium, so cation-cation interaction, as well as cation-oxygen, for instance, contribute constructively to this pre-peak, while cation-anion and anion-oxygen pair distribution function would contribute destructively to this peak. And here I come to the Anomalous experiment, because from conventional scattering measurement, we cannot benchmark this information that we have from the MD simulation. Instead, our idea is that by using anomalous rescattering and adding element sensitivity, we would be able to disentangle atom pair contribution and therefore benchmark in a better way our theoretical models. And I should also say that I was particularly interested in see what is the utility of ASAXS for dilute chemical system, which I think are less explored with this technique. And so in the presentation, I'm going to show simulation and data from erbium bromide in aqueous solution. We pick erbium bromide to start with because erbium is a strong scatterer, and because at 15 ID at the beamline, we could access both the erbium L edge as well as the bromine K edge. And these are conventional ASAXS data on the left, and you can see this stronger pre-peak, which shifts to higher Q distances, also for erbium bromide as a function of increasing concentration. And on the right, I'm comparing with our preliminary MD simulations, which qualitatively reproduce the pre-peak, but they don't reproduce the shifts. And so meaning that there is some room here for improvement of our force fields. I want to also point out that the correlation from this MD simulation, you can see that the correlation extends beyond this pre-peak region. And so ideally, we would want to measure an extended Q range. And I'm going to come back to this point in the very last slide. So from our, I just said that the MD simulation are not perfect, but for now we have these, and we can start with this to see what would be the anomalous effect, the expected anomalous effect for this system. So I picked here 1 molar erbium bromide, and this is the, I included the anomalous effect, and this is the expected changes in the pre-peak region. So it's basically a decrease in intensity of this peak as a function of increasing distance, increasing energy approaching the erbium L edge. And these three contributions are the exactly the atom pair distance that containing erbium are therefore the one that contribute to the anomalous effects, and these are different different scattering signal normalized to the peak. So basically the erbium atom pair distribution function would contribute the most, almost 30%, to the change expected to be seen in the pre-peak. And indeed, this is what we measured. So these are data we collected in February for 1 molar erbium bromide, and we can see that we observe a decrease in intensity as a function of increasing energy. We are working on trying to subtract the fluorescence background in a more efficient way, because you can see from the simulation there is no Q range where the anomalous effect do not contribute. So it's difficult to find a range where to estimate the offset. And in this case, I just offset with respect to the very low Q region. And we are working to improving this, but nevertheless, I think the anomalous effects are clear. At the bromine K edge instead, this is the MD simulation prediction. We can see that we should expect smaller changes. And this is because the bromine atom pair distribution function would contribute would contribute so that the peak intensity decreases, and also in this case would contribute almost 30%. But the other two contribution, erbium bromide and bromine oxygen, contribute the other way around. So they make the peak intensity to increase, and so the two contribution basically cancel out. So we don't expect to see much changes in the pre-peak. And indeed, when we did the experiment, we saw very little differences as a function of energy in our experimentally measured pre-peak. But I think we could observe consistently, and here I say consistently because here I'm just showing 1 molar, but we measure a series of concentration, and consistently I see a peak shift, which is more visible if I take a differential scattering signal with respect to the one measure at the first energy far away from the edge. And I think this could be explained by the fact that maybe different atom pair contribution contribute to the pre-peak at different, slightly different position, but this right now is not reproduced by the simulation, so we are working on it. And with this, I come already to the conclusion of this short talk. So I hope I could convince you that we can use anomalous X-ray scattering to systematically examine with unprecedented sensitivity long-range ion-ion and ion-water correlation in aqueous electrolytes, which are important for many chemical phenomena, from ion transport reactivity to nucleation. And specifically, we want to provide a new dimension, which is not accessible with conventional X-ray scattering, a new dimension for which, with which to test our theoretical models. And we are working on, for future experiment, we are thinking about a few improvements. We had to use, in this picture here, you can see the sample holders that we utilized. So basically we sandwiched the sample between the two Kapton windows. We needed a very thin sample path, but this was a longer process to make hundreds of these sample holders. So we are trying to think about a more efficient way to make these holders. And then, as I mentioned before, we would like to access a higher Q range than just a small angle. And this will be possible. This is what Marinal has said as it's an upgrade that they are thinking about the beamline. And in connection to that, we also need to think about how to correct for polarization, which becomes important at higher scattering angles. And we are, as I mentioned already, we are thinking as a better method to subtract the fluorescence signal. And with this, I wanna, let's see if I can go to this last slide. I cannot, but there was a big thank you. And I'm happy to take any question.
Thank you, Lisa. So thank you. We're going to go for coffee break. Yes.
So is that the pre-peak position is directly correlated to the concentration? I saw that it was a 1/3 power of concentration, but it's a homogeneously distributed in the solution. That's my question is.
Yes, that's a good question. We think so. At least there are some quasi-lattice structures that are big enough, right, to give rise to this periodicity. Yeah.
I did a quick little calculation. It seems like when you have 1 mole concentration, the Q0 position should be order of, let's say, 0.05 inverse angstrom. So it looks like a 10 times smaller spacing you have in the diffraction, in the scattering. So that's why I was curious. Maybe my calculation was wrong, or
1 molar concentration should be so 1 molar concentration is this magenta line on this left plot here, and it's maybe around 0.4 inverse Armstrong. Does that agree with your calculation?
I thought it's 0.04 or ish or something.
So this is the concentration 1 to the 3rd, this plot.
That I see, yeah. So, okay, thanks.
Okay, Mike, you have a question?
Yeah, that's a good plot to sit on. Do you mind if I draw on the slide?
Sure, go ahead.
So here's the peak that you saw. There's something here, and there's also something here. Right, so this is pretty clearly some kind of a shoulder of some kind that manifests also, I think, perhaps at the lower concentrations.
Yeah.
What are those due to?
Good question. I don't think we assigned those peak yet. So these data are published in this publication here, but I think there is no assignment of that shoulder in the publication. It's quite difficult, right, to start assigning at some point these distances because, yeah, when we go above 10 inverse Armstrong, we see this periodicity in the GDR, but it should be possible with this highest concentrated sample. So yeah, something to look better into, I think, something to look into.
Yeah, I mean, it's kind of a wild extrapolation maybe, but it almost looks like they're in a 2-to-1 ratio in Q. Of course, very, very hard because you're sitting on something big sloping background.
Yeah.
Okay, cool. Thank you.
Okay, then thank you, Lisa, for the talk.
Thank you.
Yeah, then we go for our coffee break. And then we'll be back in 15 minutes. How do you calculate that? I think we did evaporating in some way no, it's the at some point no, it's not that way for someone else to leave that work.
Oh, no, if I just in this class before I would do all right, but yeah.
Okay. So, okay, let's begin the session after the break. So our next speaker is Mike Tony from University of Colorado, and he will be presenting virtually. So Mike, you can share your presentation.
Okay, the standard, can you all see that question and can you all hear me?
Yeah, we can hear you.
Okay,
We
cool,
can see your
great.
presentation as well.
Okay, cool. Okay, so let me move this away. Let me go here. All right, great. So I too wanted to thank the organizers for the chance to tell you about some of the recent work we've done looking at solvation structure and electrolytes. I too am sorry that I can't kind of make it. In contrast to Stuart, I think I had to move, less than a millimeter from my office to my office chair in order to give this talk. So this is an outline. I'll briefly give a motivation. I'll give an example of how we're thinking about it. And then I'm going to finish with some kind of final thoughts in a summary. The final thoughts are meant to be kind of more general than specifically what I'm going to kind of touch on. So most of the work I'm showing you is done by one of my postdocs, Antonio, who's shown here. Some of this work was started by Julian Mars. And then we've collaborated with the Argonne team and gotten great support from ChemMatCARS. And while I won't talk about it, we've also been working together with some people doing molecular dynamics at Notre Dame. And lost it. Oh yeah, I think I lost this. This work is actually funded by J. Caesar, which I somehow forgot to put on here. So the issue of nano aggregates in electrolytes has increasingly kind of become to be recognized. And so these kind of consist of hundreds to thousands and perhaps tens of thousand ions that form kind of heterogeneous domains. So there are a couple of examples of this. One is the so-called water and salt electrolytes, where, and this is a bit controversial, you can form domains that are rich in water, and then domains that may not necessarily have a lot of water in them. And this then will depend on things like electrolyte concentration. Somewhat less controversial is the issue of with solvent mixtures. You can kind of get phase macroscopic and microscopic, as I'll show you, phase segregation into water-rich and salt-rich domains, and then in this case with acetonitrile, acetonitrile-rich and then salt-poor domains. So this is a couple of examples. This is the highly concentrated electrolytes, solvent mixtures, polymer electrolytes, very similar kinds of problems occur. And then we heard from the previous speaker, kind of another example of this. This is not surprisingly driven by the kind of molecular scale interactions. From a functional perspective, it then has a huge impact on things like where the ions are and how they transport. So depending on the exact nature of these, you're going to have an impact on the transport properties. That's kind of obvious, but perhaps more subtly, because the environment associated with the ions are different, the electronic structure and electronic properties can be different. And so from an electrochemistry standpoint, that can drive differences in decomposition properties, differences, for example, desolvation, and differences in the structure of the electrochemical double layer kind of as well. And here's a nice paper that Lei Cheng from Argonne actually published that kind of discusses aspects of this. I think this was last year. In any case, that's a problem. Here's an example. This is some work that was done by a previous group looking at water acetonitrile kind of solvent mixtures with, this is, but they did a bunch of different salts. This is magnesium sulfate. This is the phase diagram. So up here is homogeneous regions, and then down in here is macroscopic phase segregation. And then you can, of course, draw tie lines that will allow you to look at the endpoints and what the composition is at kind of the endpoints is classic kind of thermodynamics. Our interest in this really stemmed from loading control. No, what do I do here? Actually, let me just move this. I think there we go. Our work stemmed from some work that Argonne had done, where they would find now with water acetonitrile and zinc sulfates kind of mixtures in this kind of equivalent macroscopic phase segregation region. You would have a so-called bottom layer, which would be rich in zinc sulfate, and then a top layer that would be rich in acetonitrile. And if you go ahead and use this as a zinc battery, it turns out that the bottom zinc-rich region had actually very good kind of cycling properties as opposed to the top layer, which basically failed. And so this drove interest in our part in kind of understanding aspects of this. And so I'll tell you a little bit about what we've done there so far. Come on. So this is the phase diagram. I'm going to skip this in detail. So this is a phase diagram that Emma kind of found. So blue is kind of macroscopically mixed, red is macroscopically phase segregated, and then green is kind of dissolved, not dissolved structures. So that's kind of that is in fact a version of phase segregation in some form or another. And so what I'll be showing you is a couple of results then that we measure the ASAXS profile as we go ahead and fix the salt concentration. So the zinc sulfate, so I didn't write that down, concentration going from 0.01 to 1 to 2. And that just marches across the ternary phase diagram in a line, whereas we increase the concentration, you can then see, we move from a single phase region into kind of this two-phase region. You can also do things as a function of varying our constant acetonitrile concentration, but I'm not going to show that. So again, same phase diagram. I'm going to show you some ASAXS data as we walk through this line by blowing up this region here. And so these are now the ASAXS profiles. We'll just go through this. So these are now as we walk through the mixed region, or sorry, the well-mixed region. So macroscopically well-mixed, but you clearly see the peak in the ASAXS regimes increases. There is some kind of phase segregation or incipient initial phase segregation in kind of the salt with zero in pure water. But as we go ahead and increase this drives a stronger tendency to phase segregate. So this is not surprisingly right. You're approaching kind of a phase diagram. Aspects of this are related to, for example, critical behavior. And then okay, so this goes on to 18%. I forgot I had this. This goes on to 18%. And then as we go into this region here at about 20%, we move into a regime where you have macroscopic phase segregation. And if we look at these two curves, you see that there's also in each of the endpoints microscopic phase segregation as well, which is not too surprising because the composition of those two phases is pinned by whatever the tie line kind of gives. So this is interesting, right? Because we see this kind of behavior where you see these kind of microscopic phase segregated regimes. And as I attempted to talk about before, this can drive aspect of transport, which I don't really have time to talk about going forward. And this kind of summarizes then what we found is these nanoscale phase separated regimes into zinc sulfite water rich regions. That would be the bottom part, and then acetonitrile rich and salt poor regions kind of as well. And then you see that the extent of the phase segregation goes up, so the intensity of this peak goes up, and this shoulder shifts to the left, so the size scale associated with those go up as well. And I'm not going to go into those kind of in detail right now. Okay, so this is what we found. So how can ASAXS help? So first of all, these are qualitative statements. We don't actually know necessarily the composition. In principle, if we had mapped out the complete phase diagram, you could get this kind of from the endpoints of the tie line, but that's a little challenging to kind of do. So ASAXS can help us two ways, right? I've drawn you this very simple picture. It's simple, but is it correct? Are there subtleties in this that we're perhaps missing? And so by changing the scattering contrast of the zinc, by basically varying the X-ray energy, we can in principle get insight into this. And then if you think about this, we kind of have two independent variables. right? So we know the acetonitrile composition, we know the water composition, we know how much salt we put into this. If we have two phases, if I did the math right, they should give us two independent quantities. So if we can vary the contrast, we should be able to get the composition information from this. Kind of falls out of the math, which I'm not going to show you, but if you walk through this, it turns out you can get insight into this. Turns out that has to do with the fact that the scattering length densities or electron densities of the water and the acetonitrile are not quite the same. But I don't really want to go into that kind of in detail. So as I said, this is a general problem, right? I'm showing you one example of this. This kind of nanoscale phase segregation from the electrolyte problem occurs in a number of different situations. There's lots of interest now in mixed salts, particularly for multivalent ions, where now you have two salts and potentially heterogeneity as a result of that. There's not much that's known in that case. I mentioned kind of the water and salt system, and here's an MD simulation kind of that, where you again have very similar problems. And then even for lithium ion batteries, there's a concept in the battery community of using high concentration electrolytes to basically tune the interface structure. That has problems because the viscosity goes up and so the transport goes down. So I think PNNL actually developed this. You will take a high concentration electrolyte and dilute it, and that inherently creates phase segregation. And so how that impacts transport and how that impacts reactivity is not particularly well known. All of these have the potential for ASAXS to kind of start to provide some insight into. So I think this will become an increasingly powerful technique for these kinds of problems. And then last, just to close, multi-component systems are kind of ubiquitous now, right? And many of these are not homogeneous. And so from a SAXS and ASAXS perspective, this now becomes a powerful probe. So I've listed a few of these catalyst nanoparticles, so transition layer of platinum, which was some work we did actually probably 10 years ago, looking at sort of these highly active catalysts where ASAXS proved to be quite useful. Earlier, I don't remember, someone talked about the quantum dot aspect of this, which I put this in at the last minute. As part of an NSF STC, we're very interested in narrow line emitters. Many of these have core shell structures, and so aspects of the nature of the thickness of the core and the shell are always big questions. And so ASAXS has potential to be useful there. From halide perovskite structure, which is something else I'm very interested in, these are all multi-component alloys. And in principle, the alloys are uniform. We actually don't think that's the case. And you can run into regions, for example, in this particular compound, where you can have regions that are bromine rich and then regions that are iodide rich. And these have a big impact on things like stability. And so we don't really understand that particularly well. I'll skip this. And then this I also put in at the last minute. Because this is an NSF funded, I don't remember what the term the program entity used, but let's just say center. There's a lot of opportunity here to think about kind of being creative from the educational outreach activities perspective. I'm pretty sure you all are already doing this. But if you think about training the next generation of beamline scientists or training by students, then there's some interesting opportunities that I think ChemMatCARS, and even unique opportunities that ChemMatCARS can kind of leverage here from basically the access perspective. I expect you're already thinking about this, but I would encourage you to kind of think about this very, very broadly, because I think there's a huge potential to have a real impact here. And then just last, thank the people who funded this, Jay Caesar, NSF. So Emma's done the work. Lacey, who's a grad student, is taking this up. And Claire actually is an undergrad. She's a sophomore now, will be a junior. So that's an example. Then she's gone to the synchrotron, but she actually has done an ASAXS experiment. But that's an example of kind of the educational opportunities that can kind of broadly be leveraged through things like ChemMatCARS. And last, I'm just going to end here and happily take any questions.
Thanks, Mike. So can we have some quick questions, either here or online? Any questions here? Yeah, go ahead.
Yeah, hello. I have a quick question. Since you mentioned about the potential application of this technique for battery systems, could you comment on current studies on, for example, the solid electrolyte interface for battery systems? Can we actually get some new insights using ASAXS or ASAXS?
Kind of. Not for lithium because it's just too low, but for multivalent systems like manganese, right, or zinc. Again, depending on energy, there's a potential to use this to disentangle aspects of the SEI. I mean, I would think of this more in terms of an anomalous reflectivity kinds of experiment where you take a model interface and basically use that. And that's part of what the student I mentioned is going to try to be doing from the zinc side. There you have the potential now of giving you pretty unambiguous speciation kinds of information. I mean, ASAXS in principle will give you the same, or actually will give you the same thing. Assuming the length scales are kind of correct, and the right morphology kind of exists. So I think the potential is there, as is with any kind of complex and multi-component system, but the details depend on exactly how you design an experiment.
Okay, thanks, Mike. So due to time, we have to move forward. So our next speaker is May Nyman from Oregon State University.
Okay, I'm really upset you took away my speaker view, but I'll do the best I can. So thank you, Mrinal, for organizing this interesting meeting. It's great to be here where everybody understands ASAXS and also with the people who develop the programs that we use in our ASAXS. So I'm going to tell you a little bit about introduction to my lab and the role that ASAXS plays in our lab, and a specific story on ASAXS analysis of metal DNA complexes, and followed by ASAXS measurements of the metal DNA complexes in collaboration with Mrinal, and then a recent proposal that we got funded that we proposed to use ASAXS in, but no data yet. Okay, so my lab, we studied metal oxoclusters from metals from all over the periodic table, and we're interested in form-function relationships and also how they're made. So how we get from simple ions to clusters and then clusters to materials using the clusters as building blocks for these materials. So the schematic here depicts the role primarily of pH on these processes. So if you're in the extreme ends, extreme acid or extreme base, we have monomers. As you drive towards the middle through hydrolysis and condensation reactions, you make clusters, and at any point you can isolate them, especially if they're monodispersed. And so pH, concentration are primary controllers, and then generically shown here in the purple and yellow spheres is counterions and coions, which we've heard a lot about in this symposium today. And as you can imagine, ASAXS plays a very important role in all of these studies. So up in Oregon, we don't have a beamline right in our backyard. So we have a Anton-Par ASAXS instead. This is 10 years old and still going strong. So let me introduce you to it. We have a copper source, and we've got, this looks very well. We have a line collimation crack key system. That's why we've got this box instead of the long tube that you get with pinpoint. Or pinhole ASAXS. We introduce our samples in sealed capillaries, put them in the vacuum chamber at point number 3 there. And when we first got this ASAXS, it didn't have that curved extension. So we had ASAXS, which went out to a Q of 0.7 reciprocal angstroms. And we worked with fairly small stuff. So we really wanted to go further. We were missing a lot of information. So we got this wax extension, and now we go out to 2.5 reciprocal angstroms. And we have an image plate, not an electronic detector. And what this means is that we have to read out our images after collecting them in this carousel here. So, you know, think Polaroid photos or something like that. The last speaker talked about next generation beamline scientists. So the students really become experts having this at home. And not only do they use it every day, they also take care of it. They align the instruments. So this was our most recent alignment with a brand new crew in February 2023. They were successful, they're happy, and for good reason. This is not easy to do, especially with this image plate, right? Because you have to read out after every single time. Okay, so in addition to our own studies, we have a lot of collaborators, and even people we don't know contact us. They send us samples. We published about 15 papers to date doing this, and 5 other studies in progress. And I'm actually going to highlight today some of these collaborations rather than our work, because that's where the ASAXS has come in. So this is Miguel Galindo. He's a professor at University of Granada. And this is his webpage. And as you can see, he likes to study the supramolecular assembly of DNA with metals. And he contacted me around 2019. And not only did he send me samples, he brought his whole family for a sabbatical in March 2020. But we all know what happened. 10 days after he arrived, everything shut down. There was a global pandemic. So we spent a lot of time in the park with the kids drinking beer. But we did collect data. And in fact, the black and the blue scattering curve, I think we were actually able to collect before the shutdown. And I was published in 2021 in Angewandte Chemie. So this is one of the approaches that Miguel has taken for strategic assembly of metal and DNA. And I'm a little out of my comfort zone here. I don't really know DNA that well, but bear with me. Okay, so the approach here is we've got a 15 adenine DNA strand. So adenine needs to hydrogen bond to make a double helix. With thymine, there's none present, it can't hydrogen bond. Okay, so the black scattering curve is the ASAXS of just the adenine. It just flops around in solution, maybe folds in on itself. It's not very impressive. Okay, so then Miguel added this palladium complex with a ligand, and the idea is that it will take its ligand and then bind to the adenine, and that is the blue scattering curve here. So that is a 15 adenine strand, and we were also able to make a model of what we would expect to happen with the double helix, and that's the simulated data in red. And we were very happy. This is a very nice match. So that was one strategy to replace the hydrogen bonds of DNA strands with metals instead. Okay, so this is the strategy that Miguel is taking now. Okay, so here is our bases in natural DNA. So if you're to add metal to it, like palladium or platinum or silver, it really tends to bind to that N7 shown here, rather than go into the center of the double helix and replace the hydrogen bond. Okay, so instead, Miguel designed this modified DNA called the ASA, where you replace that N7 with a CH. Okay, so now when we add the metal should go to the center of the DNA. Okay, so we use ASAXS to study this. And now, since we've got all the base pairs present in our DNA strands, we can get hydrogen bonding in the absence of silver. So that's the black scattering curve here, and it actually matched nicely with the simulated scattering. And as we start to titrate in silver, so we call this equivalent silver per base pair, where we expect 1 silver per base pair to go into the double helix. we get significant increase in our scattering intensity. And we model it with a cylindrical model. So that's approximately correct. Here's our cylinder over here on the left side. And it's about 20 angstroms wide by 35 angstroms tall. So up to what we found that we cannot model the data with just a single cylinder population. We needed a second population, especially as we go to higher concentration. Okay, so we only see something approximately the size of the model that's shown on the left with one silver per base pair. And then we start getting much longer strands up to almost 200 angstroms. Okay, so this is a benchtop ASAXS. We had to use very high concentration. So what we think is going on, and because there is literature precedence, is what's happening when we get that elongated strands is you're actually getting end-to-end polymerization of your DNA through silver-silver interactions, maybe silver phosphate, we're not sure. And so this is a crystal structure that was published in Nature Chemistry in about 2017, I believe, with natural DNA, and they were able to isolate these infinite silver chains within DNA helices. Okay, so we want to go to lower concentration to eliminate this polymerization, and we also wanted to compare the modified DNA to the natural DNA to see if there's a difference in where the silver goes when you eliminate that N7 where the silver really likes to bond. So we contacted Renal because we needed a brighter light source, and of course Renal said, let's do ASAXS. So we did that. We sent them the samples. Again, this was 2020. There was no visitors, plenty of beam time available. And we collected data from below the silver K edge to at the silver K edge at 25 keV approximately. And again, here's our hypothesis with natural DNA. The metal goes to the N7. With modified DNA, it goes into replace that hydrogen bond in the center of the helix. Okay, so in every case here, the black scattering curve is at the silver edge, the blue scattering curve is below the silver edge. Okay, so this is the modified DNA with no silver, exactly the same, that's what we expect. As we start to add silver, we start to get absorption and we get decrease in the scattering in the low Q region. And we continue to titrate in more silver. It becomes more complicated because we start to see fluorescence. But like everybody else, Renal used his black magic and modeled the ASAXS component, the resonant component, the cross component, and got us some interesting, very compelling data. Okay, so the red data point is the natural DNA, sorry, the modified DNA without that N7. The blue data point is the natural DNA. The first thing that we see for both systems is a decrease in the radius, and that's because we're putting silver in. So our Z or electron density is concentrated in the core. Comparing the modified to the natural. Okay, so now with the modified DNA, so we're looking at how much silver we added versus how much is inside the core according to the models. With the modified DNA, it about maxes out at one equivalent per base pair. So that means you got one silver replacing each hydrogen bond. But in the case of the natural DNA, we still get that polymerization. And this is non-specific coordination of the silver to the N7, in addition to in the center. Okay, and then one final thing. With the if we look at the length with the modified DNA, we see only that one population, which is about 20 angstroms wide by 35 angstroms high, and this is a total of 12 to 15 base pairs. And in the case of the natural DNA, we still see that polymerization even at low concentration. Okay, so last part of my talk. Together with my colleagues, Kara Knope at Georgetown University and Paramiro, who's a computational chemist at University of South Dakota, we recently got DOE funding to study metal-metal separations using metal oxide clusters. And here's the general concept. So if we've got two metals that are similar in cluster forming conditions, when we put them together and we make clusters, what happens? Do they form clusters together? Do they form different shape and size clusters? And then how do we exploit these differences to separate them physically, either by liquid-liquid extraction or by crystallization? Okay, so we've studied zirconium-hafnium separation quite a bit already. In fact, this was the inspiration for getting this grant. This is the process that is used, or this is the chemistry, the process that used for zirconium-hafnium separation in the nuclear industry. So thiocyanate plus ammonium plus zirconium and hafnium, it makes these oxocentered tetrahedron for both hafnium and zirconium. But zirconium is poorly behaved. It also makes larger clusters. And we learned this by a combination of ASAXS and crystallization. Okay, so this was studying separate systems, and now we're interested in studying the mixed metal systems. So from our benchtop ASAXS, we can see with the mixed metal systems, we can see a beautiful march to larger species as we put more zirconium into the system. Okay, so from anomalous ASAXS, we want to know in these mixed metal solutions, where are these metals going? It's like, is the hafnium going into the larger clusters with zirconium? Are they completely separated? And we actually call this narcissistic self-sorting. So do they sort themselves or do they mix? And these are the two most similar metals on the periodic table. So it's a great system to study. So this was in the proposal. It's not data yet. But we just were simulating some scenarios of the lanthanides with different known cluster forms and what happens if you have mixed metal systems versus separate metals, and how does the ASAXS data change depending on where the metals are. Okay, so I'd just like to finish up by acknowledging the students who collected the data or who are currently working on the projects that I presented, and I'll just leave that here for questions.
Okay, time for some quick questions. Yeah, go ahead.
When you calculate the cluster scattering, what degree of fluctuation, positional fluctuation you guys are assuming?
I'm sorry, I don't understand the question.
For your cluster calculations, probably it's not gonna calculate the scattering pattern from just the crystal structure, right? I guess a certain degree probably you have to assume the like the positional fluctuation or thermal
Simulation.
Yes.
Okay, so the only simulation I showed was for the palladium DNA.
Okay, I thought the last slide you showed was a simulated curve.
Oh,
Yeah.
yeah. We're not actually did these.
Oh, okay, you did.
Yeah, so yes, there is a possibility we can, in the function there, we have a possibility to add this kind of a random fluctuation surrounding that position, and that will decay those kind of high Q features.
So can you say how much actually they fluctuate from their ideal positions?
Well, we don't have a data to show it, but
Okay.
we can do that, yes. Once the data is there, we can simulate and see how this thing changes. Yeah, any questions here? No, I don't think so. Anyway, so thank May, and then we will move forward to Chong Liu from University of Chicago.
Okay, good morning. I'm Chong Liu, assistant professor at the University of Chicago of PME. And today I'm glad to share some of the efforts that we put into understanding the ion transport in confinement with the help of anomalous XRD as well as anomalous ASAXS. Okay. So the motivation, as we already hear from 3 talks, is driven by that we have a lot of critical elements that we need to build renewable technologies. For example, the electric vehicles need a lot of batteries, then you need lithium transition metals, as well as for wind turbines, then you need the magnet, then you need a lot of rare earth elements. So all these are not in a percentage of adoption that we desired for a carbon neutral society. But then here's the question of how can we make sure we have the raw material to provide to the manufacturers to secure the supply chain. And so this is the motivation for our group to study how can we better separate these materials to diverse the sources that we can use more dilute and more complex chemistry to extract these elements, as well as whether there are more environmental friendly and energy efficient ways that you can separate the element, and then you don't have initial carbon footprint of that heavy load from the beginning. So our nature does have a very effective separation way that's based on ion transport. This is an example of potassium ion channels, and it can transport potassium at a very high speed, almost to the limit. And it has a very high selectivity of potassium to the other monovalent ions, such as sodium, which is very similar in chemistry. So the reason for that is that it has a particular selectivity filter that's made of a precise chemistry, and then it allows a very optimized energy landscape for the potassium only to transport without the dehydration too much, but with a optimized binding so that it can quickly go through with this dehydration rehydration pathway and to transport inside and outside of the cell. So the question we have is whether we can mimic something with artificial materials, for example, solid state materials, to create such confinement, and then we can dial the size and the chemistry and to achieve the separation via just ion transport. So if you think of a ion transport based separation, actually you can drive it by many methods, but since all the things are confined in one type of solvent, ideally, so the energy consumption could be drastically decreased. So that's the benefit of that. So what we need here is several key physical parameters or key physical chemical processes that governs the optimized energy landscape. So what you can do here is, for example, here are two examples of confinement shown. One is a two-dimensional confinement, and the other one is a three-dimensional confinement. So you can dial with the size of how you construct this confinement. And basically for ion separation, especially in aqueous environment, you want this confinement to be less than 1 nanometer. So it's comparable to the hydration first hydration shell, or in general, the hydration radius of the ion. So you can control the dehydration barrier. And then is what kind of chemistry you pick for the material that you want to hold the ions inside. So that's the binding energy, which is coming from thermodynamics, as well as when you have this is a solid material, so there will be dimensions. So the transport of ion in this solid ionic channels will have a barrier. So it's either through hopping or through different kind of mechanism. So you can tune this barrier as well. So basically, if you combine all these processes together, you actually have a great deal of knobs that you can tune, and add them together, you optimize the energy transport landscape. Okay, so to really figure out in the ion transport measurement, what is governing the selectivity, you need to figure out two things. One is the thermodynamics, and the other one is the kinetics. Okay, so for thermodynamics, what we want to know is how ions are really sitting in the membrane, either a membrane or an electrode, whatever physical processes you are involving for the solid. And for that, the binding mode or binding site can be characterized by many ways, such as you have a specific probe, you can use FTIR or Raman, or you have this ion binding that would induce a structural change that's observable. So you can use XRD. And anomalous XRD is very good that it simultaneously gives you both the chemical speciation as well as the structural change. So you can tell exactly where the ions is sitting in what kind of site inside of this crystal. So for the kinetics, you want to know a spatial, you want to know your element, where are they, with a good spatial and temporal resolution. So usually what we do is we can do snapshots where you use TM, for example, to look at the spatial distributions of these ions with a good resolution, 2 nanometer, right? But sometimes you only can look at several particles. So it's good for single particle analysis where you want to pinpoint some key features on the atomic level, but including a lot of samples, ensemble information, whether all these particles are behaving samely, and then you want to use a ASAXS method to look at chemical structural information at the same time. Okay, so now I'm going to give an example of material we studied. So this is a Prussian blue analog material, which is a 3D framework. What it has is it has 2 metal centers. One is iron and one is copper. So each of them, so there are like 2 metal centers, and in between of them is a cyanide group that links them. So when you do material growth, they will self-precipitate out into this kind of cubic structures where it's alternating copper ion site with a C and N coordinating to different kind of metal. So this would give you a structure that has multiple different kind of confinement. So for example, in a regular perfect crystal, you have iron, you have copper, and then it builds into this, what we call A site in the material, where the length scale is around 4.6 Armstrong. Definitely this is tunable if you change the metal center. And what happens during the growth is if you want to balance your charge, usually there will be iron vacancies that formed during the growth. So the iron vacancy, if you look at this 2 picture, is actually just a missing site of the iron inside the center. So this gives you a larger storage site of bigger than 5 Armstrongs, sometimes can be 7 to 8 Armstrongs. So you can store a larger thing in those type of sites. And then when you are talking about ions to go into the structure, then it has to go through different kind of pathways. So one pathway will give you, like if you go along the 100 direction, so the confinement at the narrowest point is around 3. So if you look at what kind of ion you want to study and their hydration radius, you can see this confinement actually would be small enough to disturb the hydration shell of all the ions. So you have to pay your dehydration energy for your ions to go in. And when it's go in and you don't really know how many coordinations are left and how the ions are binded in the structure. So this is what we want to know at the beginning, how the ions, all the ions are sitting inside the structure. Okay, so first we did indeed pick all these, all this different element to probe. And okay, so for the alkali metal, so they are all monovalent. And this, the way how we drive the ions into the structure is by electrochemistry, where you can reduce the iron from 3+ to 2+, and then to balance the charge, you can incorporate the other cations into the structure, where the going in is compressed of all the physical process I mentioned. So in here, you can see if you drive the entrance of the ions into a particle, they show different energy requirement to sit into the structure, where the higher voltage is indicating a better selectivity because you will always, in a reduction reaction, you will always go from a higher to lower voltage. So whichever shows a higher voltage, it means that it can go into the structure first. So within this group, it follows very much to the hydrated radius trend, that smaller hydration radius would indicate an easier is going into the structure and with a higher binding with a better selectivity. So it's the reverse trend of the bare ion radius without hydration. So it definitely means that the dehydration energy and the migration barrier is playing a key role in here. But when you go to all these ions, actually only the monovalent ion that follows this hydration trend, which means as your ions going bigger and bigger in their hydration shell, and the thing gets more complicated. For example, if we look at all the divalent ions in the green series, the higher curves that are actually calcium and strontium, but you are not looking at barium as the one that's the most selective, or they kind of show similar selectivity around here, but are all better than magnesium. So the lower selectivity of magnesium, you can still attribute that to the dehydration penalty, but for the other three, the trend is blurred or is reversed, which is complex. And the same thing for the trivalent. So if you look at the trend, actually there's no trend to the hydration enthalpy anymore. Okay, but this curve of thermodynamics plus kinetic information does tell you selectivity at a relatively correct trend of which ion would be better to sit into the structure than the others. And the selectivity does follow, we tested, it does follow the trend of the voltage curve. So then the question is, there are two types of site, as I mentioned from the beginning, that the ions can sit in. And would they be different for different type of ions? They prefer different kind of site to go into the structure. So then we did the anomalous XRD. And if we only look at the potassium one where we probe the indium edge, which means that there's no indium, so there's no absorption effect to the X-ray diffraction. So what you see is, even though you move from the indium edge and there's no change of the XRD spectrum. So that means this is, if you are not probing the element, there's no response. But then if you are targeting to certain type of element and using their absorption edge, and then you can get different kind of, like we see here, different kind of shifts of peak intensity that's indicating the information of that element in the structure. Okay, so why can we use this? So if we are looking at the general patterns, they are all looking the same, which means that this type of 3D structural material doesn't have too much of a structural change by incorporating ions, which is hard to use ASAXS to differentiate, which ASAXS is very sensitive to morphology size. But then you have to go to a smaller length scale in here, where, let's say, if you look at diffraction on the 200, which is the first peak, and actually you are looking at the planes that directly across the center of this lattice. And if you look at the 220, actually it crosses all the A side and B side together. And if you look at the 400, it also across all the A side and B side together. It's only this 200 because it only cross one time at the middle, and then you are having a sensitivity to the B side instead of the A side, because A side is only covered in the 400. So that gives us an indicator of saying, if we probe the divalent versus the monovalent, can we tell which one prefer to sit in the A side versus the B side? And if you look at the rubidium data, okay, so the rubidium data, if you look at the 200, the response by going away from the edge is not very obvious. But the response to the 220, which you can see across this A side, is increasing, because when you are at the absorption edge, you decrease the scattering or diffraction intensity. But if you're away from the edge, then your intensity recovers back. So that definitely indicating this rubidium ion is sitting preferably on this A side, because the response to the 200 is not strong. So it's not an indicator of B side occupancy. But if you look at the strontium instead, and then both the 200 and the 220 have a strong increasing intensity in their diffraction, that means these two are always crossing where the strontium are sitting, and especially the 200, where is the center of this cubic. That's saying that the divalent strontium have a preference in the B side, much better than the rubidium. So that's the conclusion if we use elemental sensitive XRD, and then we can get this correlation that monovalent prefers A side, divalent probably prefers B side. But we cannot exclude the possibility that the divalent also sit on the A side. But then this becomes interesting, and then we look at the redox response. Okay, so there are two transition metal centers. One is iron and one is copper. And if you look at for different monovalent versus divalent, here we have potassium and magnesium. And you can see for potassium, and most of the redox is run on the iron. And for the copper, most of the for magnesium, most of the redox is running on copper. So that tell you another thing, that whether this A side and B side occupancy is also correlated with your ions redox. If that's the case, it makes the picture even more interesting, that you can dig deeper as whether your ions site occupancy is driven by the redox potential, which redox element you use, or is the reverse way of the ions that's preferred to sit in here to balance the charge. I prefer to run some redox. So this correlation versus causation is the thing that we want to keep digging deeper in the future, where, like say, if we figure out that the redox is driven the occupancy, then can we change the transition metal to really tune which transition metal runs first to tune the selectivity of the material? Okay, so finally, I just give a quick example of showing like how ASAXS is helpful for us to review the transport pathway. So ASAXS is very good, sorry, here, okay. So ASAXS is very good in terms of giving you multiple information at the same time. So for us, we look at the crystal material. If you have structural features, and then you want to look at always X-ray scattering, X-ray diffraction. And if you want to look at element information, sometimes you have to go to TM or electron microscopy to look at the distribution. But ASAXS can give you that all at once. And this can be even like with the upgrade can be in operando. So then we can have a in situ chemical structural information at the same time. So with this, we first want to validate whether the chemical and the structural information we get is correct. And we use a different system where it has a 2D confinement, and we can put different rare earth elements in it, and we can accurately using ASAXS to pull the chemical information by probing the edges of two different elements, and they match exactly as what we test via ICP-MS ex situ. So which means in situ, if you probe ASAXS, you can actually know the composition of your material and how it evolves as the selectivity is changing. And then another thing we care about is the structural thing. Okay, so when we have ions going to 2D structures, sometimes it will expand, and the expansion degree is elemental sensitive. And by that, usually we use XRD to characterize the expansion of the structural indicator. But with ASAXS, it can also tell us the L in here, which is a accumulated thickness of this particle, because each layer is expanding. And then you have, for example, 10 layers, 20 layers. So with this accumulation is a good enough signal for ASAXS to tell you. So, and then we correlate this two, and it shows very good correlation that no matter you probe with the small scale using XRD, or you probe using the larger scale using ASAXS, it can all give you a consistent structural change. So in that case, if we have a case where we can tune the dynamics to fit into the characterization timescale of ASAXS, then we do operando test. Then we can have the structural and elemental information at the same time, where we can probe how these ions are going through and whether when they go through, whether they have dynamics of exchanging or not, which is complicated to get via different techniques that you have to combine and correlate. So for example, in here, dysprosium is showing that it's going from the edges, and this is a snapshot, and we quench the sample where you can see it's all concentrated at the edge and it start to go into the system. But if the time scale matches, then it will be a good example for the ASAXS operando test. So with that, I want to close my talk and thank all my collaborators and funding source, and especially for this topic, a lot of support from Miro. And thank you all.
Out of time, but maybe one quick question, if there's any.
Sorry about too.
I don't see any question here, but in any case, thank you, John,
Yeah.
and we'll move
Thank
forward to the next talk. So our next speaker is Daniel Fessenmeyer from Purdue University. So he's from UYON group.
Yes, so thank you all. As you mentioned, I'm a PhD student and Professor Yuan Wan's group at Purdue University. And today I'm going to speak to you about how we're trying to use ASAXS to explore the fundamental behavior of a novel polymer lung surfactant therapeutic. So a brief background.
Yes.
Oh, okay. Why we're exploring this topic is when during severe respiratory conditions, which can be brought about by a variety of indirect and direct causes like COVID-19, as we all know. The native lung surfactant, which lines the air-water interface within the alveoli, becomes deactivated by the presence of certain blood proteins, as well as phospholipases. And this serves to degrade the function of this natural lung surfactant substance. And this contributes to the poor respiratory behavior during in this state. And currently there are no effective therapeutics, surfactant replacement therapeutics. So we're exploring this novel polymer lung surfactant. And as shown in the diagram on the right, the polymer lung surfactant is proposed to form a more insoluble monolayer, which is resistant to these types of interactions with compounds in the subphase. And we've shown that this therapeutic is effective in mouse models of lung injury. And therefore, we are very interested in exploring further its fundamental properties at the air-water interface. So our current formulation is blockcopolymer, amphiphilic blockcopolymer micelles. And these micelles have a hydrophilic PEG corona, which provides colloidal stability as well as prevents these types of interactions with surface active proteins. As a very highly hydrophobic core, which helps strongly anchor the micelles at the interface, giving it the ability to produce high surface pressure, which I'll go into the next slide. So our current formulation, our model material is PSPEG, roughly 5K molecular weight. And these nanoparticle micelle structures have a diameter of around 30 nanometers. So just a brief background in terms of their the surface mechanical behavior. So for the lung surfactant formulation, we are primarily concerned with its properties at the air-water interface. And so we study this using Langer trough technique. And so when these micelles are spread in water solution onto the water surface, they readily absorb to the interface until the interface becomes saturated with micelles. And this occurs around 10 millinewtons per meter, which is associated with the equilibrium spreading of a PEG film. Additionally, when the micelles are compressed, they can produce very high surface pressure. So approaching 70 millinewtons per meter, which is roughly zero surface tension. And additionally, these films collapse and wrinkle at high compression, which is shown here in this booster angle microscopy image of these wrinkle structures. So in addition to these surface mechanical analyses, we want a more in-depth understanding of this micelle film. And therefore, we want to use, trying to use X-ray reflectivity, X-ray scattering measurements. So we can use X-ray reflectivity to probe the vertical structure. So this can give us information on the how the micelle core is wetting the interface and how this changes during the compression. Additionally, this can help us understand the effects of changing this core chemistry or micelle morphology in terms of its structure at the interface. And additionally, we want to use grazing incidence X-ray diffraction, or GIXD, to probe the horizontal ordering of these micelles and how this changes with compression. Additionally, looking at the same factors I just mentioned, if we change core chemistry or change morphology, how these all are affecting the fundamental behavior. So we have done one beam time where we measured the GIXD of the pristine micelles, so just the bare PSPEG micelles. And what we found was that these micelle monolayers do show hexagonal symmetry, which occurs at this 10 millinewton per meter surface pressure, which is shown here in this GIXD plot of this peak here. And this the spacing, hexagonal spacing, scales linearly with trough area up to about 30 millinewtons per meter. So which implies that there's no desorption occurring in the surface pressure range. However, above the 30 millinewtons per meter, this peak disappears. And so we think this is due to the increase in the roughness of the surface, as well as because the low contrast between the core and the surroundings, electron contrasts. Additionally, for X-ray reflectivity measurements, we did find that there was a change in the reflectivity profile with surface pressure, as shown here in the different colored X-ray reflectivity plots. And we found that there's an increase in the peak of this reflectivity plot, as well as ultimately the decay increases with further, decay is more rapid at higher surface pressure, which indicates surface roughening. And from the electron density plots, which was used, we used a 3-slab model fit, there was a slight increase in the surface excess with increasing surface pressure. And there are some subtle changes. As you can see, the absolute magnitude of the change is relatively low due to the low contrast. So the preliminary model fits, so we reconstructed the electron density profile for the 3 components. And what we found is that this suggests that there is the blue curve is the volume fraction of PEG, so that there's a highly concentrated PEG layer at the surface, and that the core domains, which are shown in the red, are primarily submerged into the subphase. And as you compress the monolayer at higher surface pressure, the PEG becomes more concentrated in between the micelle cores. So although we were able to get this preliminary results, we would like to improve the signal to noise ratio of these measurements. And this is because there is low contrast of the core material. So these are different potential copolymer materials. They all have relatively low electron density values compared to water. And so we would like to increase the core electron density to allow more, to reduce the uncertainty in our model fitting for X-ray reflectivity. So to do this, we are exploring encapsulating, want to take advantage of the fact that these micelles can encapsulate hydrophobic compounds into the core domain. So we can encapsulate a hydrophobic compound with a higher electron density. This can help improve the contrast for X-ray reflectivity. So essentially, this compound must be very hydrophobic in order for it to be stabilized in the core domain. So we've looked at preliminarily using this bulky hydrocarbon compound with two iodine components, which I will call DIB. And we use this rapid mixing technique, which is known as flash nanoprecipitation, where a water stream and an organic stream containing the polymer and hydrophobic compound are rapidly mixed. And the material, the iodine containing compound, is encapsulated. However, we don't know the exact distribution of this iodine compound within the core, and we need this for proper X-ray reflectivity fitting. So that is why we want to use ASAXS in order to obtain this exact distribution. So as you all know, but it does not need to be said, but we can use ASAXS to analyze the structure factor for the specific element in order to obtain the exact distribution of the iodine within the core. And we did some preliminary measurements, and we used a spherical core model. However, we did obtain a radius fits. However, the Q values associated with this radius were well into when the data was very noisy, and we think this is because the concentration was too low. So however, this does seem like a feasible technique, but we need to increase the concentration in order to obtain a more reliable fit for the ASAXS data. And additionally, we tested these loaded micelles using reflectivity, and we saw some interesting features occur, especially compared to the non-loaded micelles. We see this peak, which occurs around QZ of 0.2 inverse angstroms, and this doesn't really match up with our predictions using this spherical core model. So we think the distribution may be more complicated than what we originally thought, and so we need to do more characterizations in order to really properly understand this. So moving forward, like I said, we want to repeat using higher micelle concentrations. Additionally, we want to explore maybe using different hydrophobic compounds, specifically maybe using some fraction of iodostyrene, which has a relatively higher electron density. So using some fractionated of this along with non-iodide PSPG. And then ultimately combine this with our X-ray reflectivity and GIXD to help us with more fundamental understanding of different effects like core chemistry, core morphology, as well as maybe changing the subphase conditions. So with this, I'd like to thank my group, especially Seong, Kim, who helped me obtain this data, as well as special thanks to Dr. Weibu and Muriel for their help with the reflectivity and the ASAXS measurements. So with that, thank you for your time, and happy to answer any questions.
Okay, so time for some quick questions. And anybody online having any question? Okay, if not, I just want to ask one question about, so they formed this kind of ordered structure and you saw the GID, right? But is it possible, it's because you're thinking of increasing the concentration of this particle for the ASAXS measurements, right? Getting more particles there to increase the intensity, but is it possible they can aggregate there if you increase the concentration in the bulk?
That certainly is something we need to consider, but we think if we have a high enough like PEG graphene density, the corona, it will prevent the aggregation of the micelles in bulk solution. So we think 50 milligrams per milliliter is achievable with our formulation. So
Okay, so far you have tested the similar condition as you use for surface experiments, right? Is that right?
yeah.
Okay.
Yeah.
Okay. Yeah. Thank you. And yeah, meanwhile. You can just speak from there.
Question. It's not the wrinkling of
Mhm.
your film. What is the wavelength of those wrinkles?
So it's like 7 microns.
So that you can get out the bending rigidity of those films, right? From
Mhm.
that number. Is that very elastic? Can you pick it up from water in single piece? It's very elastic.
Yeah, it's fairly elastic. We've never tried picking it up before, but
Can you tune that elasticity by changing your polymer sizes?
so I've done a study, and the bending does change slightly if I, let's say, like decrease the PEG corona thickness, the film becomes stiffer. But the changes aren't that significant. So, but maybe if we change like the morphology of the particles or something, that could give more greater range of changes. So yeah.
Very
Yeah, thanks.
Okay, thanks, Daniel. And so we will move forward from here to Xiaobing from Argonne National Laboratory.
Hi, my name is Xiaobing Zhu. I'm beamline scientist at the TIDB, which is a high throughput ASAXS beamline at APS here. Today, I'm going to talk about the 2 projects that ASAXS can help. The first one is we want to use the ASAXS to do the element specific mapping for the material nanoparticle. Those something like the Mike mentioned in his talk. The second one, we want to use ASAXS for the biomarker to measure the labeled, for this case is this good nanoparticle labeled site in the RNA molecule, and also measure the distance between the label. So for the nanoparticles, you have a uniform density. We can use those software, the ASAXS software, to reconstruct the 3D shape from the ASAXS data. That's for the uniform nanoparticles. For the non-uniform, they have a different density. We can try those. The dense is designed to deal with these density variations. And another software you can use is called Mensa. You also can do the multi-phase reconstruction. This you need the multiple set of data. This only need one set of data to do the reconstruction. The system we are interested is gold silver core shell nanoparticles. We synthesize from the gold sheet, and they deposit the silver seed, and they deposit the gold on the surface. Then we do the heating. Then we look at the alloying process. So you can see for the alloy particle and the core shell particle, their photonic the property is quite different. So we want to know during the process how this the gold atom is migrated and how where is it distributed. So all this work done by the very talented visiting student Sui Wu from Temple University. The sample we started with, you cannot avoid they have some polydispersity. That's the data we have. So we start. That's the sense data. We heat it up and collect it at a different time. Of course, the size distribution there. So you figure out the method to pull out the consider all these the distribution you know, then figure out the form factor for the particles, then do the reconstruction use this program that we get. This, can you push the button on those? The first one, the last one. Here. I mean, we are doing. So this is.
Don't use that button.
Oh, we are doing it from.
It's here.
Oh, okay, that's fine. Okay. 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 So here we can see that there's a high density shell here we start with, and then we heat up a long time. You see this the diffusing into inward. So this makes some sense, but not totally makes sense. You know, we expect them more uniform. Here we're not sure is a should localize a certain region or not. So that's we want to use ASAXS to verify this result. So we start with the extraction measurement. That's the sample with different amount of gold is coated on the silver seed. That's the ASAXS data. So the last one, this one that we use, that's this sample we use the heating experiment. So we collect the ASAXS for all the samples. That's one example shown here for this sample. So you can see the signal change. We collect the different energy. This we can see better. There's the signal change at the different Q position. So we are working on the data analysis for the ASAXS data. But here's our plan. We know for the ASAXS, we can write into 3 terms. First, that's the ASAXS term for the whole particle. And the third one is the resonant term that only for our case as gold element. This is the cross term. So we get measurement, we have the many equations, we can solve it out and get the pure complement for the whole part, and only the green one is for the gold itself, gold alone. So we can feed this to the program. We want to, we hope we can get a higher resolution than the one we see the dense construction. That's the first part. The second one, we want to use the ASAXS for the biomarker. You know, we use that to measure the internal distance and also identify the site of interest. We know the FRED is a popular the biomarker tool, but the signal is decay quite fast along the distance between the donor and the receptor. The working distance normally is around 1 to 10 nanometer. For the size label, we think so we have an advantage because they have a bigger distance dependency, and also we expect can measure the distance between 1 to 100 nanometer. So this method have been proposed by Dave Scott group in 2014. They propose use the nanoparticle to label the biomolecule and use an ASAXS to measure the distance. He did a simulation on this double labeled DA helix. And in year 2016, a young left group, they make the sample and measured at the 12 IDC. Here, this is another ASAXS. The beamline can do ASAXS. Then from the data, you can pull out the contribution only from the label. That's the signal. So you can measure for the red one, the distance is about 12 nanometer. So the signal here is still quite strong. So that's a very impressive. In recently, a group at Tsinghua University, Professor Xinyang Fang, they developed a very efficient method to label the ion molecule with the good nanoparticle. That's the chemistry they developed. And they also use the ASAXS to figure out the label, the distance between the label, use the normal SAXS. That's what they do. They did. They need to synthesize 4 molecules, the ion alone, and the ion with one label, ion with the other label, and then double label. So you can do the math on those. Now you can figure out the contribution just from the 2 labels. Now that's what they did. Now from this signal, SAXS signal, you can do the perturbation analysis to get the distance between them. Yeah. So here we need 4 molecules. So we want to repeat that, but just use the ASAXS that only require 1 molecule. Okay, the double labeled. It's another sample. The first batch sample is not that good. So here I can show you some simulations. So that's this stem loop labeled by the good 25 cluster. This stem loop is 30 kilodalton. The label pretty heavy. Is about 5 kilodalton for each. The software I use, that's SolX. That's me at the WTDV developer here. They can include almost all elements. I can do simulation, all atom simulation for the bio and the single molecules. Can do the normal SAXS wise, also normal SAXS. So you're interested, the software can find it here. So here, that's the result. The thick line is for the normal SAXS simulation for each component. For example, here, the magenta one, that's a contribution from the 2 cluster. The black is RA alone, and the sign is for the whole things. Of course, we also do the simulation, the ASAXS, that's this rainbow thick line, the thin line here. We can do analysis on the simulation. That's what we get. We can solve the equation that gets 3 components. It's normalized, showing here. So from this one, that's that last time is supposed just from signal from the double label. So we can do the perturbation function analysis, and we get the distance. That's exactly it should be. The first one is just the single particle itself. So we can do further. So here we get the scanning for the whole system, and also we get only the RA, the label. So this one we can is easy to measure. This is the RA itself. So we can use, that's our plan. We want to use these 3 together, fit it in the program. We want to do the 3-dimensional shape reconstruction. So in that way, we not just give you the distance between them, we also know where this label site is. So we did a simulation here. How is it compared to we solve the equation here? Okay, that's the one. Okay, they write on. So that's confirm my the SolX software is accurate. So here we use the exact the number we do the simulation for the F prime of F double prime. So what if we have some error there? So this is some simulations. We can put 1% error on these parameters. So for this term, you get about a 2% error. So we will solve that. Now this is still, as a whole system is still right on, but for the resonant term is off a little bit. Okay. You will put a 2% error there. The two chart, you can see it jump. This is still right now because it's dominated by the whole thing. But this one you can jump around. Okay. But still, you even use that, so those green curve, you still can get a distance, but not good enough for the three-dimensional reconstruction. We haven't considered the error in the ASAXS data yet, so you can imagine, this is not trivial. We have some idea how to restrain, what kind of restraint we put to get a more accurate the solution, but I haven't have time to implement that. With that, I want to thank other people involved this project, especially thank to Marina and Sanka for the support for the ASAXS permit.
Okay, thank you. Yeah, go ahead.
So it seems like as you have a greater range of sensitivity at r to the -1, there'd also maybe be some issues with crosstalk. So when you have one pair of nanoparticles talking to another pair on a separate molecule, is that an issue at the concentrations you're at, or do you never see that?
I'm sorry.
So if you have, say, an RNA that has a pair of nanoparticles, and then you have another RNA that has a pair of nanoparticles, it seems like at higher concentrations, just because of the range of interactions, they start to see one another.
If you high concentration, you may have those. So this simulation is for the one molecule.
Oh, you don't have any others.
Okay.
Yeah, go ahead.
Yeah.
Oh, yeah, thanks. So about the dense method-based reconstruction, what is the spatial resolution?
I forgot. I think generally for the ASAXS, the size low resolution technique, but generally is a nanometer.
Nanometer. Oh, that's actually quite amazing. So for that to work, does it require the sample to be very model dispersed?
Yes.
I see.
So kind of my question is a related question. How much, so all the ASAXS formula that I saw was more coherent scattering formula is how much the incoherent sum, so like a fully dispersely affects. And
yeah, I did some calculation on those things, and it is like there is two regime. Like if it is monodisperse, it works fine, and if it is polydisperse, it works fine. It is in between some region where this kind of separating component doesn't work properly. Yeah. So that is sometimes happening with nanoparticles also. If we try to, there are artifacts coming out of this due to your noise which you're showing or due to the polydispersity.
in the dense deconstruction, did you try also different polydispersity?
That one is, so you figure out the way we know the known distribution, you can pull out the form factor. It's supposed to be that's the uniform. It's the monodisperse.
Yeah. Okay, thanks. We move forward. And the next talk is from Mark Swihart from University of Buffalo. Yeah, it is a little bit jumpy. It doesn't work properly.
It's all good. Well, but then the Zoom people can't see it, so this works sort of. We have a similar system. Sort of. So I should go back and just say this is almost entirely the work of my PhD student Shua Liu that I'll show today, including he mostly made the slides and everything else. So getting to that career stage where sometimes the really old professor looks at the slide that he's presenting like he's seeing it for the first time. I'm not quite there yet, but I see it on the horizon.
And also I'm the first person I think with like absolutely no scattering data of any, well, no APS, ASAXS usage at all. So it's more just a little quick tour of some of the stuff we make and trying to listen to everybody's talks and figure out if there's ways that we could use ASAXS and AXRD to understand some of this better. So I want to talk about one part of our lab which uses this aerosol method to make nanoparticles. So what we do, I'll try not to linger on it too long, but down here we have a hydrogen oxygen flame, and we suck that through a converging diverging nozzle. So we sort of accelerate those combustion products to sonic velocity, but they're already done burning. And then as they go through the nozzle, we pump in an aqueous precursor of metal salts. And then typically we run this with excess hydrogen. So there's sort of a reducing environment here. So like the things in the middle of the periodic table that can be reduced by hydrogen in the presence of water come out potentially as metals, other things come out as oxides. And then, they fly along in here for 50 milliseconds or so. And then a cold quench flow comes in and sort of stops them from growing and sticking together more. And this can occur either in a mode where these tiny droplets evaporate completely and new particles nucleate from the gas phase, or in much of what I'll talk about today, just each droplet gets converted to a little particle or a little hollow shell. Some things that are nice about this are that we can use, because we can use aqueous precursors, we can buy inexpensive aqueous salts to put in here. So we have access to, essentially the whole periodic table. I cringe a little bit when the students ask for rhodium or something, but at least it's easily available. And so, that's what it looks like in the lab. It's, I forgot to point out it's glowing. So it's, it's in the, sort of 400 to 800 degrees C kind of range, typically in the synthesis chamber, of course, hotter below where the flame is. And we can make we've made all sorts of stuff with this over the years. So if we have, you can get over there. If we have gas to particle conversion, so everything goes in the gas phase and new things nucleate, and especially if we put in some capping ligands in that quenching flow, we can get these small, not monodispersed, but relatively uniform particles. A lot of the time we're making, hollow shells of one kind or another. And because this all happens very fast, we can mix things together, whether they would like to mix or not. Anyway, we'll boldly keep going. So again, as things go through here, most of what I'll talk about today is a process like this, where we spray a little droplet. Because the solvent, the water, evaporates from the surface, the highest concentration of precursors at the surface, and so that's where things nucleate and grow. They grow inward. If we quench this fast enough in that thing at the top that looks like a time temperature transposition curve, if the trajectory is fast enough, but not too fast, things can crystallize, but they can't really phase separate. So we can mix things together even and make all sorts of metastable structures. So the first example of doing that, and again, I've got 6 months worth of work shoved in one slide in a few cases here, but this is just sort of a set of examples where we're making binary oxides. So we're taking a parent lattice and putting 10%, or for some of the rare earths, just a couple percent in. And the way we always characterize these is that our collaborators at Molecular Foundry, so Chao Dunn and his advisor or colleague now, Jeff Urban, do lots of elemental mapping, so TEM, EDS kind of mapping of individual particles. They always just sort of show everything's uniform. And then, we do just regular laboratory X-ray diffraction and, look to see if any separate phases are nucleating other than the parent phase that we expect. In some cases, it's a little hard to see in our little picture down here all the way on the bottom right. So here with a couple percent iridium, we can get isolated iridium atom atomic sites on there, which is of interest for single atom catalysis. And over here in the set of XRD patterns, that's just doping or adding increasing amounts of palladium into Syria, and we can get up to like 40%, 40% of the cations being palladium before we start to see any hint of phase segregation. So of course it would be nice to look at these in ensemble measurements where we get a little more information about distribution than what we're getting here. A challenge would be that these tend to be hollow and polydisperse and kind of messy samples. One application of these that we've looked at a number of things, is we can make catalysts for the dry reforming of methane. So reacting methane with CO2 to make synthesis gas. Something cool we can show is that essentially we make this metastable solid solution and we put in a pore templating agent as well. So we get hollow shells that are now porous. And then when we heat these in a reducing environment, the nickel is exsolved. It comes out as little tiny particles. And they're really, really well anchored because they're embedded in that porous structure. And so, skipping over a ton of things, these boring flat lines here basically just indicate the tremendous stability of these catalysts over hundreds of hours, when more conventionally prepared catalysts will deactivate fairly quickly. So again, maybe challenging samples, but it'd be interesting to see what, even just using ASAXS to look at the distribution of the smaller exsolved particles, if possible. Something we've been playing with a lot recently is these high entropy oxides. So if we keep adding more things in roughly equal amounts, right, we get the entropy of mixing counts more, and we get, among other things, high temperature stabilization. Again, we look at these things in the same kind of ways, except instead of having, 2 cations, we have 6 or 8, or I think his record is 22, right? Cations mixed in. And at some point, it becomes very hard to see them by EDS, right, if you have too many. But again, they always look about the same. The XRD patterns get broadened and noisy, both because these shells themselves are polycrystalline, and because now there's a lot of disorder in the lattice itself, because we've mixed together, half a dozen cations of, different things. So we've done this for lots of different phases. Some of them, rock salt, basically every site is the same, but something like a spinel phase, there's different sites in there. It'd be interesting to know if particular elements are going on to particular sites, for example. One example of application of this is for another energy related application. So CO2 hydrogenation, to take it back to CO, that then we could go make something out of, I guess. And here we're using the effect of that entropic stabilization to improve the catalyst stability. So this little tiny writing up here, we have, if we, all these are platinum catalysts, so like about 1% platinum on a support. If the support was just nickel oxide, under reaction conditions, it ends up getting reduced. We make lots of nickel. If we make binary nickel oxide, magnesium oxide, under reaction conditions, it still gets reduced. So this peak here is the nickel that's coming out. And in both of those cases, it also sort of centers into a glob. So C is the nickel glob, D is the nickel manganese oxide separated glob. If we make the 5-component magnesium, cobalt, nickel, copper, zinc oxide, we get the same, rock salt structure support. but now it stays happily as this panel E over here. In panel F, it's the one with the highest CO yield. Panel G, it's the one with the longest stability, and panel H demonstrates the student's ability to keep the reactor system running for 670 hours or however long that is, and taking its samples and doing its thing. Other things we can do, if we have the precious metals or some of the transition metals in the middle that can be reduced, we can produce those directly as metals. And again, we can mix them together, whether they like it or not. And if we do that, just with the pure components, they mix. We're gonna go all the way over there. Okay, they mix, but they're kind of big globs. If we put a support in, so this is sort of a activated carbon-y kind of support, then they nucleate heterogeneously on the support, and we get little tiny particles, of interest for electrocatalysis. These from image analysis are a couple nanometers. Again, interesting things to be learned, especially when these are, multi-component. They don't have to be 5, they could be 2. How really well mixed are they, right? We see, these broadened XRD patterns that only have the peak for one FCC metal phase, but we could do that on that carbon support. We can do it on these iron nitrogen doped carbons that are good for like oxygen reduction reaction supports. We can do it on porous silica, where we generate the support in situ. So we put in the mesoporous silica precursors and templing agents. We put in the metals, and the desired thing comes out. We have to calcine it to get rid of the templating agents, and things pretty much mix. But again, our evidence of that is always just the TEM mapping. We've also made sort of mesoporous materials. Some of this is, most of what I've shown is not published because I don't get my students' papers edited and out the door quickly enough. But some of this is published. So this mesoporous silica work, and that's sort of, that's the support that I was just talking about for the metal loaded mesoporous silica. We can make some MOFs in here, some not so much, right? The temperature's low enough that you can sort of treat it like a spray drying process, and some can assemble fast enough, some can't. We've got some sort of preliminary Prussian blue analogs. That's very early work. You can use these hollow shells for insulation. That's less recent work. We've made some sensors, done some electrochemical experiments with those high entropy alloys. And I think that's the end of the tour, right? So we have this wonderfully flexible method that we've gotten to a point where we can make all these things fairly quickly, turn out new materials. My students in chemical engineering are more motivated by the applications, right? So we spend a lot of time doing catalysis experiments and things like that. But at least for the things that are performing really well, it would be nice to be able to go in and characterize these in other ways beyond just saying, okay, it still looks like one phase of tiny crystals, and things look like they're well distributed, which is mostly what we have. Lots of fun applications of these. So Chow Chow and Jeff, really Chow Chow's been doing almost all the electron microscopy that you saw. Ya Chao and Gong Wu from our department, collaborating with them on the electrochemical stuff. Junjie and Alina on a lot of the thermal catalytic stuff. Lu An and Shenqiang Ren on the insulation applications, and Yao Li and Thomas Thundat on the sensor applications. And somewhere in here, so Shua is the guy here, not him. That's the guy that did all the work. I should have circled him in the slide. And with that, I'm happy to try to answer any questions.
So yeah, let's have some quick questions. Okay, great, go ahead.
So for your high entropy nanoparticles. Can you say something about the stability? Can you then, are they stable enough to, feed them back into the precursor and then put another shell on it, or do they start to anneal too easily?
We haven't tried that, but I think they'll be fine. I'm sure it's system specific, but the ones especially that we're using, like the high entropy oxides that we're using for catalyst supports, like we, heat those to 800 degrees C and run them for many hours, and that's much harsher. That's at least as harsh as the reactor conditions. So it's and even the carbon supports we put in preforms, we can put in like graphene oxide, anything we can sort of get dispersed in water, and they survive the process. So I think the definitely the high entropy oxides will survive. The high entropy alloys I think will survive in general, but they're the ones we're doing. They're the precious metals that are pretty happy to mix anyway. When we first started doing this 15 years ago, we were making a lot of copper silver particles, and those guys really want to, copper and silver really hate each other. They really want to phase segregate. So it's not that everything always mixes.
Yeah. Okay. Any questions here? Okay, so I think we are running out of time anyway.
Yeah, I think we're now one full talk
yeah,
behind.
Yes, so let's move ahead, and we have a virtual talk from Benjamin Xiao from Stony Brook University. Yeah.
Can you hear me?
Yeah, we can.
Well, I want to thank Matt Marino for organizing this workshop. I'm learning a lot, and full disclosure, never done ASAXS before. But I've been doing SAXS on nanocellulose for over 10 years. And when I put down this title, I just put it down the title. Then I realized that maybe I have to say something about end-use research, because the title is From Basic Science to New Circular Solutions. And so I'm also very happy that Stuart gave a beautiful talk. I don't have to explain nanocellulose very well. This is the part you can defibrillate any biomass as long as they consist of this building block of fundamental microfibril filaments. You will have some kinds of nanocellulose materials, and it takes very little energy to achieve these type of nanomaterials. So very recently, we wrote a Chem Reviews review paper, and we have assembled over 800 papers and printed pages, about over 90 pages of this nanocellulose. It's a future sustainable water purification. It's a really good scaffold to host a catalyst, as a sorbent, as a membrane, and as a coagulant, fluctuant. If you have interest in this area, you can take a look. Again, that in this review, we actually talk about X-ray scattering as one of the unique tools to study the behavior of these materials. So, so actually I'm going to talk about it. And the trick about preparing this nanocellulose is really by introducing the charge group. This is an excellent work by Isogai's group. It's a seminal work using tempo oxidation method to only selectively oxidize the C6 position. Tempo is a weak oxidizing agent, so it can selectively only change this OH into carboxyl group. But if you do a proton exchange and you get this salt, so in the neutral condition, these fibers will be charged, and we use this charge repulsion interaction as a way to defibrillate materials. And we've been working on this type of materials for over 5-6 years. Then I come across with a different system using nitric acid as a major component. We call this nitro oxidation process. If you tune it right, you can also defibrillate this materials. The chemistry part is actually complicated, but pretty straightforward. Actually, people start doing this type of chemistry since 1930s to 60s or so. But what's unique that generate my interest is, as you know, that nitric acid is a toxic acid, but it's a key ingredient to make fertilizers. So with this findings, we start exploring using this materials as a way to defibrillate all kinds of feedstock and target specifically all the bio waste. And this is just talking about the typical nutrient cycle. It's a broken cycle. Right now, you spend lots of petroleum-based system, you generate ammonia, you make fertilizers, and most of the fertilizers run up in the water, polluting the system. You eat food, and you eat small part of food. Mostly you end up in the waste and landfill, just where cycle is not a sustainable practice. So we have got quite a bit encouragement from National Science Foundation. For example, the most recent is Convergence Accelerator, who's actually allowed us to pursue this zero waste process. So the solid part of part that cannot be dissolved or pulped by the nitric acid type of solutions, you end up with solid. And solid, you can make them into a gel-like materials because they charge. And the liquid, you can neutralize back to a fertilizer. So it becomes a zero waste process. The process is very cool. Right now, we actually changed the biomass from woods to non-woody plants to even a food waste to even a cow manure. So let's go back to the science. Nanocellulose. Obviously, it's not a fibers at all. This is just from the nano crystal, like in between crystals or fibers. And you can use small angle X-ray to determine the width or cross-section area very well. But if you look at the height can be only about 1 to 2 nanometers or so. Very, very thin. So so it's actually a ribbon-like materials. That took us about quite a few years to really understand why this is the case. And so we have done solution ASAXS measurement. It's absolutely featureless. It's almost like doing protein in solutions. And the only scattering factor we can determine is actually the form factor from the cross-section of this materials. So they're just tremendous of example. They're actually ribbon-like materials. And so this caused us to puzzle for a long time. And until that, we start actually doing small angle and wide angle X-ray measurement using X-ray diffraction to find out. So we actually build an 18-chain building blocks of this microfibril and using all kinds of ways to assemble them. Then finally, we reach a conclusion. They really start with the building block. They assemble them in different ribbon-like structures. So this has been published in the ACS Nano in 2020, if you're interested in it. And this is the typical biosynthesis that you, because the enzyme terminal, this rosette structure, you always have 18 chains. This is a molecular machine that actually takes monomers and make them into cellulose chain materials, and they aggregate into some kinds of crystal structure. And we believe this probably is more representative to what we have observed, and they do aggregate. They aggregate through their hydrophobic interactions, so they form this ribbon-like materials. And this is how defibrillation goes. So it depends on the process. You get different degree of defibrillation process. all these defibrillated materials, they charge. And because they charge their cationic charge, so they're really good coagulants or absorbent that can just absorb and of all the materials. So this is the initial work we published quite a few years ago to remove heavy metal ion. Once they are coagulate, they form this gel-like material. So in the beginning, we only focus on water purification, but throughout the years, and we realized there are other type of application. For example, we start spinning them into fibers and using multivalent ions to cross-link them, because we start learning how to cross-link these materials. We start with aluminum, then iron, then zinc, so forth. Then we realized if we're going to do that, we might as well make them into biogel for farming. And as you know that for farming, if you have a drought, water will evaporate, and we can use all the micronutrients to do that. So, and this is currently where we are. We actually, we have this nitro oxidation produced CNF. We're using different type of cations, and they range from monovalent ions to divalent to trivalent, and we're focusing on different applications. For example, aluminum is a very good cross-linking agent that actually can make this very, very strong gel for infrastructure application. For example, that if you have a coastal area that lots of sand, every storm event, there were just with coastal erosion. So we can actually come up with a gel structure to retain the water and to secure the infrastructure materials. Iron is a micronutrients, and this is another primary, secondary nutrients. So we can start working on using different nutrients to make this biogels as a growth media or as an infrastructure materials. So, and that's the foundation of this larger National Science Foundation Convergence Acceleration Project. Of course, we can also use it to remove heavy metal ions too. And also we're start interested in diffusing nanoparticles and or in situ grow different nanoparticles. And the nanoparticles can be absorbent photocatalysts and catalysts. And what we're good at is small angle X-ray. What we're really also interested in is ASAXS. So I particularly enjoyed today's talk. I never got any experience with ASAXS yet, but my student Jia-Jun is there and learning. And I'm sorry about this. And Jia-Jun is right in the room. And this part of good part of work was a collaboration with Thomas, who's my postdoc before, currently is working in KTH. Part of the research is also funded by Swedish funding agencies. But particularly, I want to thank National Science Foundation for really funding the fundamental work, and to some end-use research like PFI, TT, CBAT to today's Convergence Accelerator. With that, I close my talk. I'd be happy to answer any questions.
Thanks, Ben. So yeah, we can have some quick questions for Ben. Yeah, so if no questions now, I want to ask Ben something about this cellulose. So we did some work with Stuart, as you have seen.
Beautiful work.
Yeah, So there you have seen, like, we have introduced this ions there. So now you are indicating that we can put nanoparticles there too, right? In the similar kind of, not in nanocellulose, but some other kind of systems.
Yeah, we have a student who try to understand between the diffusion versus in situ growth mechanism. We have done quite a bit of XPCS, knowing the dynamics of those particles, try to probe the voids between this network. I think Jiajun attending the workshop in person can tell you more. But we're very much like to see that what ASAXS can help us with.
Okay, thank you. Okay, I think if there are no more. Yeah. Okay, so let's move ahead and thank Ben. Let's move to Cecilia Leal from University of Illinois Urbana-Champaign. Okay, Cecilia, it's yours.
Can you see this all right, or?
Yeah.
Okay, good. So thank you for being here. Thanks, Matt, for inviting me. So similarly to some of the other speakers, we've never done anomalous scattering, so we have only done very normal scattering, but we've done a lot of scattering. So here's an example of diffraction pattern of a material that we've heard about before. So it's actually a pulmonary membrane extract from cows, and that can give, under certain conditions, it can give this really beautiful diffraction pattern. So I'll tell you a little bit of the things that we do in my laboratory, and that has throughout, the last 10 years here at Illinois, brought us to Oregon to do scattering. And I will just start right away by acknowledging the people that go up there. So I have a fantastic group of students that go up to Oregon 2-3 times a year. This is possible because of this phenomenal Oregon staff, and we mostly go to 12IV, and I really want to shout out for Byung-Do and Sanka and Ivan and Xiaobin that have been really amazing at helping our students, and that has resulted in I don't think there's a single paper that comes out of my lab that doesn't acknowledge APS. So what do we do? I just want to sort of like summarize here in 4 overarching themes. We do a lot of work on lipid nanoparticles. We always have, even before they were popular. What we do that is different is that we focus on how we can tune and prescribe certain complex structures to these LNPs, and how that helps regulating and sometimes completely dictating on their ability to deliver a cargo to cells. And in this respect, ASAXS in combination with cryo-EM has been critical to determine the nanostructures, of course, of these systems. One thing that oftentimes we struggle with is really locating the cargo. We try different things, but obviously it would be wonderful to see exactly where DNA is, where RNA is, where the drug is located within the 3-dimensional structure of these LNPs. Completely different and very new topic to my lab. We only have a bioRxiv paper that has been in review for like 2 years, bouncing around. I think it's super exciting. It looks into how diet affects fat packing. And I'm going to tell you a little bit more about this topic as I give two examples in which I think potentially ASAXS could be useful. And this is one of them. So I'm not going to say much because I'll give more details later. The other topic where I think ASAXS could be very useful is this topic of hybrid membranes, where we've done a lot of work on this, where we basically do a co-assembly within the same membrane of synthetic blockopolymers and biological phospholipids. And when you combine them in the same membrane, you get synergistic structural, mechanical, permeation. We see a lot of things basically that if you have a pure polymer membrane or a pure lipid membrane, they have a certain characteristic. When you make a hybrid on the two, they have unexpected behavior. So I'll give a little bit more details on these. And finally, and just because we heard another talk about lung membranes, we do a lot of work on pulmonary membranes, and APS has been quite literally instrumental into many of the advances. So we work with tissue as well as with extracted lung membranes, both from bovine as well as of late from Navy dolphins that mostly die or get very sick because of pneumonia and other respiratory diseases. And with SAXS and microscopy, we've been able to, as well as with, lipidomics. This is a huge project. We've been able to identify critical structural transformations that happen in lung membranes on animals affected with pneumonia. And so that has been a really, really fun project. So I would say that in all of these, in pulmonary membranes, definitely being able to locate possibly protein aggregates that give rise to these structural transformations would be really nice. I would say that in all of these, potentially ASAXS could be useful to us. I'll be honest, I don't know much about ASAXS, but if I go to the UChicago ASAXS webpage, there's a few things that, of course, immediately come to me as potentially interesting. Obviously, the ability to do elemental specificity, that we can locate where things are. We can have, spatial distribution, locate, again, where things are in a biomaterial that is highly complex, right? We have membranes, we have membrane proteins, we have nucleic acids, and we also have structure, extremely rich structure in many of these systems. We can, of course, inspect these things from SAXS to AXS. So those are things that are highlighted here in this page. And then, of course, sample environments, right? So all these samples are biological, and sometimes they are challenged with temperature. Sometimes they are challenged with radiation. Sometimes they are challenged with a different buffer or salt conditions or what have you. But the idea to be able to tune the conditions at which these samples are equilibrated is attractive to us. Okay, so I'll give you two examples. One is purely biological, and that's this business of crystallization of fats in adipose tissue as a function of diet. And another one is in these materials that are hybrid. So they sort of co-assemble a block polymer and a phospholipid into a single hybrid membrane. So we saw some really interesting data here where this is in collaboration with people that actually know the biology. So with Cyanak here at UIUC, what we saw is that, that if we look at this tissue here, if we feed mice with a normal diet, the fat cells, so this is an histology of fat cells, they have a certain size. If you feed the animals with a high fat, calorie excess, not surprising, the cells are bigger now. What was very surprising is that if you feed the same amount of fat, but now you provide sugar to these cells, this is called the Western diet. I didn't name it that way, but it seems fair. These cells grow even bigger. We also, and it was the question of why, what is happening? And there's a lot of things that is happening in terms of genetic cues, which we are not involved in studying, that size domain, but even at a physical or a physical chemical level, we are seeing changes. So the first thing that we see is that this Western diet tissue is a lot stiffer. That's the first thing. We calculated the Young modulus. We also isolate the organelles in where this fat is packaged. So what is this fat? It's mostly neutral lipids, triacylglycerides, sterile esters, and where cells organize fat, all cells do it, is in this sort of organelle that is called a fat droplet or a lipid droplet. For those of us who are materials, chemists, physicists, it's essentially an emulsion. Think about fat being stabilized in the cytosolic, in the cytosol, right? So in the aqueous environment of inside cells, and they are stabilized by a monolayer of phospholipids and certain proteins. All cells, or most cells, have these organelles. Their function is not really well known. They are perceived as the garbage trucks of the cells. But in adipose tissue or in fat cells in adipocytes, they're critical. If you look at the volume of the cells, they are the main component. It's this giant droplet that packs fat, right? And there are different types of fat cells. There's white, which is mostly for fat storage, and there's brown, which is mostly for heat homeostasis. So we're going to ignore that for now. And so when we isolate this organelle, which is quite hard, but we managed to find a protocol to, out of this tissue that we characterize as having really high rigidity with a Western diet, if we extract these organelles and we pull on them, so we build a micropipette aspiration machine, and if you pull on them, you see that it's just like pulling butter. And so what we found by ASAXS was that, in fact, these are not emulsions at all. So there is an emulsion peak, a liquid peak coming from this fat. But what happens with the Western diet is that this fat literally crystallizes on the rim, we think, on the rim of the lipid droplets, and making it really rigid and making it really hard for the cells to metabolize this fat. But it makes it very easy to grow big because, as we all know, if you want to pack more in an object, you just need to make a crystal. So this is highly packed. But what we don't know exactly is what fats are packing. We have very strong evidence by the position of these peaks. If we compare with a lot of the diffraction that is done in fats, food technology papers, we have good indication that these are mainly tags, but really what crystallizes, what kind of lipids, neutral lipids crystallize, and where are they? And are there phospholipids in that crystal as well? Where do the phospholipids go? Of course, we cannot say any of that because we don't have element specificity. So it would be really nice if we could resolve that, and then just teasing out, challenging these organelles with different temperatures and buffers as well for sample environment. So in the next 2 minutes, less than 2 minutes, I'll just tell you the other example that's where I think ASAXS could be useful is again in this business of mixing a hybrid lipid polymer membrane. We've seen throughout, in a series of different papers throughout the years that there's a lot of things going on. We see synergistic phase separation. So that we have three-dimensional phase separation. So we have domains that form in these hybrid membranes that are either lipid rich or polymer rich. And when we stack them together, these domains align in full registry. So polymers align with polymers, lipids align with lipids. And that creates a very interesting material with very high interfacial energy here at the domain boundaries that becomes super interesting to encapsulate drugs, to control permeation of drugs. We've seen synergistic mechanical properties. I don't have time to go into that, but we've seen synergistic permeation of drugs. So polymer only delivers drugs at a certain rate, lipid only at a certain rate. But when we have a hybrid, it delivers faster. And so we've seen also some crystallization, also synergistic, we could tune the orientation of crystals, of polymer crystals when we use polymers that crystallize, and we could see actually synergistic heat transport properties too. So anyways, are there really lipid-rich and polymer-rich domains at the nanoscale, or are they macrophase separated? If we have drugs, where is it located? Where are the polymer crystals when we crystallize polymers? So last slide here is questions, right? Is element specific, but it's really for just high Z atoms. What does that mean for soft and biological matter? All small is the spatial distribution that we can, what's the resolution that we can achieve? Can we do grazing incidence? And okay, we have sample environments, but can we have flow cells? Sorry, I'm a minute late, but if there's time, I can have questions.
Thanks, Neil. And we can take only one quick question.
Well, it was interesting to see that. So this is Matt.
Hi, Matt.
That your last project is supported by CBET. One of our objectives is to broaden the constituency at NSF for interest in ASAXS. So that's a helpful piece of this.
Yes.
There's not much getting around the fact that this is a mechanism, that this is a scattering mechanism that gets contrast based on,
right,
gets information based on the absorption energy of X-rays. So, but
yes.
Often you can infuse these things with metal atoms or other elements that go certain places, and perhaps bring out some kind of contrast in that way.
Exactly. And so, I one of the things very quickly that this CBET grant is actually the polymer component, Matt, is a polypeptide, where now we can conjugate, right? We can conjugate high Z atoms. So I think there's exciting opportunities there. I just didn't want to reveal too many specifics, but definitely something we are very interested in.
Okay.
Thank you, Cecilia.
Thanks. So due to time, we have to move ahead. So there is one more talk before the lunch. Kian Chen from University of Illinois at Urbana-Champaign.
Hello, everyone. This is Chen from UC. I'm Cecilia's colleague. First, thank Matt for the invitation. I actually don't have SAXS or ASAXS data in my slides. Today's workshop is really a mind-blowing experience for me, so I really look forward to becoming a user in the future. So for my group, what my group does is actually electron videography, what we refer to as electron videography, to understand the formation mechanism of shape and dynamics at nanoscale. So our unique capability is that we can actually take movies using electron microscopy at nanometer resolution. We have several different ways to do that. So here I'm showing 2 ways to do that. So one is using liquid phase electron microscopy, which actually allows you to load the liquid sample into the high vacuum of TEM. And then by doing so, you can look at the dynamics, but at the same time at nanometer resolution. And then showing below is the technical electron tomography, which allows you to actually take a lot of snapshots of the 2-dimensional projection of one 3-dimensional object for you to reconstruct the 3-dimensional morphology. So showing here on this in this example, that's actually a polyamide filtration membrane. It has very complicated morphology. We can resolve it at nanometer resolution. So with this technique, we can actually look at a lot of interesting dynamics. So we can look at, for example, how nanoparticles in a suspension can interact and self-assemble with each other. We can look at biomolecular dynamics, which I'll talk more later in the talk. We can look at how polymers adopt different conformation and aggregation state on a highly curved nanoparticle surface. We can actually also look into battery materials, can actually look at how strain propagates, how microstructure propagates as you do the cycling. So for the talk today, I'm going to be more focused on macromolecular dynamics. I have 2 examples. So the first example is a hydrogel nanoparticle. So hydrogel nanoparticle is essentially a polymer network. It has a lot of empty space, so that's why usually we use it in a solvated state. And that's also why traditionally people use scattering methods to actually characterize the size and shape of those hydrogel nanoparticles. Of course, at the same time, you can also use transmission electron microscopy. And usually how people characterize them is you freeze the hydrogel sample and load it in. So by doing so, you can still have a very beautiful view of the size and shape, but you don't really have dynamics. So that's why we see a unique opportunity for this electron videography method using liquid phase TEM. I want to mention with TEM, now what we can do is while imaging the dynamics, we can also add electrical bias. We can actually do flow cell experiment. We can also change temperature. Okay, so this is just an example we published earlier where we can actually use a liquid flow to trigger this crystallization of nanoparticle sub-assemblies. And then in this unpublished work, we apply it to a hydrogel nanoparticle. So the hydrogel particle we use is polylipin. It has about 5% cross-linking density. It's dispersed in water. So to begin with, through a lot of optimization, we can actually get liquid phase TM images of those polylipin particles in liquid suspended in water. And then in this other movie I'm showing is when we do the imaging, we can actually also increase the temperature. So for polylipin, as you increase the temperature above the critical temperature, you actually collapse its volume. So that's what we observe. So with this kind of real space information, what we can now try to retrieve from these TM images is this very well-established picture by the picture for a really long time. So when you think about polymer networks, there could be 2 types of structure features. So one is essentially composed of a distribution of blobs of different sizes. The blobs could be cross-linkers, could be entanglements. And another aspect is this is a network, so it has network topology. So we hope that we can actually use TEM to actually see this directly. So the way we process our data is we start from our raw liquid phase TM image showing here. And then we can do electron optics principles to convert this intensity map to a local mass density map showing here. So in this map, you can already see some fluctuation in intensity. That means it's actually heterogeneous. And then what we can do is we can apply this blob analysis that people use in computer vision. So this can give us essentially decompose a photo like this sunflower photo here into a distribution blobs. We can also get a fingerprint out of this image. So we did this to our image. We can actually get a blob size distribution out of liquid phase TM image. So I want to just mention the size of the blob actually matches with the expectation. And then we can also apply the same analysis to our liquid phase TM movies. So for this one, we already established that within the nanogel particle, the structure is actually heterogeneous. So it turns out this collapsing induced by temperature is also heterogeneous. So that's the discovery here. And then how I see ASAXS can help is that for all this imaging here, we don't really have chemical composition. So if eventually we want to relate the structural heterogeneity back to composition, we need a way to actually map out composition. And also, as I mentioned, our way of doing this in situ characterization is kind of limited. So it would be great if we can actually do it on a more ensemble level to eventually relate what we observe with the synthesis condition and also with application conditions. So this is the first example. And then the second type of example is a more complicated macromolecule, which is a protein sample. So similarly, the motivation for us to actually to image a protein sample is because protein dynamics is actually very important for a lot of applications. And then using our electron videography method is better than cryo-EM because we have dynamics. It's also partially better for the super resolution optical microscopy because the resolution is higher. It's also can be also more advanced than AlphaFold because AlphaFold does not predict dynamics. So the system we study is membrane proteins. You have heard that quite sometimes, but now it's very important in our human body. It's pretty large, pretty bulky, not really easy to characterize using other methods. And then specifically for our electron videography method, we have this sample, have this membrane protein in the form of nanodiscs. So this is a very smart way to actually have your membrane protein coated with lipid bilayer, which is closer to the native environment, and then circle it with a belt protein. So by doing so, you can actually have this single nanodiscs, each with one single membrane protein and then suspending in solution. So with this liquid phase TM technique, what we use is actually a graphene sandwich. So here, this is actually a zoom out view. By zooming out, still the scale by is 100 nanometer. So you can see a lot of species rotating in the solution. So that's actually many nanodiscs. And then we can zoom in. And now the scale by is 20 nanometers. So you can really see a nanodisc rotating to really show different projection view to show this is a really disc-like shape. So we can do some analysis. For example, we can look into the detailed profile of this liquid phase TM image to recorrelate with molecular dynamic stimulation to actually get some structure details, like what is the position of the membrane protein and how they diffuse within this nanodisc. We can also really discover that this nanodisc itself is actually very dynamic, it's fluctuating. That's actually partially because the lipid bilayers, the molecules are diffusion, and also these membrane proteins are also diffusion actively. So we can tell this from this boundary fluctuation of the nanodisc. And then what more we can do is we can zoom in into the boundary of fluctuation, quantify this boundary fluctuation into a series of Fourier transform modes. So I'll skip the detail, but just highlight on this diagram here. So this diagram is our experimentally mapped Fourier transform modes as a function of the wave factor. So out of this, we can actually see, first of all, our experimental data matches with simulation. Second, we can actually see very distinctive power law dependence region. So that power law dependence region can actually allow us to fit parameters from our movies, such as the effective surface tension for the fluctuation and also the bending modulus of the protein. So for this, you can see there are a lot of open questions because all that we get is just electron microscopy movie. So for example, what is the interaction between the membrane protein and lipid molecules? How to rethink about the diffusion? And another practical questions as we do such imaging for biological molecules using electron beam, one question is always what is the invasiveness? What is the damage? So it'd be great if we can have another correlative method to actually show on the ensemble level, our conclusion is actually relevant and important. Right, so that's basically the two stories I have to tell. Right, that's my group, but let me come back. So I really feel there will be a great opportunity here to actually conjugate those two ends of nanoscopic imaging. So on our side, we have high spatial resolution, but at the same time, we have limited sample size. We don't have chemical sensitivity. And for ASAXS, that's a perfectly complementary. Yeah, thanks.
Thank you. So if you have any, yeah, go ahead. So when you do the construction
are you tracing one single particle? You do the average for the sample.
Yeah, we do both. So if so with liquid phase TEM, if you just trace one single particle, sometimes that one particle can actually rotate for a lot of different orientations. That can be enough for reconstruction. That actually can work really well if your sample is heterogeneous. But if the sample is relatively uniform, we don't need to push that far for really long movie, and then we can just do average, do the reconstruction based on average.
Yeah.
So it's interesting to see what is the Q to the fourth power of that dynamics is.
Right. So
Yes,
it's actually based on this equation shown here. So the Q to the -4 power is actually related to the bending modulus, which is actually the belt protein bending modulus. And then -2 is more like an effective surface tension term. So you can imagine for the bending modulus is a molecular property. So it's actually more manifested in the small length-scale fluctuation. That's why it's actually following this power law dependence on the high Q region.
Thank you.
Okay, so we should conclude this session then, and thank all our speakers. And we are going for lunch now, and we are already 15 minutes late. So we'll anyway have lunch in quick 45 minutes. And there is one thing at 2:15, 2:15 to 2:30. Another speaker who could not come due to medical reason. So we'll have a photo session that time. We'll take some pictures.
And we probably don't need that second break. I mean, we could take a break around then and then and finish up or something like that.
Okay.
Lunch is out here, right?
Yeah, lunch is just outside. Okay.
And people online, if you want to come back at 1:30.
Yeah. See you at 1:30.
Which one they want?
Hello, Natash. Hello, Bob. These are folks I know.
Hey, Mark.
Bob, you can hear me. That's good. That's test one.
Test one works.
If I actually try to share my screen in case that is applicable, all that looks okay?
Looks great. Yep.
Okay, that sounds good too. Great.
Perfect.
Dare I start my video? Is that me?
That's Natash. I just
Hey,
wanted
there's
to-
Natash. Okay, I see and recognize Natash. Okay, looks good.
I'm going to also just check my screen share.
Can you see the room, Natasha? This is Matt. So,
Yeah, I can very clearly.
okay.
Everything has been working out. So
Okay.
yeah, I think this works as well.
Yeah, looks good.
Good.
Yeah.
You look the same as when I talked to you last week. So,
Yeah, some things don't change. Yeah, I was noting your absence. Now I know why.
yeah, I was. Hey, Mark.
We haven't seen each other in a while, Matt. It's Mark Hillmeyer. Hello.
Yeah. Hi, Mark. I
Hello?
said hi. Hi.
Have the participants who have been presenting, Matt, been sharing their own screen?
Yes.
Okay.
People are slowly coming back in from lunch, Mark. I'll get them going. So,
Sounds good. Thank you.
Yeah.
Is that microphone pointing that way? That's the only one on mute now for the in person? Yeah. Since we have everything else muted. Yeah. He just came on it and tested this.
everybody ready? So the first after lunch talk is by Mark Hillmeyer from the University of Minnesota. So take it away, Mark.
Thank you so much, Matt. Thank you for having me participate in this event. I'm happy to tell you about work we've been doing on polymerization reduced microphase separation toward ion-containing materials. The work is for my group in terms of membrane materials has been motivated by global water issues. And it's estimated that about 4 billion people experience severe water scarcity during at least 1 month of the year. Block polymers will not solve that, but they could contribute to solutions, for example, purification of wastewater or ultrafiltration to rid water of bacteria and viruses. And the idea really with block polymer self-assembly is to ultimately get something over here, if you can see my pointer, what are referred to as isoporous membranes, where each of the pores is basically the same size, which can lead to both high fluxes and high separation factors. In fact, plotting separation factor as a function of water permeability, one can predict that at ratios of surface porosity to membrane thickness, you can follow this blue line, which allows for very high fluxes and very good separation factors. And these are some approaches here in the colored symbols that are trying to get toward that. In fact, some of this is being pursued in startup companies. That's always intrigued me, and we've been pursuing our own efforts in the nanoporous block polymer realm. It led into an interest in putting ionic materials into polymeric membranes, and in particular, a class of materials called charge mosaic membranes. And the idea is that under high pressure water feed, if you alternate positive and negatively charged domains, is that under high pressure, a salt-enriched permeate can come out of a charge mosaic membrane. And without much detail here, I will tell you that the flux of salt across that membrane is inversely proportional to the domain space. So the idea of getting domain spacings where you have alternating positive and negative charges in a self-assembled material could be interesting for a charge mosaic membranes. And there's some very early examples that are supportive of this, this potential. Well, we took this approach of trying to put a pre-anionic block and a pre-cationic block. It's tough to have them both anionic and cationic because, of course, they'll, in solution, make a coacervate, have them self-assemble, and then do a post-polymerization functionalization. And David Goldfeld, as a PhD student in my group a couple of years ago, tried to look, and this bottom block polymer is what I want to tell you about. This sulfonate ester buffered between a benzyl chloride unit using this chlorinated styrene, in principle could self-assemble to have domains, distinct domains of the blue and the red. And I think the clever idea that David had is that with a single reagent, in this case trimethylamine, one could turn the pre-anionic block into a polyanion and the pre-cationic block into a polycation with one reagent in one conversion. I'm not going to tell you about much more about this other than it was very difficult to prove that these things self-assembled in that way, but we had great evidence that these post-polymerization functionalization reactions worked well. This led David into a little more straightforward process for making domains bicontinuous, in this case, domains of a neutral block and a charged block. And in this case, we used what I refer to as polymerization-induced microphase separation, where the idea is you have some polymer that's dissolved in a monomer, and in one process, you polymerize the blue monomer to make a block polymer, but also have some cross-linking so that the structure that emerges is chemically fixed. And the idea was, again, to use pre-cationic, I'll call them, or pre-anionic polymers to make charged materials. You were able to make these kind of charged materials, and this small angle X-ray scattering says they look like they're self-assembling on the nanometer length scale. These are, we know from other studies, bicontinuous structures without much long-range order. But what was interesting is that if you made this kind of cartoon structure here where there was a polycation in the orange, those monolithic materials, transparent because of their nanostructuring, were able to uptake, in this case, anionic dyes through exchange of the anion chloride for the dye. So we felt comfortable that we're able to do this using a polymerization-induced microphase separation strategy. And what I want to tell you about today is trying to do two things, really, is taking that strategy of a pre-cationic polymer, but combining it with the idea of nanoporosity. Now, the nanoporosity that I was referring to earlier, in our case, typically comes from a sacrificial block. So getting a self-assembled, in our case, again, bicontinuous self-assembled structure in which one of the blocks, this case red, can be removed through treatment of a chemical reagent, in this case, a simple base. That will remove the red and leave nanoporosity. So I want to first show you that, and then I want to show you how the combination of nanoporosity and incorporation of charged materials could lead to some charged polymers could lead to some pretty interesting materials that ultimately we think could be applied in, for example, desalination of water in a charged mosaic membrane. The story continues, but I'll tell you where we are to date. So we use, I don't know if the movie is showing, but the movie is basically a suspension polymerization taking water and monomer, except this monomer has polymer dissolved in it. So you might imagine styrene divinyl benzene, low molecular weight organic liquids, a peroxide, you would make cross-linked polystyrene. However, if you dissolve in this macromolecular chain transfer agent that uses raft polymerization by virtue of the trithiocarbonate, you can polymerize off of this PCl chain, generating polystyrene PCl that cross-links during self-assembly in these suspension particles. The idea would be then those particles could be etched to leave nanoporous beads. So I'm introducing, instead of monolithic materials like I showed you in that dye experiment, bead materials, which could add to some interesting advantages for separations. We made those beads. It works pretty well. Their size distribution is tens of microns to a little over 100 microns. This is the as prepared beads in the upper left. And when you put in, sorry, that's without the PCl, excuse me, just the opposite. This is with the PCl and without the PCl chain transfer agent. That doesn't really factor into how the beads come out. And when we look at the ones with polycaprolactone by confocal Raman spectroscopy, we can see that in these materials, the PCl seems to be evenly distributed through the bead at this long length scale, and so is the polystyrene. So this is all consistent with microphase or nanophase separation in the material. Here are the beads, again, now after removal of the PCl. So this is an SEM image. These SEM images, you can tell these beads are, maybe 100, maybe 20 to 100 microns. And the PCl has been removed. We know that by looking at IR spectroscopy. The carbonyl stretch for the PCl is no longer there in the etched beads. And by optical microscopy and scanning electron microscopy, they retain their shape and their structure. However, there's no PCl in them. So when we look at small angle X-ray scattering, we depending on which polymer we use, under which conditions and how much PCl we use, we do get domain spacings here that are on the order of 20 or so nanometers. And depending on what molar mass PCl you use, you can change the domain spacing in the material. And the domain spacings are consistent with these SEM images, which we took, which I'll show you, excuse me, in the next slide, that show the nanoporosity. Nanoporosity was also confirmed by an etching experiment. And using nitrogen adsorption. So the unetched material has very little surface area, but once you remove the PCl, you're leading to hundreds of meters squared per gram surface area. And the differential pore volume analysis says that's consistent with all the PCl being removed. Well, this is what I was referring to. This is what the beads look like. On the surface, you can certainly see porous materials. And when you do a cross section, you can clearly see that through the material, you're generating porosity by virtue of removal of the PCl. So now we have a spherical particle, 100 microns in diameter, that's nanostructured, that's been rendered porous by chemical etching. And so now to combine the two possibilities of nanoporosity and having a pre-cationic, in this particular case, domain, is that instead of doing the polymerization-induced microphase separation with only a PCl raft agent, we made a PX PCl raft agent. In this case, it's vinyl benzyl chloride. And then we put that dye block as the chain transfer agent and made dense polystyrene, but now the other domain is actually a dye block. Once you remove the PCl in gray, now you've decorated the walls. You've decorated the walls with the pre-cationic polymer, in this case, polyvinyl benzyl chloride, which we treat with triethylamine to render it cationic. And here's the chemistry. That's the block polymer that we use to put it onto the cross-linked polystyrene. In this case, we're using triethylamine in water to displace that chloride to generate the polycation. Also removes the PCl at the same time. And I'm going to tell you only about the results for the cation, but we can also do it in an anionic sense. We remove all of the carbonyl, so all the PCl is gone. We see some evidence of carbon-nitrogen stretches from the functionalization. And then when we look at uptaking this dye, I'm going to go over this briefly, but basically at very low concentrations of dye, the dense beads take up almost no dye. But if you make them nanoporous or you functionalize them, they can take up a lot of dye. Now, in this specific case here, the polymer has not been rendered cationic. So there's clearly some non-specific dye absorption here. And so we decided to change the concentration of dye that we were probing, increase its concentration by a factor of 100. Now, this is methyl orange that's at 3 millimolar. And the punchline of this presentation is that if we compare our beads that were quaternized only without removing the PCl, yes, they will uptake some dye. If you take amberlite, which is a polycationic porous resin material, it also takes up some dye. However, if you combine nanoporosity and high cationic charges, you can imagine the mass transport limitations are reduced. The cations can do, polycations can undergo exchange, and you rapidly take up all of the dye in solution. And here's a shorter time scale to show the rapid decrease in dye concentration over time with our nanoporous cationic materials. In fact, really orders of magnitude greater rate constants of uptake. Moreover, our capacity the expected capacity was about 405 milligrams based on the number of cationic charges, and we got something close to that, and that's almost an order of magnitude higher than amberlite. So this story was meant to tell you that we use block polymer self-assembly, in situ block polymer self-assembly, to generate nanoporous materials that their pore walls can be readily functionalized, in this case, with polycationic or polyanionic domains, ultimately leading into, this was a pretty simple dye sequestration experiment, leading into hopefully making membrane materials that include both polycation and polyanion dense materials. It was Colin and Jay and Hyung K who did this work. It was published last year, and I thank the Department of Energy through the BES for support of this program. I'll stop sharing, and if there's a moment for questions, I'm happy to entertain them. Thank you.
Thank you, Mark.
I have a question here on Zoom, if I can.
Please. Go ahead, Cecilia.
Yeah, I'm just curious about the state of that PCl. Did you do wax? Is it crystalline in any way?
In some cases, yes, it depends on the domain spacing. I'm not remember this exact, I'd have to look at the exact analysis for this particular one. But yes, you can actually see a crystalline peak in wax, and also in DSC, you can see melting. However, that melting transition can be depressed because this crystallite size is confined by the nanostructure domain. So you get very small crystallites, which have lower melting points. So we can use DSC in melting to actually as a proxy for size of the domain. So it's a nice reporter that way.
And do you know the orientation of those microcrystallites?
I do not. This is a very good question. People have studied semi-crystalline block polymer self-assembly, whether the stems are parallel or perpendicular to the face. I don't know the answer in this case.
Okay. Thanks, Mark.
In the charge mosaic ones that you showed at the beginning, the schematic, Mark, is it either imposed by the macromolecular architecture or hypothesized that there's a regular arrangement of both the positively and negatively charged channels?
The idea would be imposed by the architecture of the tri-block. So you'd have A, B, C, and you would just naturally expect the B to be between the C and the A domains. The reason I didn't show you much of that, Matt, it was very difficult to understand what the self-assembled structures were in those tri-blocks. I don't think that the organization is going to matter that much, only that there are positively charged domains that are adjacent to negatively charged domains. The crystallographic order of those, I don't think will matter all that much.
I mean, it does strike me that there's an ASAXS experiment here for at least looking at the negatively charged domains by binding with some cation that X-ray absorption energy.
Absolutely. For example, yes, agreed.
Thanks a lot, Mark.
Thank you.
I think we go now to Robert Moore from Virginia Tech.
That's correct. Can you hear me and see the correct slide?
Yes, we can.
Okay, let me see if I can get my system to it. Sorry. Is it still the correct slide?
Yep, and we got the right presentation view too.
Okay, great. Perfect. That's good to know. Okay, well, Matt, I'd like to thank you and Myrna for organizing this workshop. I've learned a lot. I haven't done ASAXS since, gosh, the late 80s, when we were looking at random ionomers back in the heyday of random ionomers. And that'll be the subject of my presentation today. Okay, so when you put ions on the backbones of polymers, you get this really interesting morphology. Let me get my pointer here. Let's see, there we go. Where the ions aggregate and they create these multifunctional physical crosslinks in the material there. We know from small angle X-ray scattering that we get structure associated with these ionic aggregates. We also see a really interesting upturn, and that's going to be the subject of my presentation today. But also as a result of this nanophase separation, you also get really interesting biphasic behavior where we have regions of restricted mobility in this model that I developed back with A.D. Eisenberg long ago. The regions of restricted mobility create a high TG phase, polystyrene well separated and removed from the ionic aggregates is just the matrix material having its kind of standard behavior. Okay, so the hierarchical morphology of these types of ionomers stems from the electrostatic interactions, of course, that cause the formation of these multiplet structures here. And then in long range, we have for random ionomers, a rather random distribution of these multiplets in the matrix. But this cluster phase here is really what I've been interested in for quite some time, because you think about membranes, ion-containing polymer membranes. You need this percolation pathway to transport water and ions across a membrane. Perhaps this cluster phase, this heterogeneity, long-range heterogeneity, is responsible for that desirable characteristic of ion-containing polymer membranes. So long ago, there was quite a bit of interest in the small angle upturn and the origin of this upturn. It continues into the ASAXS region. Models have been employed to kind of estimate the sizes of the long-range heterogeneities. In this particular study, the heterogeneities were on the length scale of about 130 nanometers or so. For this sulfonated polystyrene system that we have here. But these correlation lengths vary significantly over different Q ranges that you're looking at here. So there's still a lot of question as to the precise origin of that. So that led to people employing ASAXS. Was this long-range heterogeneity associated with the ions, in a heterogeneous distribution of those ions in the materials? These studies prove that, of course, the ionomer peak was associated with the ions. We kind of knew that. It's well known. But the small angle upturn here is still present in this difference pattern. So it is associated with, in this particular case, for a sulfonated polystyrene system with the long-range heterogeneous distribution of the nickel ions. Now, is that the neutralizing salt that gave rise to that? Was it voids? Well, ASAXS proved that it's not voids. The initial assignment of the small angle upturn was a inhomogeneous distribution of the neutralizing cations, as you can see by these dots that have here. So long range domains that exist there just from the random neutralization of these materials. But later on, it was suggested that, well, it could be an inhomogeneous distribution of the ionic aggregates as well. And this has been kind of the focus of some of the things we've been interested in. I've also been very interested in semicrystalline ionomers from the Nafion type materials, of course, Serlin here. ASAXS really gets interesting with semicrystalline ionomers in that you have an electron density distribution profile shown here from the crystalline domains, the amorphous material containing the ionic aggregates. And then, of course, if you go across one of the ionic aggregates, you see a spike in the electron density there associated with the counterions that are neutralizing that material. So ASAXS provides the ability now to determine the sign and potentially the magnitude of the electron density differences here in these different morphological features. And so this is essential for the assignment of these features, knowing that information. The scattering attributed to the polyethylene crystallites are also clearly distinguished from the scattering attributed to the ionic aggregates. And this is really where ASAXS is extremely valuable for this class of ion-containing polymers. So here showing here near the nickel edge, the ionomer peak decreases due to attenuation. And the contrast across the crystalline long period that you're seeing here effectively increases as you see the attenuation near the edge. So ASAXS has proven in this particular case that the electron density of the crystalline component is greater than that of the amorphous component. And this is the average electron density, if you will, of the amorphous containing material that we have there. So that brings me to the subject of my presentation in the types of ion-containing polymers that we're making, and it gets us to taking a look at gels, polymer gels, semicrystalline polymer gels. The model system that we're looking at here, I can move this thing, is syndiotactic polystyrene. It's a stereoregular form of polystyrene that is very high tech. So it's a beautiful model system. Much of what we've known about ionomers is from atactic polystyrene. So this is a beautiful model system for semicrystalline ionomers that we're making in our lab. The gel network, when we create a gel, we dissolve in a solvent at high temperatures, you cool it down and it gels by crystallization. So the semicrystalline network that you have here is linked together by these amorphous chains that trap the solvent in that gel structure. And oops, let's see what happened here. And the chemistry that we're doing in the gel state is shown here. If I have a homogeneous solution and I add a functionalizing reagent, such as a sulfonating reagent in this particular case, those sulfonating reagents can access every monomer on those polymer chains, and you end up with a random copolymer. But if I first allow the system to gel, so go across here, now allow the system to gel, then add my sulfonating reagent, it can't get it, those reagents cannot get into the crystalline component of the gel, and you only functionalize the amorphous chains over here. So that leads to a blocky structure, a very simple way of making block copolymers, if you will, where I preserve the crystallizable segment between these highly functionalized regions of the amorphous chains that originated from the gel structure that we have there. And that's going to be real important to us in the membrane area where we want crystallizability to limit the water uptake, for example, but we also still want a very high ion content to facilitate transport in these materials. And so we've done a lot of work with this model system. This is bromination of syndiotactic polystyrene, looking at the random 31 mole% brominated system versus a blocky. C13 NMR is a beautiful textbook case of polymer sequencing. We've been able to map out all of the triads and pentads and know exactly what these peaks are associated with. Simulations of these chain structures then that exactly fit the NMR spectra are shown here. You can see clearly random type microstructure for the random solution state functionalization, but the gel state functionalization, this is what our blocky structures really look like. Again, preserving that crystallizable component there and also grouping up the functionality that we have there. So now back to ionic polymers, comparing, looking at the random sulfonated atactic polystyrene. So this was formed in solution. We neutralized with cesium ions to give us a lot of good scattering contrast. Here we see at 3, 6, and 10 mole percent, we have a prominent ionomer peak, as we'd expect. We've seen this many times with standard atactic polystyrene, sulfonated atactic polystyrene ionomers there associated with the ionic aggregates. But now we start to see some really interesting structure here at low Q values. And we're wondering, could this be the cluster phase that we're seeing now in these systems that we have there? Okay. Getting back to ASAXS that was done on sulfonated atactic polystyrene many years ago, looking in this region with this particular data set, everything's kind of jumbled together, but there might be something there. I'm wondering if they were seeing that feature as well in their ASAXS data that we see there. We've done some estimations of the domain sizes of this feature that we have here, still not knowing what the origin is, but if it is the heterogeneous distribution of these ionic domains that we have here, we're thinking that at low ion contents, we might have these large domains that look like this, but in a greater density of these multiplets, at higher ion contents, perhaps we have smaller domains that look something like that. Now, comparing the random system to the blocky system gets really interesting now. The random system behaved just like we've seen for many years for typical ionomer behavior, increasing the scattering feature associated with the ionic aggregates as you put more ions on the backbone of the polymer there. But we really have a diminished ionomer peak in the blocky structure. And we don't see any features associated with what is potentially that cluster phase that we're looking at there. So no clusters were visible in this system, diminished intensity of the ionic feature. And then at high ion contents, we also see this multi-feature here. We don't know what that's attributed to. Is that associated with the ions as well? This is right in the vicinity where you'd expect to see some crystalline scattering, but at this high ion content, these materials are completely amorphous. For the more crystalline systems, we really don't see that much. So ASAXS would be perfect for helping us to try to understand what these morphological features are associated with. The behavior that we observed with our model syndiotactic polystyrene system is also essentially the same when we go to backbone materials that are a better choice for fuel cell membranes, for example. We've been, the field's been sulfonating PEEK materials for many years. We wanted to see if we could do this in our gel state, and certainly we could, to make a blocky speak versus a random speak. When we look at our small angle X-ray scattering, we see for the random speak a very prominent ionomer peak that we have there, but a greatly subdued ionomer peak that we have there. A much stronger small angle upturn in the blocky speak too is another interesting aspect of this that we hope to probe with ASAXS. Really the reason that I'm so interested in this small angle upturn and these really low Q value information features is getting back to those transport properties again. When we look at our sulfonated PEEK membranes, the proton conductivity here versus the degree of sulfonation, this is what the field has been doing for many years, sulfonating PEEK and getting conductivity values that are, you know, about half that of Nafion, the benchmark material there. This lambda value is the moles of water per mole of sulfonates. So we have a lot of water. It's about to dissolve any higher than that because it's not crystalline. But our semi-crystalline blocky microstructure now has a much lower water content because those crystallites impede the swelling of the material. We also have conductivity now that's approaching Nafion. I can tell you now that we're actually exceeding that of Nafion as we improve our synthesis. But it's really, we believe that that long-range heterogeneity that's likely giving rise to this enhanced proton conductivity in these materials. So to wrap it up, you know, these ionomers continue to have very interesting scattering features and scattering questions. We looked at with ionomers ASAXS, but then it's been, a few decades since we've really looked at this again. So I'm really grateful to this workshop for making me think again how we might be able to resolve some of these complex structural questions, particularly when we start affecting the microstructure of the materials. Are we seeing scattering features associated with clusters? That's pretty cool. First time to ever see that. How does the microstructure of the backbone affect the development of these ionic domains, and how are the ionic aggregates then distributed throughout the crystalline domains? Certainly, we're interested in employing ASAXS to help us resolve these questions, and I'm particularly excited to know that you can do cesium, because that's what we do all our scattering with. So I think that'll really answer some really interesting questions there. Okay, so with that, I realized I didn't have my video on there. I'm sorry about that. I wasn't hiding. Happy to answer any questions.
Thanks, Bob. And I think we'll give you a much better instrument to work with in a year.
I think so.
You have a question here.
Hi, Bob. So that feature that you're trying to link to some sort of structure, in our inorganic systems, we see that a lot for kind of donut shape or ring structures. Is the chemistry conducive to formation of rings?
Well, I don't think so, but there have been some early studies of kind of vesicle type structures of the ionic aggregate itself. But that longer range heterogeneity, I don't know, but I don't think so. We don't have the precision, really, I don't think, in that structure.
Anything else? Okay, thanks again, Bob.
Sure, thank you.
And we'll turn the floor over to Natasha Balsara at Berkeley.
Thank you, Matt. I'm getting my ducks lined up here. Far less organized than my friends.
You're doing okay.
Before me. Okay, so I'm here to tell you a relatively old story. I have not used anomalous scattering for a while, and I thought I'd tell you about the morphology of block copolymer films as we were able to see using what we call resonant soft X-ray scattering, or ASAXS. And this was done with Jeff Courtright, who is, of course, a leader in the field of anomalous scattering. We did this work under the support of the Office of Vehicle Technologies at Department of Energy, and I have joint appointment at UC Berkeley and Lawrence Berkeley National Lab. I'm going to tell you about a block copolymer, polystyrene, polyethylene polystyrene. We call it SES, and these are the molecular weights in kilograms per mole. The volume fraction of styrene is about 25%. And not surprisingly, because of this triblock has a lamellar morphology as you determine by small angle X-ray scattering. What the Department of Vehicle Technologies wanted us to do, or the Office of Vehicle Technologies, was to see if we could make separators for batteries that were better than the ones that you can buy. And the approach we used was to take a homopolymer, polystyrene, add it to the block copolymer, and then see what happens, keeping the void fraction at 0.43, which is the volume fraction of the added polystyrene, and then we would wash the polystyrene out with some series of solvents that none of them attack polyethylene, which is crystalline at room temperature. And so our objective was to make things with 43% voids. And the main variable was the chain length, the homopolymer chain length to the block copolymer chain length that we used. Learning from a lot of previous studies, starting with Ken Cho and perhaps even Ludwig Leibler, telling us that if your chains were very small in chain length, like 0.13, 10%, then 13%. Then they would be in both phases, and they would start getting aggregated into the phase that you want at reasonable values, like 0.22 and 0.36. And these are SEMs of the washed films that we made after the homopolymer was extracted. So really the structure that we are looking at is a polyethylene garbage bag with holes in it, but the holes are lined with polystyrene. Looking at Mark Hillmeyer's talk, one can imagine lining these pores with more interesting materials. But for the moment, I'm going to tell you about the polystyrene story. Want to acknowledge that this is, many people use similar approaches, and we were, standing on the shoulders of these folks as we were working on it. Here's the, if you soak these membranes in an electrolyte and measure conductivity as a function of this alpha parameter, you find that you get reasonable conductivities, or good conductivities actually, in a range of alpha values. And unfortunately for the Department of, it turned out to be exactly the conductivity that you get when you soak the commercial state of the art. So these materials could not exceed what you can buy, but nevertheless taught us something interesting. So in anomalous scattering, what one does is vary the energy of the incident X-ray beam. And I'm showing you data using 10 keV, which is just a simple ASAXS, along with some resonant soft X-ray scattering data from the same film at different energies. And what these different energies do is allow you to contrast match different parts of the film. These contrasts are calculated here based on this wonderful paper by Harold Ade and co-workers. They are given as a function of the energy over here. Energies of 300 and greater EV are basically ASAXS, and you get a big contrast between the two polymers and void. So what we are contrasting is polyethylene and polystyrene with this dashed little red line, how it would change with energy, and then polyethylene and void, which is this blue line that goes down and then goes up, and then this other dashed line, which is polystyrene and voids. And this is a log scale for the calculated X-ray contrast as a function of energy. And you can see two interesting energies at 280 and 284 EV. At 280 EV, you're essentially wiping out the contrast between styrene and the void. And at 284 EV, you're wiping out the contrast between the polyethylene and the void. And then, as usual, if you go to the hard X-ray limit, you would start seeing the contrast between the styrene and the polyethylene would disappear. So, so these three energies give us different views into the same material. And of particular interest is this 284 EV work, where you actually can now hopefully begin to see the domain structure of the block copolymer in the presence of the void. So you don't get your signal doesn't get wiped out by the presence of voids. And that is this lowest curve here that shows this peak that in fact actually is quite coincident with the block copolymer peak. While it sort of disappears when you don't have this contrast match condition. Jeff Courtright and I thought about what we should do when refractive indices are imaginary. We knew we wanted to get a real answer, and we just proposed that we can take the refractive index and its complex conjugate to calculate the various contrasts that I showed you in the previous slide. using RSOX, we could tell when the templating was successful and when it wasn't successful. For example, this is an alpha of 1.65, which means the homopolymer molecular weight exceeded that of the block copolymer. And here you see essentially just one peak corresponding to the block copolymer, and that's this analysis of domain spacings, for example, is shown here as a function of this alpha, the sacrificial homopolymer. The domain spacings that we get at 1.65, is that the domain spacing that we get from resonant X-ray scattering is identical to ASAXS, and this agreement actually tells us that the templating has failed. When the templating succeeds, we get two different length scales from these two different experiments, reflecting the porosity of the material as well as microphase separation. And that's really the moral of the story. The complete data set is shown here as a function of that alpha parameter. And what you see is that when you get very long chains, you get macrophase separation. What I think, to me as a scattering person, what was interesting is that we could study microphase separation in such a material in this polymeric domain without too much interference. It turns out, of course, that these cartoons are simplistic. There's all sorts of phase mixing and broad interfaces, and so contrast matching never works perfectly, and that's a subtlety that we deal with. In fact, we were looking at microphase separation in that little region, in those macrophase separated block copolymer regions. So in conclusion, I wanted to say that RSOX is particularly useful when there are more than two components, and contrast matching is possible. It's not always, the energies may not always be available. They sometimes can be too destructive from the point of view of beam damage and so on and so forth, but it is possible in some cases. If you want to study mesoporous structures with lining that is different from the support, RSOX is one approach. Thank you.
Yeah, I mean, what Nitash pointed out is that this can be done to some effect with soft X-rays, and the element you're looking at is carbon. It's a little different kind of scenario, but it certainly is the same principle that we're talking about here. So, any questions?
I actually have one, if I could.
Go ahead.
Hey, Natasha, great talk. You got me excited about something. So I'm wondering, and I didn't quite catch it, with really highly porous materials, how applicable is this? With our gels, if we extract the solvent, we have an aerogel, but I'm very interested in the crystalline component within that phase. Could I do the same type of thing there?
Absolutely. In fact, in this, I don't know if I have this plot, but it turns out that when we contrast match everything except the polyethylene, we see two bumps. And then when we heat it above the melting point of polyethylene, we see one bump.
Wow.
Okay. We just made a statement about that in the paper because we thought it was somewhat boring, and but indeed you can see crystallites, and of course the crystalline polyethylene will have and the length scale sort of matched up with folks like you have published. So yes, you can see those subtle features in spite of the presence of the voids.
Excellent. Thank you.
Anything else? Okay, thank you, Nitash.
Thank you, Matt.
So we're up against the missing speaker, right?
We can have this photo session now.
Is there any chance that we could get the photo array of the people online and we could take the picture up here or something like that?
You'll get a lot of names.
Stay on, Cecilia and you guys and whoever else we have. I don't know. So I don't know if we can get all of us in front of that screen, but
Would you like me to take the picture for you? Oh, okay.
gallery.
Everyone just needs to turn their camera on.
Does this make sense?
Yep,
Okay, well, let's try it then.
gather around.
Whoever is online. Whoever is online, please switch on your cameras. We are taking a picture.
We want you in the picture.
Put the shortest person in front of Chris.
You could, like sit on our shoulders here. Well, we can't force you. I think we might need 2 rows. Yeah. I think we can see where you get.
First thing is, everybody has to turn at an angle, so we can try and
Yeah.
squeeze you all in.
Come this way.
No, don't, because you're blocking that window.
Well, I'm not sure what we can do about that.
We'll figure it out. Yeah, well, there you go. Excellent.
Okay.
Get this microphone on. Okay. Nobody's wearing Argon ID. Excellent.
I know, but I got it covered up.
Here we go. Oop. Now I got everybody. Thank you very much.
Thank you. Thank you. So I think we're gonna let's take our break now and then have the final 3 talks. So we're gonna start again at 2:30, 13 minutes from now. I don't think we'll have an hour.
Yeah.
Hey, Natasha, are you listening to me?
Yes.
I'm writing a letter on behalf of Megan Robertson, who I don't know very well, but somebody asked me to write a letter on behalf of her. You think highly of her?
Very much.
Good. That's all I need to know. I'll pull out my extra special adjectives.
No, she's a, yeah, she's always been solid and one of those who doesn't, the only thing I didn't teach her well is how to sell herself.
Nice.
But
I guess we should ask Mark too, but so.
Oh yeah, I mean, in fact, Mark, might have rescued her in that respect.
I don't actually know her. So I think this worries me because, I've been through a period of my life where I thought I knew everybody. And now I think I might be getting too old to know everybody. So.
Well, this is one way to do it. I don't know what Mark would say, but
Yeah.
I have, she's been the same, since she graduated, as far as I can tell.
Good. And I see she went to work after her PhD and then came back with a postdoc.
Right.
Right.
She got, her idea is to, similar to Mark's perhaps, influenced by Mark to make plastics sustainably. And that's, she came to me wanting to do that. at a time when, that I didn't, all I could say is that plastics recycling is a mixing problem, and I know something about thermodynamics of mixing. So that was enough. So I think that, it's a hard problem, the things that she's working on. Yeah, I have respect for her.
Well, thanks for joining us today. We'll be back in about 10 minutes. So.
Sure. Yeah, I look forward to the next two talks. I was actually looking forward to the one right after mine. Yeah. Anyway, thanks, Matt.
Yeah. Yeah, I don't know what happened to that guy, but anyway.
Good to see you, of course.
You're going to be next, Sumit. I'll get people in here, and I'll get people in here in 2 minutes.
You are muted, Sumit. If you said something, we can really hear you.
Yeah. Can you see the presentation?
Yes, I can see it. Not in presentation mode though.
Yeah. 1 second.
That's it. So when we give our presentation, are we sharing from our own laptop or a computer? Is that right? We
Yes.
open our own. Is that what you just did?
Yes.
So, okay.
Yes.
Okay, so our next talk is by Sumit Kawalarami from Northwestern.
Hello, I'll talk about today about using ASAXS to determine the distribution of counterions surrounding highly charged bioconstructs. So all charged objects, be it proteins, DNA, polyelectrolytes, etc., when they are in solution, they are surrounded by counterions that neutralize their charge. These counterions can affect their conformations, their structure, stability, and activity. Here's a simple example of a large linear DNA that condenses into a toroid in the presence of multivalent ions. Similarly, the fibers of actin can condense in the presence of divalent ions. While there are many phenomena that are related to how the ions interact with these charged objects, the exact knowledge of the counterion distribution is lacking. Today, I'll show you 2 examples. One is the case of DNA-coated proteins, and the other case is chiral assemblies of peptide amphiphils. So the work on DNA-coated proteins is done with Professor Merkin, who's a pioneer in DNA nanoparticles and their assemblies and their applications. And the work on chiral assemblies of peptide amphiphil is done in collaboration with Professor Stu. So why are we interested in DNA-coated nanoparticles? So when we functionalize nanoparticles densely with DNA, the properties of DNA are slightly distinct from those of free DNA in solution. These DNA become more resistant to enzymatic degradation. Despite their large negative charge, they can be directly taken up by cells. All these properties are related to how the counterionic cloud on one DNA overlaps with the neighboring DNA. However, there's no proof of that. Now, I'm not going to answer any of those because I don't know the answer. What I'll give you a proof of concept that if we change the nanoparticle core by a weakly scattering object, such as a protein, we can reduce the distribution of counterions in these systems. So the basic idea of ASAXS, you guys are all experts, but basically, as we approach the binding energy of the element of interest, the scattering power decreases because the real part of the dispersion correction goes down. In both the cases, I'll be looking at rubidium. So And the scattered intensity can be divided into two parts, resonant and a cross term. In previous talks, you might have seen a second term, which is quadratic, which is purely resonant. But because of weakly scattering objects in these systems, that term is like less than 1%, so we ignore that. So our system in the first example is a protein, which is CG catalase. It's about 9 by 9 by 7.5 nanometers. It's roughly spherical, and it has floppy single-stranded DNA, 40 of them coating it. And we are studying the distribution of rubidium plus in 50 millimolar salt. So we do the measurements at 4 different energies below the edge. And as you can see here, the scattered intensity is different at the different energies. When you are farthest from the edge, the intensity is the highest because F prime is the lowest. So we analyzed it, and we can extract out the resonant and the cross term. And without doing any analysis, you can see that the minimum in the scattering is at different positions in the two curves. And that's because the ions don't penetrate the core, and their extent is larger than the nanoparticle itself. So this alone tells us that we are sensitive to ionic distribution. Of course, we note another problem with ASAXS, that we cannot extract beyond Q equal to 0.8 nanometer inverse the cross term because the error bars are too high. So it's essentially a low resolution technique in that sense. So we take the resonant and the cross term, and we fit them simultaneously. We find that the linker length, which connects the protein and the DNA, is 4 nanometers, and the DNA is 5 nanometers. And we also find we can also fit the cross term pretty well with the same parameters. And so the picture here shows in the black on the top right shows the distribution profile of rubidium. In the red, it's shown what the profile would have been if each base on the DNA was accounted for by one rubidium within the shell. And since nature doesn't like such sharp gradients, the ion distribution profile is more smooth. So by doing this, we are able to show how much of the charge is compensated within the DNA shell, what's the extent of ion distribution beyond the particle, and what are the lengths of the weakly charged linker and the strongly charged DNA. Of course, to answer any of the questions or the big issues that I had talked about earlier, we'll need to extend these studies to biologically relevant conditions or to the when the nanoparticles are assembled. The second example I'll show is the case of chiral assemblies. And in particular, we are looking at twisted ribbons, or these are bilayers which have twisted because of chirality. So the molecular chirality is being shown at manifested at mesoscopic scale. And it's the assembly of the peptide amphiphyll shown here, which has at the terminus 2 charged groups, which are the glutamic acids. So these ionization of these depends upon the solution acidity. So they will be not charged in acidic conditions, getting charged at high pH. But there's another way to change the charge regulation, and that's by incorporating large organic cations. So when Professor Stoop's group did that, and they got the same qualitatively the morphology of twisted ribbons, but when they tried to grow neurons on these templates, when these large cations were present, the neurons matured and they were well differentiated, and in the absence of them, they aggregated. So the quick question then came that how are these large cations affecting the ion distribution around these membranes? Are these competing with the small cations like rubidium, or are these large cations incorporating in the bilayer, increasing the degree of ionization of the membrane and increasing the rubidium concentration? So that's basically the mathematical model of a twisted ribbon. And I had to steal my daughter's Jenga pieces to illustrate it more clearly. And so she thinks she has made the biggest contribution to this. But we use this simplified digitized model of the twisted ribbon and do ASAXS just as before. What we find is that on the average, the ribbon width is 117 nanometers. The bilayers are crystalline. And in the absence of these large organic cations, we can fit the data pretty well with 50% of the molecule's charge, or one glutamic acid group out of the two is charged per molecule on average. And on the right is the distribution profile of rubidium. Now we tried to do the same thing with the large cations in, and surprisingly, we found that we can fit the data with 50%, even though the ribbon width decreased to 13 nanometers, the tail height of the bilayer reduced. So that doesn't make sense, but it tells that there are some pitfalls to avoid in ASAXS analysis because it's you're getting low resolution data. So there are many parameters that may be able to fit the data that we get. So what we did was, based on the structural parameters, since the tail height is reducing, the ribbon width is also decreasing. That means the electrostatic repulsion has to be stronger because electrostatics wants to stretch out things. So we relaxed these parameters and redid, and we could fit it depending upon the electron density of the tails. We could fit it with 75% of degree of ionization and even higher. So while we can now qualitatively show what's happening is that these large cations are at the interface of head groups and tails, and they are effectively increasing the degree of ionization of these membranes. We don't yet have a complete quantitative picture, as we are missing complementary experiments that can verify, that can pinpoint which of these fittings is the correct one. So what I've shown are two examples of how to determine the counter-ion distribution surrounding charged assemblies and some of the pitfalls. But this is a broad area because the chiral assemblies, for example, they come in various shapes, helices, cochleates, etc. And still, because their assembly structures, such as the pitch length, the diameter, etc., they change with the ionic conditions, but it's not known. There are no scaling laws yet known. So the ASAXS can prove to be a very useful tool in figuring out how ionic conditions is affecting the structure of this. So with that, I'll take any questions. Thank you.
Thank you. Anybody want to ask some questions? I mean, this is an excellent example of a kind of thing that we've been touting as a use of this instrumentation. Right.
Hi. So maybe this isn't an ASAXS question, but you had mentioned that one of the two glutamic acids was charged. Is this a consequence of the fit or the pKa or
so both the things? It's a combination of both. So basically the whole idea of doing these, so you can change the degree of ionization just by changing the pH. The idea was that we want to keep the pH the same, but still change the degree of ionization. So all these measurements are done at neutral pH, around 7.5 in that region, where you would expect that only one or so would be charged. It's not at very high pH. So that answers like why in the absence of the cations, you would get 50% roughly. And to be more precise, from the fitting perspective, we did do a careful analysis of what should be the charge. You can see if we use 30%, the cross-term fitting goes bad. It's lower than the simulated is lower than what the data shows. If we go 90%, it goes higher. In fact, error analysis shows that if you are in the 50 to 60% regime, then you have the best fitting.
So I'm sorry, I have a question, if that's okay.
Yeah, go ahead.
You know, the profiles you show, of course, it's a very, very interesting subject because you have these high contrast ions distributed around the object. But the profiles you showed are pretty similar. You know, is there some cases where you can get violent disagreement between the anomalous and the regular data? Like the ions are on one side, or I mean, some, you know.
Sorry, I didn't fully understand the question because you're saying, so for example, if you look at the previous one, the profile from the dude which has the ion part in it, which is sensitive to the ion distribution, which is in red in bottom right, you can see the minima has been shifted to the left compared to the non-resonant profile.
Yeah.
In the scattering profile. So that's because the ions cannot penetrate the cores and the ions extend beyond the particles, because smaller minima at a smaller Q mean that you are dealing with a larger extension object, which is the larger extent of the ions relative to the size of the object around which you are studying. So these are subtle changes like this. So, but you can differentiate between non-resonant and resonant just by without any analysis also. Am I answering the right question, or I misunderstood?
No, it's you answered it perfectly. Thank you.
What else? Okay, thanks again.
Thank you.
Oops, next week, sorry, I had my thing off. Stephen from O'Brien from the City College of New York, you're on now. Thank you.
Thanks. Yeah, just firing up here. Hold on. Can everybody see my screen okay?
We can see it. We see more than we need to, but it's perfectly visible. We see some of the bar stuff is what I mean to say. So.
Sorry.
You can go ahead this way. It's fine.
Is it fine? Okay, good. All right. Thank you.
Very visible, so don't worry about it.
Okay. Great. Well, thank you for the opportunity to speak. Thank you for the invitation to this meeting today. Thanks to Matt and to Renal. I'm going to give a short talk entitled Complex Oxide Nanocrystals, Nanocrystal Composite Films and Exploring the Potential of ASAXS. I've never used ASAXS before. I have experience with synchrotron work and powder and time-resolved synchrotron stuff, but I haven't done much small angles. So I'm very intrigued by this and very excited about the new instrumental capabilities at the APS. And so I guess I'm still kind of exploring and looking for a fit. And so I thought I would just tell you a little bit about the work that we've done, going back probably about a decade, actually, and summarizing some of that, and then seeing whether or not some of this could apply in the context of ASAXS. And as mentioned, I hail from the City University of New York, which is this large, sprawling urban university with many hundreds of thousands of students and many institutions. But the work that we do is located at the Advanced Science Research Center of CUNY, which was established in 2015. And it's a wonderful building. If you happen to be in New York City, please come and pay us a visit. Okay, so the work that I'm talking about is the design of nanocomposites. And this work was funded by the Department of Energy, the Advanced Research Projects Agency for Energy initially. And what we were looking at is the idea of using capacitors in power electronics. And so, of course, there's been this big interest in designing power electronics to be more efficient, to be faster, and to operate at a wider range of frequencies. And one of the things that they were funding projects in at the time was for LED displays, but of course, power electronics extend to all kinds of renewables as well. So we were looking at LEDs. We published fairly extensively on the topic. sort of on the chemistry and the material side of things and the design of the composites, and also in partnership with our electrical engineers and designers, we published in IEEE a couple of things there too. So if you're interested in looking at our literature, it's also on the links below, or please feel free to email me. So this was for the design of better DC-DC converters, designing new power electronics, and the reasons being cheaper, smaller, more efficient, longer lasting. And certainly the idea of integrating discretes, as they're called, right? You have, you know, the basic discretes, inductors, capacitors,
Maybe we are not
and
seeing
resistors. And so the idea here is, can you embed these more efficiently in circuits? You know, can you actually make prints?
Is that right? Sorry, I guess things aren't going as well as I had led on. Your slides aren't advancing, Stephen.
Oh, I see. I didn't realize. Thanks for telling me.
Right, too.
I'm using a PDF, and I guess that's not working. Is that right?
I guess, yeah.
Okay, so let me just see. I'm glad somebody said something. Let me just get out of presentation mode then and see if that works. Are my slides moving now?
No, we're just seeing you now.
Oh, okay. All right, well, my lesson is learned. I shouldn't use Adobe, but I had a keynote presentation that was gigantic, and then I converted it to a PDF. How about now? If I advance, does the people see that now?
So pick it up, you know, I think maybe on this slide, if you don't mind. We're not pressed for time. I don't think we're going to, we only have one more talk.
Yeah, sorry, but are you able to see my slides advancing now?
Yes.
Okay, great. Sorry, I didn't hear that. Okay, thanks. Sorry about that. So yes, I was just talking about the work that we've done, and the link is on the bottom of this page. There's a number of publications in materials chemistry journals and also in IEEE type journals about using printed thin film capacitors for power conversion. Okay, and the sponsored project was better DC-DC converters, and I was talking about the kind of general goals of designing new, more efficient power electronics and having them be more integrated, either with a sort of novel device design or on chips, you know, on ICs, actually making an entire integrated circuit that is a power electronic chip. And of course, that has a lot of relevance for today with the CHIPS Act and where the US is going and what it's looking to put funding into. So of course, and this is about otherwise having discretes, you know, that you would have a discrete, you know, discretes. I'm referring to inductors, capacitors, and resistors. And those are things that you have to normally add to the circuit later. They're not that easy to add within integrated design, although there is a number of different technologies out there that try to do so. So we were attempting and continue to attempt to work in that space. And so the idea here was that we could imagine using a nanoparticle-based film, and that we would try and rely on high dielectric nanoparticles and synthesize them, and then make multi-layer printed thin film capacitors in order to increase the energy density, and have those be integrated in a hybrid switched capacitor resonance circuit with the kind of design that you can see there. And the test model was really for testing and powering LEDs. So the material selection process that we went through was that we were looking at barium titanate and analogs because it has very high dielectric constant in the bulk, they're ferroelectric. And then there's been a lot of research and physics surrounding what happens when you make these things nano, and if they retain their remnant polarization, or at least retain enough dielectric moment and polarization to make them useful in capacitor technology of this kind. And so we settled on barium strontium titanate, and we developed a kind of a sol gel process for synthesizing them. And then you can see on the right hand side that you can generate films of this kind of thicknesses on the orders of 50 to 100 nanometers. And it's essentially a self-assembled nanoparticle film, in the sense that it's kind of a sedimentation process, evaporative driven assembly of discrete nanoparticles. And the advantage of doing this was at the time to consider the parameters that you would need for a circuit that would work in power electronics. And one of them is high frequency, low loss. So if you can operate at very high frequencies, but with low losses, that would be a useful type of power electronic device. And nanoparticles lend themselves very well to that idea, because the switching of the polarization can be very, very fast. And so the loss is very low. So actually, this might be an instance where you don't want remnant polarization. You just want a high dielectric material. But of course, when you're talking about making nanoparticle films into capacitors or into other devices, you run into a lot of problems. And so there's a lot of literature out there that you'll find about making nanoparticle films, which are usually composed of polymers and nanoparticles. And the nanoparticles have their features, namely, for example, in this case, dielectric constant, but there could be other properties that you're wishing to exploit. And then the polymer usually serves to be the medium. And of course, the idea is that if you have a polymer with a low K, but it's a good medium, and then you have a particle with a high K, then you can combine the two and get the best of both worlds. But unfortunately, in the world of nanocomposites, it's very often the case in properties, for magnetics, dielectrics, and these other things, that you kind of get the worst of both worlds. And this is something that the community has long since been trying to address in order to improve. And this is an example of that, in the sense that on the left-hand side, you just have a nanoparticle film, which does show a high dielectric constant, a low frequency, but this is due to space charge effects. It's not really due to the intrinsics of the nanoparticle. And what you then need to do is make it a nanocomposite film, and then you get a lower dielectric constant, but you get a much more stable, low loss dielectric constant, which is what you actually need for electrical circuits. And so that puts you at a K, in this case, for these particles of about 20, and a loss, a tan delta around about 0.05. And of course, then you have to worry about actually making a device. And this is a case where, good contacts versus bad contacts plays a big role in the actual ability to make something that is a capacitor. So a capacitor is a very seemingly simple device. It's just some things sandwiched between, or even nothing potentially sandwiched between two metal plates. But when you start to design capacitors of this kind, especially if you're thinking of using them for printing and integrated with circuits, it quickly becomes a complex problem with a number of parameters that you're trying to optimize around. And as I said before, there's a large community that explores making dielectric nanocomposites. We tried our hand in a few areas. For example, the one that I just showed, which was barium strontium titanate and polyfluoryl alcohol, which was one of our sort of optimized and preferred materials. But there are also lots of other examples. And then there comes this question about, well, what are you doing with the polymer and the particle? How are you mixing them? And how is the interface actually working out? Are you just mixing them mechanically and then making materials of that kind? Or are you in fact chemically bonding them together? And what we have learned through our research is that covalently bonding the polymer to the particles and making a three-dimensional network is much more effective for generating low loss capacitors. And that's illustrated on the bottom there with some of these pictures. But basically, what we have found is that you can generate nanocomposites, but they don't necessarily make good capacitors. They can be very lossy and very poor in quality. And there's a large bulk of research that is endeavored in this regard. When you're actually calculating the effective medium approximation, there are a number of basic equations that you can use. Based on the set of equations that are derived for this. And what you do learn is that quickly the packing density matters a great deal, that the dielectric constant of the filler may actually not affect the dielectric constant of the film. So once again, you're left with this idea that making the nanocomposite and making a pure, well-integrated nanocomposite is very important much more important than, say, just trying to optimize the dielectric constant of one of the two components. And so making a fully integrated mixture of the components becomes very much the goal. But if you can do that, there are some interesting things that you can do. For example, in collaboration with Dan Steingart, who's now at the University of Columbia, we were able to use a spray coating facility and make a multi-layered capacitor, something that you could consider doing in an integrated fashion. And we were able to demonstrate in this case that mean capacitance can increase with the number of layers. And so this is like the BST PFE capacitor material. And then we went on to demonstrate those multi-layer capacitors can function in devices that you can. I guess I won't go to the display, but these are actually movies, but they're embedded movies. But I'm not gonna show them. But you can see the movies on the website. But basically, we're able to show at the final stages of the project that we did with ARPA-E the ability to use these kinds of capacitors in electrical circuits. Now, just moving on a little bit, because dielectrics is one form, but there are more exotic or more interesting films that people can consider and are considering making. So multiferroics has also taken off in the past few years as an important field of inquiry. And that's where you have the combination of magnetization and polarization within one material, different forms of another. One way to do it is to actually combine elements in one material in order to generate a multiferroic material. Multiferroic meaning that it has one or more, that it has two or more order parameters, such as ferromagnetism and ferroelectricity. But one other way to do this is to make nanocomposites. And so again, this field is looking towards the materials community for the design of nanocomposites that could be effectively used. And there are also demonstrated devices such as the one from Ian Young's group from Intel, in which they did demonstrate a scalable magnetoelectric spin orbit logic device a few years ago. To that end, then we also look at making nanomaterials and nanocomposites that are multiferroic, and we tried our hand with nanoscale perovskites of europium barium titanate, looking at the phase space and understanding how to synthesize it, and then trying to make nanoparticles of this material, which are not completely uniform, but we were able to do so. And we also generated some interesting novel core shell structures here, for example, in this case, that I'll spare you the details, but we were quite fascinated by some of the materials that we made there. So what can ASAXS do for us? I think I want to just move to speak about that. So in my survey of the literature, going back several years, I looked at different things. And I was quite interested with some of the work that people had done on things like supercapacitors. And this is a case of a carbon manganese oxide structure, where they're able to analyze the size of the particles and then thicknesses of different layers, and then looking at the assembly of these microstructures and correlating that to atomic scattering factors of manganese, and looking at the structure factors and so on with samples loaded with different weights of manganese dioxide. And so I thought that this could also be something relevant to what we're doing here, that if ASAXS can reveal information along these lines for these kinds of materials, then this would apply well for what we're doing. And also in general, for the field of nanocomposites, that if you are making a polymer nanocrystal material, that these things they're not that well ordered. They have a fair amount of disorder in the structure, but you might be able to self-assemble the particles into some kind of level of ordering, and then you're considering backfilling with a monomer and making the nanocomposite. And it would be nice to be able to analyze this polymer inorganic matrix more effectively than just looking at it by scanning electron microscopy. And it seems to me that ASAXS could be a very useful tool for doing this as well. And so we would be excited to try to explore this further. And certainly because ASAXS has this contrast given the different kinds of elements present and doing cross sections where the elements are lying within the material, then that would seem to be something that would apply well to what we're doing. Okay, so that's the end of my talk. Sorry about the the earlier problems with the presentation. There's a lot of people to thank in the background for some of the work that we did as part of the ARPA-E project, and also my group who worked on the dielectrics, Fred and Julian and Nassim, who worked on the dielectric and multiferroic nanoparticles, and thanking also the NSF who continued our support after we completed our ARPA-E project sponsored by the DOE. Thank you very much.
Thank you, Stephen. We can. Sorry. I have to clap first. Questions for Stephen? I'm guessing this isn't a technical question, but part of this has to do with providing ammunition for our NSF program monitors. What were the programs that supported your work at NSF, Stephen, if you don't mind me asking?
Sure, the DMR, ceramics. So they funded us, and we also had an interdisciplinary research, an IDR with civil and mechanical.
Great, great.
And in the case of the civil mechanical one, the partner Jackie Lee, who was also at City College in the engineering school, she was doing modeling of the nanocomposites as we were making them.
Thanks.
And certainly I would be very happy to provide, any further support. I probably need a bit, I think I need a bit better education in ASAXS, but maybe some conversations could help move that forward. And, I'd be very happy to provide you with some more information if it would help the ChemMatCARS course.
Yeah, obviously this is ChemMatCARS, but as you heard from John Papanikolaou, chemistry is carrying the ball.
Yeah.
They need partners within NSF to do what they'd like to do to help support this instrumentation. So,
right.
Thank you.
Thanks.
Last but certainly not least is Caitlin Landy from Northwestern.
Okay, great. Hello, everyone. My name is Katie Landy, and I'm a fifth-year PhD candidate at Northwestern University. And I'm really excited to be here today on behalf of my advisor, Professor Chad Merkin. And what I'm going to tell you a little bit about today is how we can use DNA to program multi-component colloidal crystal architectures. And so in the Merkin group in general, we're really interested in precisely controlling architecture on the nanoscale. And one of the major approaches we use to do this is to repurpose DNA from its genetic function as the blueprint for life to a blueprint and a construction material to assemble larger structures out of nanoscale building blocks. And so our fundamental building block in this approach is a nanoparticle core densely functionalized with a shell, a dense shell of oligonucleotide ligands. And so we can independently design the DNA with respect to the nanoparticle core in terms of composition, nanoparticle size, shape, and so on. And so how this works is that our DNA functionalized nanoparticles contain sticky end, short sticky end sequences that can interact through specific DNA hybridization events with strands on neighboring nanoparticles. And as the system seeks to maximize the number of these DNA hybridization events, we can assemble really highly ordered colloidal crystals with lattice symmetries, interparticle spacings as defined by the DNA design, and in many cases, faceted single crystals on the microscale. And so overall, this approach has allowed our group and others to establish routes to stimuli responsive lattices using some different DNA motifs, as well as 78 different lattice symmetries to date, and 8 different crystal habits. So we've really been able to explore a wide design space with this approach. And so today, what I want to talk about is multi-component crystals. And so I won't show any ASAXS data in this presentation today. I will show a lot of ASAXS data, but I think multi-component colloidal crystals are potentially a really interesting system for further study with ASAXS. And so I'm going to give a brief summary of 3 recent directions in our research, which includes making colloidal analogs of substitutional alloys, core shell structures, and intermetallic lattices where these colloidal crystals have nanoparticle building blocks and nanoscale periodicity compared to atomic systems. And so the first approach that I wanted to talk about today is programming substitution within colloidal crystals using the DNA design. So previously, it's been known that if we combine nanoparticles with complementary DNA, they'll form a cesium chloride type lattice. And so if we imagine a system where we simply use 2 different nanoparticles with the same total size DNA design, and so on, in this case, a gold and an iron oxide nanoparticle core, we know that these will form a cesium chloride lattice. But what we hypothesized in this work was that if we were instead to combine a equal ratio of each type of nanoparticle composition with each type of complementary DNA shell, it should be equally likely for either composition to occupy a given lattice site, resulting in a random arrangement within the overall same parent lattice symmetry and crystal habit. And so we tested this experimentally with gold and iron oxide nanoparticles, and we can see from scanning electron microscopy images of these crystals that we see the same type of crystal habit. This is a rhombic dodecahedron shape, which is what we expect for this parent lattice symmetry. And then we can start to distinguish these 2 types of substitution based on the ASAXS data. And so for the cesium chloride lattice, where we have an ordered arrangement of the gold and iron oxide within the lattice, we do see peaks in the ASAXS trace that correspond to a cesium chloride structure. And we've simulated this with tools from Byongdu. And so it matches pretty well with the peak positions for the expected cesium chloride lattice. Whereas in comparison, you can see for the random structure that we see a BCC lattice. And this corresponds well to simulations where we used a 50% site occupancy ratio of each of those compositions in each lattice site. And this also gives us some indication that the 50-50 ratio that we intended to incorporate was incorporated, as if one of those lattice sites had a slightly off ratio, we should start to see the appearance of the 100 peak in that trace as well. And so ASAXS gives us a really nice global measurement to look at these differences in types of substitution. But to look at this on a more local scale, we turned to energy dispersive X-ray spectroscopy. And so in this initial experiment, we were mapping whole crystals, which are embedded in a silica matrix, and we observed signal throughout the whole crystals of both gold and iron. So there's no indication that we have any phase separation in either of these cases with ordered or random substitution. And then furthermore, we can also embed these crystals in resin and then thinly section them to do these EDS mapping experiments to probe more local order. And so with these EDS maps of sectioned crystals, we also see the ordered lattice and random substitution that we were expecting based on the DNA design. And so I'll move to the next topic, which I mentioned for that previous system, the crystal habit here is a rhombic dodecahedron shape. But one aspect of how we actually assemble these crystals is kind of important in terms of the size and uniformity of the resulting crystals. And so typically how we actually assemble these crystals is we'll combine the DNA functionalized nanoparticles, DNA linkers in a salt solution and slowly cool it over the course of 2 to 3 days from about 55 C to room temperature. And what this results in is these faceted crystals. But you can see on the left here, we get a pretty wide range in sizes. We have some crystals that are relatively large. And then if we were to go to a higher magnification, these smaller ones are still faceted, but there's a pretty wide range in sizes. And so what we were interested in this project was to use this DNA design to try and overcome this limitation by programming separate nucleation and growth steps. And so we think that this kind of size polydispersity arises because nucleation and growth are not well separated typically in our process. And so what we did here was we designed the initial complementary DNA sticky end sequence, which is pretty much the same from the previous project. But then we designed a second weaker interaction that's identical except for a single base mismatch. And so the hypothesis was that this particles functionalized with the stronger DNA sequence would be able to nucleate first, followed by heterogeneous growth of the nanoparticles functionalized with this weaker growth DNA sequence. And so to test this, we can do in situ ASAXS measurements where we're cooling the solution from 45 to about room temperature over the course of about 3 hours. So this is a bit faster than our normal crystallization process, but we're able to compare for these different cases where we have the different DNA interaction strengths. And so what we see in the structure factor versus the temperature as we cool the solution is that for this case where we have the stronger DNA interaction, we see BCC peaks start to appear at a much higher temperature than if we have this weaker DNA interaction that has the single base mismatch. And so we can also then combine all of these DNA functionalized nanoparticles in this process and look at kind of this two-stage assembly process in situ ASAXS. And so we do see an early onset of peaks and then increase in intensity. And we have about 1 nanometer difference in lattice parameter between the two. So we also see some effects of that. But one analysis that we did just to kind of compare these temperatures in a way that's a little bit easier to compare is we took the normalized peak intensity of the 110 principal peak. And that way we can compare kind of the temperature at which these transitions are occurring. So if we have only the stronger DNA sequence, we see a higher temperature transition. And then if we have only the weaker DNA sequence, we have a lower temperature increase in this peak intensity. And then for the system where we have this kind of two-step process, we do see an initial increase in the peak intensity, followed by a slow increase at higher temperatures than we would otherwise expect to see it. And then finally, I'll just briefly mention that we can also use nanoparticles with different compositions to apply this approach as essentially a one-pot synthesis of core shell colloidal crystals, which have been made before, but typically require some post-synthetic stabilization of the core before you can grow a shell. So this is a bit of a simpler approach to make these types of architectures. And that way, with different nanoparticle compositions, we can also use that as a handle for characterization, where we can, in this case, since we used gold and silver nanoparticles, we can look at different wavelengths for the LSPR peak in a UV-Vis melting experiment as we're changing the temperature in the solution. And then again, we can use EDS mapping to confirm this type of core shell architecture in these crystals. And then finally, I'll really briefly mention one other recent direction in our lab that we've been thinking about using DNA, more complex DNA architectures to also design more complex lattice symmetries, hopefully, in these types of crystals as well. And so here, instead of just DNA functionalized nanoparticles, we also have these branched DNA dendron structures with orthogonal DNA sequences on the two different colors. And then we can either hybridize them together with just duplex double-stranded DNA, or we can use a three-way DNA junction to attach multiple of these together. And then one of each of these dendrons can interact with a different DNA functionalized nanoparticle. And so there were a few interesting things that came out of using this new type of architecture. One of them being that in these structures, we observed a simple hexagonal type of lattice symmetry with respect to the DNA functionalized nanoparticles. We aren't able to see the location of these DNA dendron junctions, since the scattering cross-section is so much lower than gold or whatever metal nanoparticles we're using. But one interesting thing we can do with this type of design is extend the DNA length in between this junction. And so doing this allows us to increase the lattice parameter along a single crystallographic direction. So we have the same type of symmetry, but we've just increased the parameter along the C direction. And then finally, we can also use different nanoparticle compositions in this approach as well, where in this case, we used gold and silver again. And in the same type of simple hexagonal structure, we're able to see this confirm this type of layered structure of gold and silver nanoparticles and in the EBS mapping as well. So to conclude, using DNA, we've been able to access a couple of different types of multi-component crystal architectures, including analogs of random solid solutions, core shell crystals, and intermetallic lattices as well. And throughout this talk, I've shown a combination of ASAXS characterization with electron microscopy and EDS mapping to really characterize the location of different nanoparticle components in these structures. So this is where I think ASAXS would be potentially really useful to give us more information with a ASAXS experiment versus the type of EDS mapping that's a very local, time-consuming technique. So I think that's a potentially really exciting direction. So with that, I'll just acknowledge my advisor, some of our collaborators, research and administrative staff, as well as the funding sources. This is funded by the AFOSR. And with that, I'm happy to take any questions.
Thank you.
Yeah, okay, so I have one question regarding the polydispersity of the particles itself. So in general, the nanoparticles, they do have some polydispersity inherently, right?
They do, yeah.
So is there any dependence on this polydispersity to the overall, supercrystal size?
Yeah, so we do need to have the polydispersity below a certain level in order to get these highly crystalline structures out. The number is about 12%. CV is reasonable to use in our approach. So it has a little bit of a higher tolerance for polydispersity than some other nanoparticle assembly methods, just since the DNA can kind of correct for some dispersity, but that's kind of the number that, as long as we're below that, it varies a little bit for the different compositions, what it actually is, but below that threshold, we can get pretty crystalline materials out.
Yeah, one more question. So in your page number 8, I think there is something like I saw the peak changing with temperature, the peak width changing with temperature, the first peak, here.
Yeah.
So like at 23 degrees Celsius, the first peak becomes very sharp and it becomes very narrow.
Yeah.
And in the other higher temperature, these are broader and maybe two peaks together. So is there any kind of crystal transition happening here? And yeah, the structure is different, right?
Yeah, so what I think is happening in this is that we do have a slight lattice parameter difference between the fully complementary DNA design and the one with a single base mismatch, which we attribute to just slightly increasing the distance of single-stranded DNA versus double-stranded. So it's a relatively small difference, but as this second peak starts to come in, that's sort of what we're attributing it to it. I think also in the first few layers of the growth of the second set of particles, there's probably some strain. And so as we incorporate a larger amount of the second set of crystals, I think that's probably just washed out, as my impression. These also, we weren't super quantitative with this because obviously we're doing this in solution in a capillary tube, so it's hard to quantify how many crystals and what area of the capillary you're shooting. So yeah.
Okay, thank you.
Yeah.
I have my question actually, but the maybe related question is that typically when heterogeneous nucleation is faster than the heterogeneous nucleation growth probably is a little faster than the homogeneous case, right? So do you see though in general these has a faster growth?
Yeah, so one thing that we see in this plot especially is that that we thought was interesting was we do see this increase in peak intensity at much higher temperatures than we do for just the control with the weaker DNA sequence. So I think that was a good indicator that we had heterogeneous growth. The overall time I didn't score, but that's an interesting point.
So maybe going to the same issue, but maybe being able to help with the strain problem. Have you ever tried to take advantage of the fact that in the growth DNA, there is a temperature shift, so you can do like a temperature jump to nucleate and then go back a little higher to anneal?
Yeah. So I didn't show that here, but we did end up changing the temperature profile somewhat. So how we ended up doing that was we did kind of our traditional slow cooling rate through the first transition, and then we added a 10 to 12 hour just annealing step.
Yeah.
And then slow cooled it the rest of the way. And we tested a few, I think 8 different temperatures, kind of in the range between the two melting temperatures and slightly below the lower one. So we did kind of look at that, and we did see improvements for, we saw improvements for the crystal uniformity also when we added that annealing step, where we're hoping to bias it towards the initial nucleation and then heterogeneous growth without any subsequent nucleation events. So
thanks.
Yeah.
Yeah, thank you. So it seems like you have done these things with the similar size kind of particles. Is it possible to do two different sizes?
Yeah, absolutely. Yeah. So a lot of the projects that I've been working on specifically have used the same size particles. They're sometimes depending on the total size ratio, so incorporating the core plus the DNA shell. If you go off a 1-to-1 ratio, you would get a different lattice symmetry, but you can use the DNA design length to correct for that. So you absolutely can use different sizes. I think in this project, I didn't show it here, but in addition to using gold and silver, we also used 2 different sizes of gold, like a 10 nanometer diameter and a 5 nanometer diameter, to also kind of confirm this layered structure. So yeah, we absolutely can do different ratios. And other projects in the group are also interested in co-assembly of different shapes. So anisotropic shapes with spheres or multiple anisotropic shapes. So yeah.
Thank you.
Anything more for Caitlin? Yeah, sure.
Is the having the weaker strength of DNA is, particles around, does it help to limit the growth of the first component? So I'm guessing that probably it's like it's a more nucleation dominated kind of system you kind of made, so that way you were able to make a more homogeneous size of the supercrystals, right?
Yeah.
So probably the growth was limited by the falling particles around, I guess.
Yeah. Yeah.
Okay.
Yeah. I think so.
Okay.
I think you can sit down, and we obviously left a lot of time for general discussion. I don't have any particular points. I mean, we've recorded and discussed a lot as things went on. But does anyone here want to raise a general point? I might ask also, Benoit, I'm not trying to put you on the spot, but you've been with ChemMatCARS the longest of anyone in this room. So are there things you think we ought to be thinking about as we prepare for our renewal vis-a-vis the subject of today's workshop? It's okay. I mean, I really wasn't trying to put you on the spot. I'm just trying to figure out if there's any more substance that we need to discuss in this last hour or so.
Matt, this is Cecilia here.
Yeah, go ahead.
It's more a general question. I just wonder how does, the renewal and these ASAXS capabilities couple with the fact that Argonne is going to be closed for a year or so.
That was in fact a bigger issue with the last renewal, because we're shut down now in this grant period. By the time the renewal gets funded, we will be coming back up. So I don't think that is in particular a big issue right now, Cecilia.
Okay.
We'll be coming back up in a different with a whole different capability. We'll have a second beam line. With respect to ASAXS, we'll have twice as much allocation of time. The beam itself will be different, more intense and more coherent. So I think there's a lot of pluses about the upgrade, and they knew that when they funded in the last renewal the construction of a second beam line, that was likely to take place during the shutdown, which is what is happening right now. So I think we're okay on that score.
Good. Yeah, I mean, we'll definitely be looking to use it. So I'm excited. So hope it gets renewed.
Yeah, I think the chances of it getting renewed are good. I think, we really, because of the second beam line, we're asking for a large increase in the operating expenditures. And, that's honestly a heavy lift for chemistry. I think those are the exact words that John Papanikolaou used. But you heard him. I mean, they appreciate and are proud of this as really right now their only ongoing facility in chemistry. So we have to do our job and make a good case and develop the community as best we can, and I think we'll get as much support as they're able to give us.
Thanks, Matt.
Say something, Bruno?
Yeah, so I think this is open in discussion anyway. So I think Morgan she was saying she want to say something about the
I just more wanted to ask kind of the actual details of how to use the instrumentation, more of things to apply it towards, because we've seen a lot of the chemistry specifically with ASAXS, and I know some of what my group works on is exactly in that size range that might be a little too low for necessarily getting ASAXS level resolution. So I kind of want to know more details of the spatial resolution and what concentrations we can work with to kind of see if it can fit what maybe we do at Georgetown a little more, if you could speak to that
You asked for it, Myrna.
Okay. Yeah, that's fine. Yeah, certainly. So in general, means when we do it, when we discuss experiments with our users, we ask these things like from the beginning, like what kind of system you're doing, what kind of size you're getting. So depending upon that, we tweak the experiment in our beamline. So right now, as you have seen, we have some discrete ways of doing getting the Q range, and that decides the size of the system you want to probe. But that will be much improved in the upgraded version, where we can have like simultaneous ASAXS where the Q range will be much broader. So that can definitely address this issue. And apart from that thing, we still have some limitations in terms of size, like the maximum and the minimum. So minimum, we cannot go beyond like few angstroms, like 5-6 angstroms, we cannot go below that. And maximum, we can go up to, let's say, 150 nanometers, something like that. So that's the range you have. And concentration wise, yeah, this is a tricky question. Yeah, I cannot answer it until I know which kind of material you're going to use, because different material have different kind of contrast. And also the contrast coming out of ASAXS is different depending upon which edge you are doing. So there are a lot of things there which needs to be kind of clear out when we discuss with each other before experiment. Yeah, thank you.
Stuart.
So thanks. Sorry I had to leave. I had a meeting on campus. The nice thing about being close is I could drive there, but the bad thing about being close is I couldn't get out of some meetings. So I apologize for having to leave and halfway through. But the question I wanted to ask, Matt, was maybe sort of building on a question you'd asked Stephen earlier, was does it make a difference if or does it help you out in any way, should I say, if the research that's maybe being proposed has already been funded by NSF Chem? So if chemistry is funding a certain class of materials in your lab, does that help you out, or does it not make a difference?
Well, I'd say generally, yes, that's a good thing. The question is, how do we use that information, or how does NSF become aware of that information?
Mm-hmm.
And, I guess it might be of some interest, and we could do this as a follow-up to ask who among you is supported by what programs in NSF. Certainly chemistry is most concerned of supporting the chemistry community, but they also want help and partnership. Mainly they hope and expect to get it from the DMR and from chemical engineering, especially CBET, or from engineering, I should say, especially CBET, chemical, biological, environmental, and thermal, or whatever CBET stands for. Please. You got it.
I just wanted to add, maybe the contrary is also true. If we have a new idea that exploits ASAXS, maybe, the connection to this beamline could help get that funded. I mean, of course, the proposal has to review well, but.
Yeah, I think that's absolutely true.
Could I add something to
Of
that?
course.
Because you were asking me. That's absolutely true. So one of the things, we're running several workshops, and one of the things we would like to get your contribution to helping our proposal is to write a few paragraphs of your research, what you're doing, and what you think ASAXS can help you, or you hope you present some ideas, or you think the ASAXS can develop in the new directions and they can help you, and we'll put in our proposal. And that can really strengthen the proposals, because our proposal is not just to ask money for building the instrument, but also present a science case, which is a user science. And we have been doing that, and it's a very successful, collect a lot of users at different areas. And
Yeah, and what's new about what Ben was saying just now, I should have been a little quicker. We've already asked users for that kind of, and
Right.
people have actually already used the instrument, but we could ask some of you who haven't, and we don't want the writing to be a terrific burden, but I'm sure it would help if any of you have gotten ideas. And you know, many of you started off your talk that, you know, we haven't done ASAXS or, you know.
Good. That's
Well,
good, right?
no, that's perfectly okay. We're trying to enlarge the awareness into other areas, but so, you know, I think we should only ask you to do this, or you should only respond to our ask if you really do see a way that you can use it. We're not just trying to bulk up the proposal.
I just wanted to change the subject a little bit,
Sure.
but the speaker before me, and then we talked about it at lunch a little bit. This is NSF, education's important. Currently, Renal is, he is processing a lot of people's data because we don't know how to do it. So I think educating users how to process their own data, workshops, you know, maybe student internships would be really great for expanding the user community. Right, because you got to understand and know how to use the technique in order to be successful with it.
Yeah, I mean, to tell you the truth, instrumentally, this is not a gigantic innovation. You just have a ASAXS machine that you tune the X-ray energy.
Right, but the data processing is very
processing
tricky.
is.
Yeah.
And Mirnal's one of the world's experts in
Yep.
that.
The world.
We should relieve him of that title by teaching everybody how to do it. It's a very good idea. Thank you.
I think we are kind of organizing like works sometimes of like not workshop, like schools, which is part of sometimes organs, you know, they do like NX neutron X-ray school. So all those kind of schools, we are participating there, and sometimes we do it ourselves, but I haven't done specifically ASAXS, you know, even workshop through ChemMatCARS yet, but I'm planning to do it in this kind of, you know, as we are going in this start period. So I think it is a good time to start this kind of interactions with the, you know, existing user and with the, you know, prospective user, I will say. Yeah, in that sense, it will be useful, I guess. So more discussions and yeah, discussing with students mainly, where they will do the real analysis there. So main reason is like when I developed this software, the main reason is this, that we will give this software to user and they will do it, but still there is some training lacking there, and I agree with that we still need to do some kind of either through YouTube or documentation to make this thing much more you know, transferable. Yeah, I need to work on that.
YouTubes are very helpful.
Yes.
I mean, students really like that.
Yeah.
Yeah. I have another question. In seeing when people presented ASAXS data and, you know, our own data, it often looks very noisy. And I don't know if that's just the resonant component, because I don't know that much about processing the data. But, you know, like some of the fits that I see, I think, oh, that could be something different. So is there, is this inherent to the process, or is there a reason for this, or?
Yeah, I mean, it's resonant term, as you can see, like in many of the talks. And the thing is, resonant term is some sort of, you know, in order of magnitude, it is like 5 or 6 order of magnitude less.
Mm-hmm.
So it's very tricky to get that at the first place. That's why the beamline hardware needs to be, you know, and on top of that, the contrast is also important. Like if you really start doing something like polymers, which doesn't give you much contrast, even the ASAXS itself is difficult. Then you cannot get good ASAXS data on that. So yeah, it's a balance between contrast as well as the number of scatterers you are in the system. Like Daniel was mentioning in his system, that's the, I think, more you know, noisy data we see today. So the reason is the contrast is less, and he has also, because his main intention is to look at the interface. So interface, you can see a lot of particles there, but at the bulk, the concentration is much lower. So in order to use the same system to look at, he needs to raise the concentration. But whenever you raise the concentration, it comes to the limit that it can aggregate. So again, it will be a different kind of system. So yeah, it's a tricky balance between what you want to look at, and sometimes the contrast is too low to look at, so you cannot use that. Yeah, there is a limit on that as well. But the kind of system which Tianbo is looking at, it gives you huge contrast to start with. So those are the systems where you really see good ASAXS data in that sense. Like resonant term is very, reliable. But extraction wise, there are two ways you can do that. You can directly analyze the SAXS data which you get as a function of energy, and then the other approach is to split it, like we saw, like SAXS term, cross term, and resonant term. So this splitting has some inherent, how to say this, some inherent noise inbuilt, because your data itself has a noise which is due to the statistical noise you get it from the data itself. And then on top of that, you're assuming something about the system. Like, your system is made up of your resonant element and the other elements, which doesn't change. But sometimes that's not true. They might change a little bit, which might contribute here. But we are not considering that when you are splitting this. So in those cases, it might give you some artifacts. So you have to be careful about that. So the way I suggest my users to do it is to first try and simulate the system and see if this extra, splitting things works properly. Because in simulation also you will see that, like what Xiaobing was showing, like after doing simulation, when you split the things, you might not see the result which is actually there. So yeah, there are several ways you can check and balance to see how you can improve this analysis.
Can you change the X-ray energy in SolEx?
It's already there.
It's already there.
Yeah.
Okay.
So you
I
use
haven't, I use SolEx like all the time, but I haven't seen that feature because we've never needed it, but I'll look for it. Great.
Yeah.
I mean, building on that, especially for people who are not experienced or have never used ASAXS before, maybe it does make sense in terms of just good grantsmanship to create, a way, not just for ASAXS, but maybe some of the other tools at ChemMatCARS, to try a sample, because you don't want to allot like a day of beam time if you can figure out if there's enough signal to noise, in 2 hours or 5 hours, right? And it doesn't also make sense necessarily to carve up beam time. So maybe there's in terms of convincing folks that, you are a more general user facility where you can set up a mechanism whereby folks can try samples and see if signal to noise is worth, continuing, or at least maybe then modifying samples to make it, a little more effective for ASAXS experiments.
We have to figure out how that works with the overall system, beam time allocation. It certainly can be done opportunistically, but I don't know. Can you see a way that could actually be done in a formal kind of way?
Well, it's more like a mailed-in service almost, right? You mail the sample in and test out. I think from our experience over the last 3 years, we've done all of that test. It depends on the samples. Murnau had that experience. If Murnau has to prepare the sample on site, chances are, yes, it doesn't work out. With your group, that's a good example. because the users just don't believe that we get the sample right. But if you have samples that's solid or things it's already sealed, ready to go, that chance is a successful chance is very high, right? Murnau,
Yeah.
think of that. I've seen him struggling, and then Louis says, Enke, I think he says, are you sure you did this? So it really depends, but I think it's a good idea to try out before you struggle and have a full run coming here.
Yeah, I think it is. This kind of approach is implemented already in the crystallography, field, where people send their samples and quickly they screen things. Like in protein crystallography also, they do the same. They send like hundreds of crystal, you screen them quickly and then select whichever you want to take the full set of data. But it is tricky in the sense that, samples are not like crystals. So if they are like crystals, yes, we can do that. Like as Minwa mentioned, the solid samples, which you just put it in a capillary tube and do it. But there, on top of that, there is another problem, like, this kind of samples, depending upon your sample itself, it can have a lot of absorption. So then I have to think about how can I, reduce the thickness, and that brings you more, complications on top of that. So so far there is no ready-made solution as such, but I am feeling like, my feeling is whenever I talk about this microfluidics, this has some, chance of answering this kind of questions where we can do a different test in a single chip. But I'm not sure, we haven't tested it yet. ASAXS people have done, but ASAXS, no, we have to check how to do that.
Well, we've heard some very good pieces of work today, and people have really put themselves out to get here. Not so much those of us from Chicago, but those of us from the West Coast and around the country, the East Coast, and we really appreciate it. We may ask some of you to consider formulating a paragraph that we could use in the proposal, if you really, if you can, when we ask you, but also if you think you have a real story about how ASAXS would benefit your work. So we don't need to stretch, but we do need to collect every legitimate story like that. So I couldn't be more grateful to the people who came here today.
Can I ask you about the scope of the, I guess, the proposal or the instruments wise? Are you gonna expand the energy range of the instruments or?
No, we are at present, we are not, we are keeping the energy range fixed. We are not changing the line, we are building two beam lines, right? Not two, we'll have one top of another. So the existing beam line stays and it will be the same energy which we are using. So ASAXS is part of it. So no, we are not changing the energy range. But the other new beam line, we don't have that broader energy range anyway. So, but we might go to the lower energy range, probably like 3 or 3.5 keV. So 3.5 keV, yeah. ASAXS is not going to be in, yeah, it's not there.
Are you going to change the detectors for detector?
Yeah, we are planning to. We have the silicon one, so we want to add one more to get the whole range of energies. Yeah.
I have a quick question. So as you go for new capabilities such as the flow cell capability, do you expect to actually collaborate with a research group to test samples together, or you would rather just have the capability ready by yourself and then bring it to potential users?
Yeah, I think we are open to this kind of collaborations, because so far that's the way I did it. Like whatever development we have done, we have collaborated with a group and we developed it and we kind of kept it in the kind of beamline inventory that we can do those kind of stuff. Yeah, definitely. This will be really useful for us as well.
Well, I mainly want to say thank you again and let you go home and get about your business. And we couldn't be more grateful for having you here. Thank you, Cecilia, and anybody else who's still online. Bye.
Bye, Matt. Bye, everybody. Thanks for having me.
Thanks, Stuart.
Thank you. Bye-bye.
But not even that, I think about a year. So.
You okay? Yeah, I was really falling asleep that last time. Not